Welcome to the 2026 LGMD Scientific Summit. I'm Bryant Knudson, the founder and CEO of the Speak Foundation. On behalf of the Speak Foundation, thank you for joining us today. We are honored to have you. I'd also like to extend our sincere gratitude to our sponsors, especially our platinum sponsor, BridgeBio Neuromuscular, as well as our bronze sponsor, AskBio. Your partnership and commitment to advancing therapies for LGMD make today's summit possible. Here's what you can expect today. You can follow along with our schedule in LGMD News Magazine or on our streaming site where the schedule is also posted. We are organizing today's summit into 3 segments. The first is clinical trial updates, featuring companies with active clinical trials and therapeutic programs in the LGMD space. The second segment focuses on policy and advocacy. Our final segment highlights emerging treatments and the exciting scientific advances helping to shape the future of LGMD. Throughout the day, you'll hear from many of the world's leading experts in LGMD. We hope the discussions today inspire collaboration, new ideas, and continued momentum towards better treatments for the LGMD community. With that, let's begin. Our first segment today is clinical trial updates. Our first presentation is from AskBio. Please join me in welcoming Dr. Susan Sifers, Vice President of Global Clinical Development for LGMD and Pompe programs at AskBio. Dr. Sifers, thank you for being with us today. The floor is yours.
Thanks, Kat, for that introduction. Hi, everyone. I'm very happy to have the opportunity to speak here today. I am the clinical lead for the neuromuscular programs at AskBio, and we'll be discussing our limb-girdle muscular dystrophy 2IR9 investigational gene therapy program. I'm an employee of AskBio with no outside disclosures. For background on the company, AskBio is a wholly owned and independently operated subsidiary of Bayer. It is a fully integrated gene therapy company with a robust portfolio across several diseases and systems. The clinical stage pipeline includes congestive heart failure, Parkinson's disease, multiple system atrophy, and our 2 neuromuscular programs of Pompe disease and LGMD 2I/R9. First, we're going to talk about the disease. So this slide is showing the naming convention for LGMD subtypes. Limb-girdle muscular dystrophy broadly refers to a group of muscular dystrophies caused by alterations in one of over 30 genes, resulting in multiple subtypes. 2I/R9 is one of the most common types. The naming convention is based on the mode of inheritance, in this case represented as 2 or R for recessive, and the order of discovery. This was the 9th discovered subtype, so I or 9, and then the affected protein is FKRP. LGMD 2I/R9 is a rare genetic disease caused by abnormalities in the Fakutin-related protein, or FKRP gene, encoding the FKRP enzyme, which is one of several enzymes required during glycosylation, whereby sugar molecules are attached to proteins making a glycan chain, which allows alpha-dystroglycan, alpha-DG, to stabilize the muscle membrane and support normal muscle function, such as muscle contraction. With abnormal FKRP, the structural link is disrupted, causing muscle cell injury and eventual replacement of muscle with fat and scar tissue. The most common type is homozygous for the 826CA mutation, meaning both copies of the gene have the same mutation. But a subset are compound heterozygous, meaning there are 2 different mutations in the same gene. This is a heterogeneous disease, meaning there are differences from one patient to the next, but it is overall characterized by progressive muscle weakness, leading to functional decline with high unmet need in both young and adult patients. The key clinical features are a limb-girdle pattern of weakness, calf hypertrophy, cardiomyopathy, progressive respiratory muscle weakness, and normal intelligence. Children often present with incidental lab abnormalities or intermittent symptoms. Adults often present in the second decade with progressive weakness. The median age at loss of ambulation is 43 years. There are phenotype-genotype correlations. So heterozygotes tend to start at an earlier age and have greater severity. Homozygotes are less likely to lose ambulation and have later onset of cardiomyopathy, with a median age of 54 versus 18 years in heterozygotes. However, the rate of progression in adults is similar between genotypes, with a decline in seated forced vital capacity, a measure of respiratory function, of approximately 2% per year. With weakness affecting skeletal muscles, as well as cardiac and respiratory systems, there is progressive functional decline that has significant impact on quality of life for patients and caregivers. There is a high unmet need with no approved disease-modifying therapies. Next, I'll talk about AB1003, our investigational gene therapy. It is designed to restore FKRP enzyme activity, primarily into skeletal muscle and heart cells, for the treatment of LGMD2I/R9. It has not been approved by any regulatory authority, and its efficacy and safety have not been fully established or evaluated. AB1003 restored normal FKRP protein function in muscle and preclinical LGMD mouse models, and is currently being evaluated in our phase 1/2 clinical trial called Lion CS101. Regulatory designations so far include orphan drug designation in the US and EU, as well as fast track and rare pediatric disease designations in the US. This is a visualization of the investigational gene therapy. The therapeutic FKRP gene utilizes an AAV9 capsid and a synthetic muscle-specific promoter. It is given as a one-time infusion and provides instructions for host cells to make FKRP protein. This slide shows the mechanism of action. It's a visualization with the left side representing the disease state. When FKRP protein is missing, alpha-DG is not properly glycosylated. And as previously discussed, this glycosylation is an important step to maintain the structural link between the basement membrane proteins such as laminin and proteins that bind to the actin cytoskeleton, collectively maintaining cell membrane integrity during contraction. On the right is a representation of the mechanism of AB1003, which is being studied for its potential to restore FKRP function and muscle cell membrane integrity. Now we'll talk about the safety and efficacy of AB1003. So this is showing the study design. It is a phase 1/2 randomized double-blind placebo-controlled dose-finding study. Participants are given a one-time infusion of either AB1003 or placebo. Cohort 1 is the lower dose. The first 2 patients were randomized 1-to-1 active treatment versus placebo. After 45 days with no dose-limiting toxicities, the second pair was dosed. After 45 days, there was a pre-planned DSMB review before continuing. After clearance to continue from a safety perspective, cohort 1 was finished with 1 additional patient, and then the sentinel pair in cohort 2 were dosed. This is the higher dose. There were again no serious adverse events or dose-limiting toxicities, so we had safety clearance to dose the last 4 participants, now randomized 3 active to 1 placebo. 2 of these have been dosed and 2 remain. 12 weeks after the last patient is dosed, there will be another DSMB review, as well as a planned interim analysis. The primary follow-up period is 52 weeks, and then there is a 4-year long-term follow-up. There are a combination of in-person and remote visits with physical checkups, muscle biopsies, blood tests, pulmonary function tests, cardiac and lower extremity MRI, echocardiograms, and quality of life questionnaires. These are the key eligibility criteria. For inclusion, the age range is 18 to 65, and there must be both a clinical diagnosis as well as confirmation with the FKRP gene mutation, and the ability to walk or run 10 meters in less than 30 seconds. For exclusion criteria, there must not be evidence of significant cardiomyopathy, liver, or kidney disease. There must not be need of daytime ventilatory support, and patients cannot have a neutralizing antibody titer to AAV9 of over 1 to 5. The primary study objective is evaluation of safety. Secondary objectives are to assess functionality, including multiple neuromuscular clinical assessments, biomarkers, quality of life, and shedding aspects of AB1003, as well as patient-reported and clinician-rated outcome measures. Exploratory objectives include characterizing the immune response and the effect on muscle with MRI and biopsy, as well as pulmonary and cardiac function. The clinical trial was designed with patient input. Insights were provided about the daily impact of the disease, procedure preference, emotional and social needs, as well as gene therapy awareness. The design aimed to reduce patient burden with several measures, such as home-based testing with mobile nursing, travel coordination, using needle instead of open muscle biopsy, and a crossover design. So now we're going to look at the safety results. These are the baseline characteristics of cohort 1. This was based on an interim data cut in May of 2025 that included 4 participants at 52 weeks and 1 at 36 weeks. The mean age was 42 years. 60% were female and 100% were white non-Hispanic. All were homozygous. So this is the safety profile of cohort 1. There were no serious adverse events or dose-limiting toxicities. Commonly reported non-serious adverse events, so occurring in more than 2 participants, included headache, fall, and nausea. 3 participants had asymptomatic transaminase elevations, so an increase in liver enzymes without changes in bilirubin, which is a common occurrence in AAV gene therapies. None of the patients had symptoms of this, and all returned to baseline levels with corticosteroid treatment. No other clinically significant lab abnormality trends were identified, and there were no cases of TMA or myocarditis identified. In summary, the interim safety data from the phase 1/2 Lion CS101 study suggests that AB 1003 has an acceptable safety profile in LGMD 2I/R9. An independent data safety monitoring board recommended to proceed with enrollment of participants in cohort 2. 4 participants have now been dosed in this higher dose cohort, and enrollment is ongoing. We are looking forward to wrapping up enrollment of this cohort soon. I want to give a special thank you to LGMD patients and caregivers for their contribution to advancing therapies by participating in clinical trials. And we want to thank the Speak Foundation for hosting this event, and to all our partners in this incredible community for raising awareness and supporting advancing therapies. Thank you. And now we'll be heading to the next section with Dr. Rodino-Klapac.
Thank you, Dr. Sifers. Thank you so much to the organizers for giving the opportunity to provide an update on our limb-girdle programs at Sarepta. Next slide. I'm Dr. Louise Rodino-Klapac. I'm President of R&D and Technical Operations at Sarepta. Here are my disclosures. I am an employee of the company. Next slide. So today I'm going to provide an overview of our programs across our portfolio. And so on the next slide, what I'll do is I'll just start with a summary of our different clinical trials, and then I'll go to each of them in more detail. So first, starting with Journey, we have a natural history study across limb-girdle subtypes. We've presented this data at various conferences, which are listed there, and I'm going to talk in a little bit more detail about each of those. The next 3 studies listed are all related to limb-girdle muscular dystrophy type 2E, which is a deficiency in beta-sarcoglycan. We had 2 phase 1 trials where we've published the results, and then also our ongoing phase 3 study, which we presented at WMS, and I'll talk about each of these. And then finally, I'll finish with the early data from our LGMD2D trial, which is SRP 9004-102, or the Discovery trial, and this is looking at the safety and expression data for SRP 9004 and 2D. So next slide. So first, starting with Journey, this is a natural history study looking at subtypes in LGMD2E, 2D, 2C, and 2A. All of these cohorts have been shared with the GRASP consortium that I'll share in more detail about, with the exception of 2E. The 2E study is ongoing, and so we'll be using that data as our comparison for our BLA or a biologics license application. So the Journey study is designed to look and follow natural history of patients for at least 6 months or up to 36 months. And the data from the study has been very useful in the fact that it describes the population. Some of the outcomes are shown at the bottom, which shows that, unsurprisingly, younger participants are predominantly ambulatory, while many patients over the years of 8 are non-ambulatory. We see that cardiac and pulmonary complications tend to be in the non-ambulatory, the more advanced parts of the disease as well. And we also see that North Star ambulatory assessment scores decline with age, indicating disease progression. So next slide. So as part of the Journey Natural History Study, we also performed a serial prevalence study. And what this means is that we tested patients within the study to see whether they had elevated antibody titers to our gene therapy vector, which is AAV RH74. And so 127 participants were tested for this, and those that had an antibody titer of less than 1 to 400 were considered negative, while those above were considered positive. And so upon looking at the data, approximately 84% of patients were negative for antibodies to RH74, and approximately 15.7% of patients were what's called seropositive or positive for antibodies. We were also able to test patients that were negative again and found that they remained negative. We did have 4 patients that were seropositive that became seronegative after retesting. So this data was incredibly useful and helpful to both us developing this and then also to the community as well. And this data is very consistent with what we see in Duchenne. So as I mentioned, we've recently provided the data in 2C, 2D, and 2A to the GRASP consortium free of charge. And then this data will be shared free of charge with the community. There'll be a steering committee that evaluates the ask and how the data will be used, but this will help inform other academics, other companies to be able to use this data, importantly, to get the most out of it and be able to show the effect of other therapies in contrast to the natural history of the disease. And we were very happy to be able to do that and help move the community forward. Next slide. So now I'll start providing the data and the results of our 2E program so far. And so our first trial was called SRP 9003-101. This is a first-in-human phase 1/2 trial to look at the efficacy of 9003. And so in this study, we looked at 2 different doses. Both were effective, and we looked at expression at 60 days, where we saw increases in beta-sarcoglycan expression compared to baseline. And then these patients will be followed for up to 5 years. So in terms of safety, we saw consistent with what we've seen previously with RH74. The most common adverse events were vomiting, liver enzyme elevations, and abdominal pain. We did have 1 acute liver injury, which is a more severe case of elevated liver enzymes in this population, but the patient recovered. Next slide. We also conducted a study called Voyaging. This is a second phase 1 trial in which we looked at individuals that were older ambulatory as well as non-ambulatory. In this case, we used 1 dose, which was the higher dose from the previous study of 7.41 times 10 to the 13th. Again, we saw very good gene expression at day 60 and then continue to follow the patients. We have a follow-up period for function of 18 months, which I'll show you on the next slide, but we saw overall stability in function and reduced CK levels. Safety, again, was consistent with previous reports of increased liver enzymes and nausea. Next slide. So here's the functional results from both the 101 study, which is shown on the left, and the 102 study, which is shown on the right. So first, looking at the left hand graph, this is the North Star ambulatory assessment for ambulatory patients in our first study. And what you can see is that the treated patients, which are in purple, continue to either improve and stabilize over the course of 18 months, while the natural history control, we see a decline over that period of 18 months. And so overall, this led to a 7.8 point difference in the North Star ambulatory score for those ambulatory patients. And then similarly on the right, in the non-ambulatory patients, we use the test called the Performance of Upper Limb to look at upper limb function. This is the Pool 2.0. And so over 18 months, we saw stabilization of the Pool score in the treated patients in purple. And then in the natural history control, you can see there was a decline in the stability of those patients over that 18 months. And so we saw a 3.5 point difference in that Pool score in treated patients compared to untreated patients. So this is encouraging data in these early phase 1 studies. And this helped inform the design of our phase 3 study, which I'll describe next. Next slide. So our phase 3 study, and just generally a phase 3 study, is considered what's called a registrational study. It's what's the ultimate study, which is used for approval or for asking for approval for an individual drug. So this study was called Emergene, and we were looking at safety and expression in ambulatory and non-ambulatory patients. The primary endpoint for this study was beta-sarcoglycan gene expression at day 60. We met the primary endpoint of increased beta-sarcoglycan expression, and then patients are continuing to be followed for up to 5 years, and that will be included in the BLA, and that will ultimately, the BLA will be based on gene expression, so we'll be asking for approval, for accelerated approval based on the biomarker, and then the confirmatory evidence will continue to go on, and we'll be looking for the NSAD and the ambulatory patients and the Pool and time to rise and 10-meter walk as well, up to 5 years. So in terms of safety, we saw, again, similar to what we've seen previously in terms of nausea and vomiting and increased liver enzyme elevation. And then next slide. And so finally, I'd like to just describe the results of our phase 1 study in LGMD2D, which is called the Discovery Study. This again was a phase 1 trial looking for safety and gene expression at day 60. And so what we saw in terms of results of that study were expression of the full-length alpha-sarcoglycan protein at day 60, restoration of the dystrophin-associated protein complex at the same time. There was CK reduction also at day 90, which shows there's the potential for clinical benefit there. And we don't have all of the functional measurements that we have with 2E, but those data were collected while those patients were followed. In terms of safety, overall similar to the profile of RH74, nausea and vomiting and increased liver enzyme elevation. We did have one patient, an older non-ambulatory patient with acute fatal liver failure at day 77 after dosing. This was similar to what we've seen in the Duchenne population in non-ambulatory patients, where we had 2 cases of acute liver failure. And so now we're evaluating the use of sirolimus prior to gene therapy to help reduce the incidence of acute liver injury. The SRP 9004 program is currently on clinical hold, as all of the limb-girdle programs are on clinical hold. And the results of the study in Duchenne with sirolimus will help inform the path forward for getting off of clinical hold, and then ultimately submitting the biologic license application for LGMD2E, and then being able to lift the clinical hold on the other programs to potentially find a path forward with partners for those other programs. And next slide. So just to summarize, we continue to push forward and find a path forward for LGMD2E and the other limb-girdle portfolio. In terms of next steps, as I mentioned, we are conducting a study using sirolimus to help reduce the incidence of acute liver injury and ultimately acute liver failure. We're testing that in the Duchenne population first in non-ambulatory patients. We've already aligned with the FDA that they will use that data and apply that to limb-girdle as well. And so once we have that data in hand towards the end of this year, we'll be able to go back to FDA, asked to lift the clinical hold in 2E, and then ultimately align with the FDA on a path forward to submit the biologics license application. All of this data will be important for 2E, but then the rest of the limb-girdle portfolio for lifting clinical holds and being able to find a path forward. And then finally, just to reiterate, the journey data has been shared, and we will help the community use that data effectively to help move gene therapies forward, other therapies forward, and really help the community as a whole. I'd just like to end by thanking this community. Thank you to the patients and the families who participated in our trial. Your efforts were tremendous, and really, we couldn't do this without you. We're doing it for you, but also with you. And there's no way to conduct these trials without your participation and your efforts. And we appreciate that. And just know that we are here as a company at Sarepta, continuing to fight for the limb-girdle community.
We're excited to welcome Brad Williams and Rachel DeConti. We're going to be having a patient roundtable with Dr. Louise Rodino-Klapac from Sarepta Therapeutics. We'll be asking questions, and this will be included on our LGMD YouTube channel as well. Hi, Louise. Hi, Brad. Hi, Rachel. Thank you all for being a part of this roundtable. Rachel, would you like to share just a few seconds of who you are, and Brad as well?
Yep. Hi, my name is Rachel DeConti. I am the executive director of the LGMD2D Foundation and mom of Jacob, who is 9 years old living with LGMD2D. Happy to be here. Thank you.
Thank you.
Hi, I'm Brad Williams. I'm director of research at the Jane Foundation, and I am a patient living with LGMD R2-2B. Nice to be here.
Thank you. Nice to have you here. And Louise, would you like to just introduce yourself again? I think it'd be important.
Sure. I'm Louise Rodino-Klapac. I am president of R&D and technical operations at Sarepta Therapeutics and been working in the limb-girdle field for 2 decades now.
Thank you, Louise. And this is just a discussion. So we are adding features like this to our summit so that we can bring more Q&A from our patient community with a lot of our researchers and clinicians and industry partners. So we have some questions that we've put together, and each of us have one question that we're going to ask. Some of us have more than one. So our first question, Louise, and thank you for being willing to share, is there has been change over the past year in Sarepta's LGMD gene therapy program, including pauses and new safety steps. So that our community understands what's going on, we would like an update, and can you help us understand in simple terms where things stand today and what have we learned so far?
Sure. Thank you. As the community very well knows, we've been developing a platform of limb-girdle muscular dystrophy gene therapies for several years until last summer. During that time, we had 2 unfortunate ALF or acute liver failure deaths in the non-ambulatory population of Duchenne, and 1 acute liver failure death in the limb-girdle 2D population. This was devastating for these families, for this community, and for all of us collectively. Following those events, the FDA placed all of our limb-girdle programs on clinical hold. And so that means no work can be conducted in clinical trials. We could not submit our biologics license application for LGMD2E, which was planned, until we could do further studies to demonstrate safety with RH74 in limb-girdle and in the older ambulatory Duchenne population. So as of right now, the programs are still on hold until we get data from the non-ambulatory population in Duchenne, and I think we're going to talk more about that next.
Okay, all right, thank you. And while we're talking, if we want to have a follow-up discussion on it, would that be okay potentially?
Sure.
Okay. All right. Brad, I think you have the next question.
Yeah, so we heard that Sarepta, in trying to improve the safety profile of gene therapy, has been planning to work with a medicine called sirolimus in DMD gene therapy. How might this help inform what happens next in DMD, especially for programs like, 2E, where you were about, to apply for a biologics license?
Thank you. In AAV gene therapy, generally, acute liver injury, elevated liver enzymes is a known risk. And so there had been some work in the field on using immunosuppressive agents. Previously, we used prednisone in our trials, but there were additional immunosuppressive agents that you could use to dampen this immune response. And so, together with the FDA, we designed a trial in non-ambulatory patients with Duchenne called Endeavor Cohort 8. You may hear it called both, to test this. And so, that trial is ongoing. We expect it to finish enrollment soon and have data by the end of the summer. And so, what we're looking for in that trial is formally, it's a 50% reduction in the incidence of acute liver injury, which is a precursor to acute liver failure. And then the agreement with FDA was once we had that data in hand, we would go back to discuss with them next steps. And this was also true for the limb-girdle 2E program. Once we had that data, they understand how small the 2E population is. And so they would accept the data from Duchenne as evidence also for limb-girdle. So once we have that data, at the end of the year, we'll talk with them early in 27 about the path forward for both programs. And safety is always our top concern. And so we're doing, in addition to the study with sirolimus, additional monitoring in our Duchenne program, both commercially and in our clinical trials, which is just increased monitoring, making sure that there's widespread education around early signals of acute liver injury so that we can best keep our patients safe while giving them the opportunity to have these medicines as well.
Okay, thank you, Louise. And then I think Rachel has the next question.
Yep. Sarepta has shared that it's pausing some LGMD programs while exploring other approaches and potential partnerships to move these programs forward. You just touched on it a little bit, but what does that mean for the future of these programs, and how are you thinking about continuing progress for the LGMD community?
So for LGMD 2E, as soon as we have that data, we'll discuss the path forward to filing the BLA. There's a chance that they may want to see a few patients treated with sirolimus in LGMD 2E prior to submitting the BLA, but we'll have that discussion first. And then that's really the precursor for any other programs. The other limb-girdle programs in our portfolio are paused. We are having a lot of discussions around potential partnering of these programs with other entities, whether that's patient organizations, whether that's companies. We want to find a path forward for all of these programs. The precursor for that is getting off a clinical hold, regardless of whether it's with us or with someone else. And so we're rapidly working so that that can be enabled. We want to see these medicines get to the community, and we're doing whatever we can to help. Separately, as part of all of this, we did a natural history study called Journey, and we've recently just shared the data from Journey with the GRASP consortium, so that data can be combined and published, and also provided free of charge to the community for use with academics, for those that can use it to help inform their research as well. So trying to do whatever we can to help push the community forward.
Thank you, Louise. And my question was about the Journey data, so thank you for bringing that up. When you talked about sharing back with the community, with the Journey natural history data, how do you think that families might benefit from that information now being at GRASP LGMD? And then, if it's possible, may I add that advocacy for the community really was effective, and Sarepta was very open as well to hearing from our community about wanting our natural history data. And I want to say thank you for making sure that happened. But can you talk a little bit more about that?
Sure. So we thought it was very important to provide. It's it's difficult. We are dealing with, as you very well know, these are small numbers and individual LGMD subtypes, and so having more data, combining it with the GRASP data, the more numbers you have, that means you have what's called more power. And so when you're comparing treated patients to natural history, that just helps you define your effect better. And so that's what we're trying to do. We know how much work went in and how much effort from the patient community to participate in these trials. And so we want their data to be very meaningful and help the community. And that was important to us. So GRASP will have a steering committee. We know that they'll be good stewards of the data. We want to make sure that it's used effectively. And we trust that they will do that and then share with um, other entities, like the CPATH, for example, is another place that can use the data, model the data, and make the most out of it.
Oh, and I really just want to say that that was, so awesome, because all of our community and our patients that participated, we really participate in these natural history studies for the purpose of progressing the field. And I think, Louise, you understood that, and making sure that that was something that could progress the field. And I know that all of the modeling now that can take place with the journey data and the previous data that GRASP has will allow us to have a more robust outcome measure as well. So, do you want to share about how you see that that could happen as well?
I think the technology, not just on our therapies, but also on the way we analyze data, especially using AI. We can't get through a presentation without talking about AI these days, but it's really giving us a lot of powerful tools to be able to make the most out of data, which is fantastic because we can't perform large, what historically were called phase 3 studies with hundreds of patients in rare populations. It's just too difficult. And so combining as much data as you have and then using tools to be able to model it and make the most of it is really going to help push everything forward.
I know. I think it's really helpful, and I know we're really excited about the potential. So thank you so much for that. I want to pivot, and I think our next question is from Brad.
Uh, yeah, I was wondering if you could talk a little bit about the sort of efficacy results that you've obtained with the LGMD gene therapy programs, and what the implications of that are for, moving forward once the, clinical hold is lifted and the, safety issues are addressed.
Sure. We talk about it in the presentation, but generally, we saw very good signs of efficacy. So, first, speaking about the 2E program, we did 3 studies in that, 2 phase 1s, one in a non-ambulatory population. Second, the 102 study was ambulatory for 101, non-ambulatory for 102. We saw robust gene expression in both of those studies. And then over a longer period of time, in comparison to natural history, in ambulatory patients, we saw a divergence in terms of their NSAD scores or North Star Ambulatory Assessment. And then in non-ambulatory patients, in the performance of upper limb. And so those data are very encouraging. And then those measures are being collected in the phase 3 study, which is still ongoing at this point. We saw good gene expression, which was the primary, and then those patients we followed up to 5 years for function. And then in 2D, we've recently presented these data as well. And so there we saw very good gene expression as well. It's early, so we don't have the same functional measures yet that we have for 2E, but things were looking promising. It was unfortunate that we had to stop that trial. Obviously, I can't not talk about safety. We had consistent safety profile and the unfortunate acute liver failure in an older non-ambulatory patient in that trial. So overall, we're always looking to balance and look at safety and efficacy. Very encouraging signs of efficacy. We have safety, which we can manage for the most part. We're making sure we can manage the acute liver injury part effectively, and that's the data we're collecting now.
Okay, thank you so much, Louise, for answering that question. That was really helpful. And then, Rachel, I think you have our last question.
Yeah. And given the promising efficacy data that you just mentioned, Dr. Klapac, is there a way for patients to get access to these therapies now through compassionate use?
So prior to getting therapies, any of the material that's used for these studies has to go through a rigorous process. For any patient to have a medicine, that there has to be an open investigational new drug application or an IND. And so really the precursor to patients continuing to get these medicines requires the clinical hold to be lifted. That's the process. It's certainly something that we're going to ask the agency as soon as we can get the holds lifted. So first, the material has to be available depending on the subtype, and then we have to have an open IND. And so those two things have to come together. So we're working hard to make sure that can happen in the subtypes where we have material available.
Okay. Mm-hmm. Thank you, Louise, so much. And we appreciate your just openness to talk with us and appreciate that. And in terms of future conversations, would you be willing to come and share again like this with our community just so that we can understand more where things are?
Absolutely. And I so much appreciate the community's willingness to listen. I know how difficult this past year has been. I mean, I can't say enough of how hard it has been. And just know that we are always working, me as an individual and Sarepta as a company, to do right by patients. And that's what we're trying to do. But I know how hard it's been, and I definitely don't want to diminish that at all. It's been tough, but we will pull together as a community and push forward.
Thank you. Thank you. Rachel, Brad, do you want to share anything before we go?
Well, I just wanted to say a thank you to Louise for, her efforts in developing gene therapy, both for, DMD as well as LGMDs. And, it's like she says, she's been at this a long time and has made major contributions. And, I think the fact that there, is an approved gene therapy for, DMD is largely due to her efforts. So, I just wanted to say a word of appreciation.
Yeah, and I'll echo that appreciation and thanks to you, especially Dr. Klapac and Sarepta, with the years of research you put into LGMD and how far we've come. We thank you for where you've brought us, and we know that you will continue to help drive us forward, and we're appreciative and thankful. So thank you.
Thank you for giving me the opportunity to speak on behalf of Bridge Neuromuscular. My name is Doug Sproule. I'm the Chief Medical Officer here at Bridge Neuromuscular, formerly ML Biosolutions. And it's a distinct pleasure to have the opportunity to participate in the LGMD Scientific Summit. I'm going to talk about a few salient things. Our transition from a name perspective to Bridge Neuromuscular. I'm going to, more relevant to this community, I think, give an update on our phase 3 study and its study status, the overview of the U.S. regulatory process and how it affects our ongoing application for approval in the United States. And then lastly, what you can do as a patient, both as an individual, as a family member, as part of the community, to really advance things for yourself as well as for other people affected by this disease as we get closer and closer to having a potential therapy available for patients. So I'm going to talk first about a name change, and it's both trivial and important. We have retired the name ML Biosolutions in favor of calling ourselves Bridge Neuromuscular. And this speaks to a broader ambition of being a potential platform company that in the future we hope to be involved in the development of multiple therapies for multiple neuromuscular conditions. That doesn't change the fact, the core origin of this company, which sprang out of the McCall Lockwood lab at Atrium Health, and is a direct legacy of the philanthropic efforts of the McCall and Lockwood families to drive forward the development of therapies for patients affected by limb-girdle muscular dystrophy type 2I. And, I can't state the immense appreciation that I personally have, and I know many other people have in the community, for the work that of those families and the work that emerged from the lab, from that lab, driving forward the creation of ML Biosolutions and our progress with ribitol, we call BBP-418, as a treatment for this disease. None of that has changed. The core ethos of our organization remains the same. We are a team of passionate, solution-oriented scientists. Every minute counts, and we're focused on driving forward and improving the lives of people living with neuromuscular diseases. And that legacy will bear forward. Hopefully first for patients with limb-girdle muscular dystrophy type 2I, and hopefully in the future we'll have the opportunity to bring forward therapies and advance the science for other forms of neuromuscular disease as well. I wanted to give an update on the phase 3 study, our study called Fortify, which is a double-blind, meaning that the physician investigators don't know the status, the patients don't know the status, we as sponsors don't know the status of an individual patient. Randomized controlled trial, meaning the patients are randomly assigned to either receiving treatment with BBP-418 or treatment with a placebo. This study has been fully enrolled. It's currently ongoing. Patients are at this point in somewhere between their second and third year on study. As many people know, this is a study that enrolls patients who are genetically confirmed with limb-girdle muscular dystrophy type 2I, teenagers and adults, ambulatory and non-ambulatory patients. And this study has produced an interim readout that we are currently working with regulatory authorities to discuss potential pathways for approval. But at this point, it remains ongoing and is ultimately a 36-month study that will generate, we expect, very strong and robust data supporting the safety and efficacy of this product for patients affected by LGMD 2I or 9. The most important thing is that we have opened and initiated an open label extension study. This is a study that is available for the patients who participated in the Fortify study. As they complete their enrollment, they have the opportunity, and I expect most people will take that opportunity to enroll in an open label extension where they will receive active BBP-418 in an open label fashion. So about this open label extension study, again, it's specifically for people who completed the Fortify study and meet the eligibility criteria. And all patients who enroll in the open label extension study will receive BBP-418. So what that means is that it's open label, so everybody knows what they're receiving. There's no blind, there's no placebo. And this allows us to evaluate the long-term safety and to some degree the long-term efficacy of BBP-4 and 8 in patients over the longer term. So again, it's only eligible for patients who complete participation in the phase 3 study. And obviously, as any study, this is optional and would occur after patients give informed consent and agree to the procedures. The study will help establish a long-term safety of this product. There will be continued safety labs that will be obtained. We'll monitor breathing, upper limb function, walking ability. We'll measure serum creatine kinase. There will not be a muscle biopsy as part of the open label extension study. And certainly if you have questions, if you are potentially eligible for this study and you have questions, I would highly encourage you to talk to the physician that you've been working with as part of the Fortify study. I want to talk a little bit about the U.S. regulatory process and specifically how it affects our program and this product. So we have submitted an application for a new drug application with the U.S. Food and Drug Administration, or FDA. And this starts a strikingly long and cumbersome process from an outsider's perspective, as the FDA conducts a systematic review of the data that underlies a drug that was potentially able to be approved for commercial use and available to the community. Our application was submitted earlier this year and is currently under review. We have received a priority review for that application, which drives an approximate 8-month review period, and something that's utilized for serious or serious diseases or diseases with considerable unmet need, of which this is one. There's continued scientific review, discussion of the indication, how the drug label will look, what data will be included in it, what patients are potentially should potentially receive this drug based on the data that's available. And then there's ultimately an NDA approval. We have what's called a PDUFA date, or a new drug application approval date, that is going to occur sometime in late November, we would expect. So then the question would be, what does that mean for patients? Once there's an approval, there is an extremely frustrating lag time that would then occur after that as well. There's a number of aspects that would need to occur. The labels and distribution needs to be determined. Negotiations often need to occur with health insurers and other and local pharmacies to ensure that this product would be ultimately covered. And this is something that takes often several months. But the bottom line is things are changing at a pretty rapid pace, and I would encourage, obviously, to keep in touch with your physicians about what's going on. Stay tuned with the advocacy organizations. And certainly, we will be as forthcoming as we're able to be with additional information as we are able to be, as we're able to ascertain it from the FDA, and as we're able to communicate it to the community. And the question is, what do we do right now? As a patient, as a member of this community, what can you do for yourself, for the broader community, as this period drags on while we're waiting for therapies to emerge for patients? I think the first thing, as an individual, is to make sure that you have a confirmed diagnosis, and I'd strongly recommend that every patient get a hard copy, paper copy of their genetic testing. If there's any questions about whether the genetic mutations that you have may be a variant of unknown significance, or whether there's questions about the pathogenicity, to have those tests redone or reevaluated, because one of the requirements that any pharmaceutical company or any payer or insurance company would have legitimately is, does this patient actually have the disease that we're we're planning on prescribing this therapy for. And so it's really critical that you have that information. And certainly it's a great time if there's any questions, if there's any doubts on things, it's a great time to get retested if that's what needs to occur. A second thing, it's again an incidental thing, but um, one of the amazing efforts of the advocacy community, Kat Bryant, Kelly Barrazo, and others, was to push forward to have LGMD-R9 as a specifically identified sub component of the LGMD ICD-10 code. And so, in as much as you encounter the healthcare system, it's really important to remind the physicians that there is a specific code that's immensely helpful to help us understand the burden of disease, and from a standpoint of the physicians, from the standpoint of potential drug companies in the space, but also especially from the payers who are looking to understand what the implications of therapies are and potentially offsetting other significant costs. Um, Another and probably the most critical thing is stay connected. This is the time to make sure that you are connected with the physicians, connected with the advocacy organizations, that you are as tuned up as possible and tuned in as possible with our physician partners to make sure that you have as much information as possible to make the right decisions, educated and informed decisions on behalf of your own care. I guess another question is, what else can you do? I'd point out some things. And again, I don't have any skin in this game other than it's something that really helps advance the science, advance our understanding of the disease, understand the epidemiology of the disease, understand how patients progress. At this point, absent any therapies in the future, in the context of therapies as they emerge. And that is to participate in registries. And I'd strongly encourage anyone who's interested to join the FKRPA registry. It's pretty easy to enroll. There's some basic information that's needed to participate and to sign up. It's a two-way street. It provides information that helps us to better understand the nature of this disease. That can help inform care, help inform care guidelines, and can provide a lot of information that can be immensely helpful in driving future therapeutic development. It also connects you in with the community as well and gives that potential access to future clinical trials and a closer contact to what's going on in the broader community. You need to have your genetic diagnosis, and you'll need to be able to provide that to a registry coordinator. One of the things that's important to note, and this is true for clinical trials as well, everything is voluntary, and that voluntary nature is at any point. And so there's a lot of work that's done to protect your privacy. This is entirely voluntary, and at any point you can always withdraw that consent as well. And so there's the information on there. I strongly encourage people to participate if they have the interest and desire. And so I think the next steps from us as a company, the Fortify study is ongoing. For people who are participating in that study, thank you. It's more critical than ever to continue to adhere to the study protocol and to continue to participate and make those study visits. We are working with our regulatory partners about next steps as far as that study. But at this point, it's critical that the study continue as scheduled. Our open label extension study is now open for patients who are completing their 36-month period within the Fortify study. And our regulatory engagement continues. We are moving forward very rapidly in the United States. We're also seeking guidance for outside the United States as well, as far as paths and next steps towards hopefully ultimately getting a regulatory approval, not just in the United States, but elsewhere. And it's our broad goal and ambition and expectation that will make this product ultimately available to every patient who could potentially benefit from. And while obviously information comes in drips and drips, we'll do our absolute best to keep the community as informed as we can be every step of the way. And I think that's the last slide. And I wanted to call out a real thank you again to every every patient who has participated in our Fortify study, the phase 2 study, our natural history study that was conducted through the GRASP Consortium. I want to also especially thank all the families and caregivers who've also donated their time and effort and energies and passion to supporting their loved ones, not just through the disease, but also through the complexity of research trials. I want to thank our community partners, the investigator site coordinators, all the other members of study staff at dozens of sites who have really, through their dedication, brought the study to life. I want to thank our partners in the DMC, but particularly I want to thank the community at large, and particularly the many advocates who pursue in such tireless fashion the pursuit of improving the lives of patients affected by this disease. So with that, I'll stop, but I wanted to proceed to the next section, which will be hosted by Byrne. So thank you and wish you a good day.
Thank you, Dr. Sproule. And I'm delighted to provide the next presentation. My name is Byrne from the University of Florida. I'm going to talk about a gene therapy program that we've conducted in conjunction with Atomayo and Genathon regarding gene therapy for LGMD R5-2C. We'll present the preliminary safety and efficacy data from a phase 1 study of ATA200. Just by way of background, ATA200 is a gene therapy product developed by Genathon and Atomayo, which is designed to address the unmet need in a form of limb-girdle muscular dystrophy due to gamma-sarcoglycan deficiency. The ATA200 product is designed to express the transmembrane protein gamma-sarcoglycan, which is required for structural integrity of skeletal muscle. Loss of sarcolemmal stability leads to muscle fiber damage and ultimately muscle loss and profound weakness. Gamma-sarcoglycan deficiency is a rare disorder with less than 10 people affected per million. Its onset is in childhood, usually in early school age years, 6 to 8 years old. And there's progressive muscle weakness, particularly in the proximal musculature. There is some evidence of cardiac involvement, and the product has been carefully designed to express at the appropriate levels in heart muscle, focusing on correction of the membrane defect in all of the peripheral skeletal musculature. In null mutations, there is no protein, and much like Duchenne muscular dystrophy, this is the most severe phenotype. Missense mutations with a reduced amount of protein can produce milder limb-girdle-like phenotype. Shown here on the right is the impact of loss of sarcoglycan in the axial and central skeletal muscles, showing a progressive fat infiltration of the musculature and loss of muscle mass, particularly in the upper limb and quadriceps muscle. The ATA200 gene therapy product was designed to achieve balanced expression between cardiac and skeletal muscle using the murine creatine kinase promoter with a configuration that achieved equivalent expression between heart muscle and skeletal muscle based on a self-complementary AAV vector configuration of the vector genome and packaged in an AAV8 capsid designed to adequately reach all of the skeletal musculature. Studies were done to demonstrate proof of concept in the gamma-sarcoglycan knockout mouse model, which showed that it was possible to restore muscle function and gamma-sarcoglycan expression to the wild-type level. Following that proof of concept study, GLP toxicology studies were done in wild-type rats that demonstrated safety up to a dose level that exceeded the clinical dose level by almost 10 times. The ATA003 GSAR study is a phase 1 study of safety and tolerability of ATA200. It's an open-label phase 1/2 study designed to evaluate safety and efficacy. It's a 2-part study in which there's a 6-month evaluation period with 4.5 years of long-term follow-up. The study population consisted of ambulant children between 6 and 13 years of age who were seronegative for neutralizing anti-AAV antibodies. Importantly, the study used a pretreatment regimen for immune suppression that included methylprednisolone, followed by 1 milligram per kilo of prednisone for 4 weeks, and then progressive tapering. And importantly, an additional immunosuppressive agent, sirolimus, was given with a loading dose of 4 milligrams on day 1 and then 2 milligrams per day for 13 weeks, followed by 1 milligram per day for another 13 weeks. The investigational product was administered at a dose of 1 times 10 to the 14th vector genomes per kilo in a 2-hour infusion. The main evaluation criteria of the study were the North Star Ambulatory Assessment, as well as the PUL 10-minute walk test, a quality of life questionnaire, as well as assessments of a muscle biopsy for vector genome copy number, mRNA, and percent sarcoglycan positive fibers, as well as the creatine kinase levels. This slide shows the demographic and baseline characteristics of the 4 study participants thus far who've received the ATA200 product. You can see the identical mutations in the first 2 participants, and then a different missense mutations in patients 3 and 4, as well as a deletion in patient 4, all leading to substantially reduced gamma-sarcoglycan levels and NSAD scores shown here at baseline. So ranging between 27 and 39 at baseline, with PUL scores between 30 and 40. Importantly, there was evidence of normal respiratory function and heart function at screening, as well as a comprehensive assessment by MR imaging, which I'll describe in a moment. The adverse events across the 4 participants were relatively common, mild to moderate nausea and vomiting in the first week. We have been able to follow up the first 2 participants beyond 12 months, and we are nearing the 6-month biopsy for the 4th participant in the study at this point. So just to analyze the CK levels that were observed in the study, at baseline, all 4 patients showed CK levels between 28 and 62 times the upper limit of normal. Of course, there's the potential for an effect of corticosteroids on the CK level, and we saw in all 4 participants in the first 60 days a substantial decline in CK from an average of about 5,500 to 1,000. This rapid reduction was seen in all participants by day 14, but most importantly, even after sirolimus discontinuation at day 180, all remained with a greater than 90% reduction in CK after 9 months. The other parameters that were tracked longitudinally were the AST and ALT. In all participants, the AST, ALT were 3 to 5 times the upper limit of normal, and these all returned to normal following treatment, which is a really encouraging sign. This is not only related to the corticosteroid treatment, but was persistent through this 1-year time point. Looking at the NSAD scores, these are represented as absolute change from baseline. And you can see, particularly in study participant 3, a significant improvement. The lowest NSAD score was in participant 001, which is the red line. And you can see there was improvement at the 4-month time point, and then a slight decline by the 1-year evaluation. In terms of 10-minute walk times, measured in seconds, it is shown as change, an absolute change from baseline. We showed remarkable stabilization even in the most severe baseline, and then improvement in the other two participants thus far out to 9 months. Looking at the PUL score shown as change from baseline, we see an improvement even in the oldest and most severe participant at baseline, and then stabilization now out to 12 months. And for the participant 002 and 003, essentially completely stabilized in an upper extremity function. The 22-item questionnaire of the Activin showed all the participants to find that there was a favorable outcome in measuring daily activity limitations from baseline to 9 months after a follow-up. This video gives us insight into the difficulty. If you see on the left panel, the difficulty rising from the floor with the Gowers maneuver, taking far longer than after treatment of 13 seconds. You can see the great difficulty there was in standing. So when we compare the pre-treatment of 25 seconds to 13 seconds after treatment. So that's very encouraging and has provided an interesting outcome in terms of confidence and ability to ambulate independently and less concern about falling. In terms of the molecular results of the study, the vector copy number was quantified by quantitative PCR. mRNA was assessed by RT-qPCR, and the protein level of expression was quantified by the method called Simple Western. So here are the first two participants with where biopsy has been analyzed now after 6 months, and shown in the first two rows for participant 001, baseline levels of vector or mRNA or transgene are below the limit of detection. And after 6 months, nearly 1 copy per cell per diploid genome, and a significant amount of RNA being expressed that leads to transgene expression that's easily measured by the Simple Western. These are just arbitrary units, but it appears that the amount of protein being localized into the membrane actually restores the wild type level of membrane function. And then in participant 2, similar high level of protein expression and mRNA expressed from 0.34 vector genomes per diploid genome. This is the most telling figure of the histological analysis of the biopsy at 6 months post-infusion for patients 1 and 2. You can see at baseline laminin staining shows the membrane perimeter in all cells. No staining for gamma sarcoglycan, or at least measured at some level about 0.3%. And alpha sarcoglycan is localization as high as 89%. After 6 months, we see 90% of myofibers expressing gamma sarcoglycan, which is quite encouraging. And in the younger participant, if you see the bottom middle on the right, up to 92% of positive fibers post-treatment, as you compare really no expression at baseline compared to the 6-month time point. Another observation from the study was the ability to do a frequent MR analysis done with the imaging NMD group, and Dr. Glenn Walter and Donovan Lott performed these studies. And in the upper left-hand panel, you can see the fat fraction measurement across a variety of the important muscles that were measured: the soleus, the vastus lateralis, tibialis anterior, adductor magnus, and the bicep femoris long head. Varying levels of involvement across these muscle groups, but call your attention in the predominantly slow fiber type of the soleus, where we saw a pre-baseline assessment at minus 10 months. There was slow but measurable progression at baseline. But interestingly, by the time the this first participant was enrolled, there was rapid progression of fatty fiber conversion, and you see the significant change from around 13% to almost 16% fat fraction in just 3 months. The rate of change between baseline and 3 months, 3 months and 6 months, and 6 months and 9 months was progressively less as the vector began expressing gamma-sarcoglycan and stabilizing the membrane. And then surprisingly, even after between the 9-month and 1-year time point, we see a beginning of a reversal of this trend, which is really encouraging. This was also seen to some extent in the tibialis anterior and the adductor magnus. In the biceps femoris and in the vastus lateralis, there was either stabilization or continued progression, but at a substantially lower rate than earlier on. And when we look at this similar data set across the two participants, you can see that the one that is younger and had less severe disease has a complete stabilization of the fat fraction in the soleus and a dramatic change in water T2 levels in both individuals. It was a little more delayed in the 12-year-old participant than in the 8-year-old. But these also, I think, represent evidence that there is adequate target engagement to prevent and reverse inflammation that had been occurring as a result of the membrane instability. So, in conclusion, the ATA 200 study for the treatment of LGMD R5 was really well tolerated with no adverse events other than mild nausea and vomiting. Based on the first 4 patients, we're really encouraged to continue enrollment in the study. 6 months data from the first 2 participants showed functional improvements. Successful target engagement is evidenced by the positive biodistribution and expression of gamma-sarcoglycan by several methods, greater than 90% sarcoglycan positive fibers. And importantly, I think this study contributes more to our understanding of how inflammation in the context of gene therapy may influence outcomes, because pretreatment with sirolimus not only enhanced the safety of the gene therapy treatment in this study, but also led to increased efficacy compared to what was expected from the non-clinical studies. Let me take a moment to give special thanks to the sponsor of the study, Rodino, as well as the foundations that help support the study financially, the Dion Foundation and the Cure Sarcoglycanopathy Foundation, as well as AFM Telethon, which through their work with Genathon and Rodino help support all the preclinical evaluations and the conduct of this study, as well as our team at UF Health and the Powell Gene Therapy Center. I specifically want to thank also our team members listed here, as well as the many participants in the conduct of the preclinical studies and evaluation of the treatment effect in the post-treatment biopsy samples, the Dion Foundation, as well as the Cure SCG Foundation. So thanks very much, and I appreciate the opportunity to present this data today, and we look forward to hearing more in the next presentation from Dr. John Vissing.
Well, thank you, Dr. Byrne. My name is John Vissing. I'm the director of the Copenhagen Neuromuscular Center at the National Hospital in Copenhagen, Denmark. As you can see, it says Genathon Atomai there after my name. I'm not really an employee there, but this is the sponsor of the study. And this is a study on gene therapy in limb-girdle muscular dystrophy type R9 or 2I. It's a dose escalation study. These are my disclosures, and the only thing really pertinent to this presentation is that the sponsor was Atomai, which is mentioned here in the disclosure. So this diagram here shows you, I'm pointing here with my cursor, the cell membrane of a muscle cell, and a lot of proteins here that are transmembrane proteins. And you can see all these antennas sticking out from these proteins. This is sugars or glycosylations, which are really, really important for every single cell in the body. in the muscle, they have a very important function in anchoring the cell to the extracellular matrix. And really, what goes wrong in limb-girdle muscular dystrophy type R9 is that you are you're breaking this glycosylation chain up to laminin here, so that you don't have this adhesion to the extracellular membrane. And this is on the right side here. This is what we are trying to fix by giving back the gene product to patients, so they can glycosylate properly and then reestablish the link up to the extracellular matrix. This picture here shows you the gene construct. So we are using a small virus here. It's very small, and you can package all genes into it. So like, for instance, dystrophin is much too big a gene to put into this virus. But FKRP, the gene in question here, is a small gene, so we can actually have the full construct put into the virus. And in this case, there was also a microRNA packaged in here to detarget the heart, because preclinical studies showed some toxicity in the heart in mice. This is the design of the study, showing you that this is a dose escalation study. There were 2 doses, one here at the top with a smaller dose, and then a dose here 3 times higher than in cohort 1. There were only 3 patients in each cohort. Patients were followed for 1 year and then had a 5-year follow-up after that. These were all, as you see on the right side, ambulatory patients with a moderately affection of the lung function. So the forced vital capacity should be between 40 and 80% of normal. As with all other phase 1 studies, this was primarily safety that was looked at, but there were a number of other outcomes, clinical outcomes, that we will mention later on. Here to the left bottom, you see that all patients received immune suppressants with the gene therapy. So on the day of the infusion, methylprednisolone bolus, and then after that, quite a high dose of prednisolone, at least for 28 days, but some of the patients had this even for longer time. This table here shows you the 6 patients that were included. The 3 top ones here are cohort 1, the lower dose, and then the higher dose here below. And in the column here on your right-hand side, you can see that, in fact, in cohort 1, patients had been followed for 3 years, and in cohort 2 for 2 years. And most of these patients were treated at our site, symbolized by the DK here. Only 1 patient was treated in Paris. First of all, safety. This was quite a safe drug. There were no serious adverse events. There were some gastrointestinal problems with vomiting and so on, and insomnia. These were probably related to the immune suppressants and not so much the gene therapy per se. But there were some transaminase elevations. These were asymptomatic, but were treated with immune suppressants. And we can see here on this one, where we look at the transaminases, the ALT and AST, we see how they fared in each of the 6 patients here. So the top panel here are the lower dose, and then the higher dose here at the bottom. And you can the bright blue here shows you prednisolone treatment, and you can see that patients had prednisolone for quite a while. Some of them up to a year after the infusion with the gene therapy. But if you look at up here in the lower dose cohort, all the transaminases came back to normal in the patients after the immune suppressant was taken off. Some biomarker response. So one is the creatine kinase response. Creatine kinase, as you know, is a muscle injury biomarker. And we see here that in the two cohorts, here the one in purple is the high dose and the red one is the low dose. We see in both groups, there's quite a marked reduction in creatine kinase, signifying less muscle injury. Actually, sort of an 80% decrease on average in the two groups. Quite impressive. If we look at some of the functional outcomes, and this here to the left is the the forced vital capacity of the patients, and the North Star Assessment Scale, which is clinical scale here on the right. We can see the course of the natural history cohort, not treated patients here in black. So actually they worsen, as you can see, for both parameters with time. But in the low dose patients, actually there was a small improvement in both NSAD and FVC, whereas the higher dose patients actually followed the natural history course. Part of this explanation for the higher dose not doing so well is that two of the patients in this cohort actually gained quite a lot of weight, probably because of the high prednisolone dose that was given here. And of course, that would make the assessments worse. Other assessments was the walking velocity didn't change much in the patients, but the active limb, which is a quality of life score, also improved in the lower dose and not in the higher dose, as we saw with the other functional measures. In summary, if we look at the velocity, North Star assessment, lung function, and the quality of life questionnaire, we see improvements in the lower dose cohort here in blue, and a deterioration in the natural history cohort here in gray, and in the cohort 2, the high dose patients. Lastly, if we look at some of the results, we can look at the, most importantly, the glycosylation of alpha-dystroglycan. And we see that in the lower dose here, cohort 1, there is 109% increase in the glycosylation. There was also an increase in the cohort 2, the higher dose, by 63%. We can't really say whether they are very much different, but at least nominally a nicer improvement in the lower cohort. And down here at the bottom, you see each individual patient increased in glycosylation with the treatment. So all in all, the study showed quite good tolerability to the gene therapy. There were some transaminase elevations, but they could be controlled. The biodistribution showed that there was a higher FKRP expression in the higher dose, but yet the increase in the glycosylation, which is what the gene therapy should do, what occurred in both cohorts, and it actually was nominally higher in the lower dose cohort. And in both groups, we saw a very nice reduction in the muscle injury biomarker creatine kinase. But clinical data only really show positive results in the lower dose cohort 1. And therefore, these data suggest that that lower dose should be picked in studies moving forward. Right now, we don't really have any certain timeline when these new studies will start. Thank you very much for your attention.
Thank you for your presentation, Dr. Vissing. This concludes the session on clinical trial updates. We will now take a 15-minute break before beginning the policy discussion session. Welcome back, everyone. Our next session focuses on public policy pertaining to rare disease and drug development. We'll begin now with a fireside chat from Neha Gandhi and Amanda Malakoff with BridgeBio. We'd also like to say a special thank you to BridgeBio for being our platinum sponsor of this event.
Thanks for that introduction. And I'm excited to kick off the policy discussion. Hi, everyone, and welcome. Before we dive in, I want to thank the Speak Foundation and Bryant Knudson in particular for the invitation and for the opportunity to join you all today. My name is Neha Gandhi, and I have the privilege of leading global patient advocacy at BridgeBio Neuromuscular. For us, patient advocacy is a strategic function. It's about making sure the needs and priorities of the community actively shape our decisions at every stage, from research to development to commercialization, and about engaging the wider ecosystem to break down the barriers that keep people from getting to diagnosis and to care. I've spent more than 20 years in rare disease advocacy and policy, and in that time, I've been fortunate to build partnerships that bring everyone to the same table. People living with rare conditions, their care partners, regulators, patient organizations, academia, and industry, all working together towards the same goals. Diagnosing people faster, moving therapies forward, and making sure care actually reaches the people who need it. And that's exactly why I'm so glad to be hosting this conversation today, because the LGMD community is one of the clearest examples I've seen of what's possible when patients, advocates, and industry work together. And I can't wait to dig into some of those stories with my colleague, Amanda. Let me start by saying a little bit about BridgeBio. At our core, we exist to develop transformative medicines for genetic conditions. The reality is that millions of people around the world live with a genetic condition and still have no treatment options, often simply because developing a drug for a small patient population isn't commercially easy. What we're trying to do is bridge that gap to turn incredible advances we're seeing in genetic science into real medicines for the patients who've been left waiting. The way we're set up is a little different. We use what we call a hub and spoke model, and it's really built for speed, precision, and scale. Each team works independently as its own spoke, focused on a single condition or drug candidate, while our central hub gives them clinical, regulatory, and commercial expertise that they need. We also partner very closely with leading academic institutions to push early and late-stage research forward, bringing the most promising ideas to light. And specifically, BridgeBio Neuromuscular, our team, actually grew out of a founder's search for answers for their own child living with LGMD2I RNA. BridgeBio's core value is to put patients first. That's foundational to who we are. But what does that really look like when it comes to policy and engagement and legislation? And how does the work that you do in DC connect back to that mission? Amanda, can you tell us a little bit about that?
Absolutely. Thanks, Neha. Really happy to be here. And really quickly, for those of you I haven't met yet, maybe I'll briefly introduce myself first. My name is Amanda Malakoff. I lead government affairs for BridgeBio. And my work here is really focused on advancing the policies that improve the lives of people living with rare diseases. And we do that by supporting policies that will enable innovation and expanding access to treatments, as well as ensuring that patient needs are really reflected in federal policy. And so what my team does is we work really closely with policymakers and offices on Capitol Hill, as well as our organization, patient advocacy organizations we partner with, and industry stakeholders to make sure that we can continue to move these priorities forward. So like Neha said, our focus is really in bringing these medicines to patients as quickly and safely as possible. And what we do to support that in DC is work to ensure that the policy and legislative environment keeps pace with the science so that patients can actually benefit from the new innovations that are taking place without hitting unnecessary barriers. And a lot of what that looks like is advancing legislation that supports orphan drug incentives, directing the FDA to review rare disease treatments with urgency and use all the tools that they have in their toolbox. And then also supporting the fact that once a therapy is approved, it's critical that patients actually have the ability to access it. And so those are all policies that our team focuses on. And our team and myself have been really fortunate to be able to partner with the LGMD patient advocacy community. That was something that I started doing day one in this and my role here, given the really close partnership that we've had with the Speak Foundation and all of the patient organizations. And it's just been really impressive to see over the past several years how this community has really started from the ground up and already had such an outsized impact in DC. And what we'll be doing today is talking a little bit more through some examples of the LGMD community's leadership in this space and how you all have really led the way in a lot of these efforts, and how we at BridgeBio have been, one partner who's been really fortunate to work closely with you on a lot of these efforts.
Thanks, Amanda. That's a really helpful frame for everything we're going to cover today, and I really appreciate that perspective. We're going to look at two concrete examples of that mission, of BridgeBio's mission in action, of collaboration and partnership with the community. First, how the LGMD community helped convene a scientific workshop that ultimately became proposed federal legislation. And secondly, how a critical drug development incentive called a priority review voucher was fought for and won. Then we'll turn the lens forward to see what's on the horizon in Washington and how the rare disease community can get involved. This work is always ongoing as the political climate and healthcare priorities keep shifting. So let's dig right in. Let's start with the scientific workshop, the LGMD scientific workshop, which took place in 2024. It was convened by the Speak Foundation, and this event brought together patient advocates, leading academic medical experts, senior FDA leaders, and drug developers to look for a pathway to regulatory approval for LGMD treatments. And really, Amanda, can you kind of describe for us what the problem it was trying to solve, and how did it ultimately lead to federal legislation?
Yeah, thanks, Neha. So yes, as you mentioned, the LGMD community, as convened by the Speak Foundation, hosted the scientific workshop in 2024. And what it did was bring together key stakeholders in the drug development process to have a scientific dialogue and be able to talk about innovation and innovative drug development approaches for clinical trials within LGMD. And I think what was interesting coming from that workshop is the rare disease patient community has done a really good job educating members of Congress and their staff about a lot of the challenges that FDA currently has and their ability to engage with experts. We've actually seen that, and FDA has themselves said that typically within rare disease, the first place where they engage with an expert on a particular rare disease would be once it gets to potentially the therapy gets to an advisory committee review. And I think we can all agree that at that point in the process, it's hopefully not too late, but it's later than we would like to see. And we would like for there to be a forum for FDA to more regularly hear from experts the way that they did in the LGMD scientific workshop. So because of the awareness that members of Congress and their staff had about this challenge, they were able to look at the scientific workshop convened by the LGMD community as actually a case study for how this type of forum could exist and help co-educate FDA on the science behind particular rare diseases. And so as a result, legislation was actually introduced called the Scientific Expert Act of 2025. And what that legislation would do is require FDA to participate in at least 4 of these externally led scientific workshops per year across distinct conditions within rare disease, so that the regulatory clarity that was sought via the LGMD community's workshop, that the community really had to fight to build this effort, can now actually become scalable for other rare diseases. So that's a really incredible example of how the LGMD community has been innovating, not just in the ability to engage and convene with the FDA, but also to now broaden this approach to be something that's more accessible for the broader rare disease community. And I just want to point out another really significant fact here, which is that this legislation was actually co-sponsored and led by the bipartisan rare disease caucus chairs in Congress in both the House and Senate. So this has been also something that's really been embraced by key leaders in Congress who are engaged on these issues. And the fact that it's bipartisan really speaks to, the value of this legislation and, its ability to gain support and hopefully move forward.
That's very cool, Amanda. That's such a clear example of how the LGMD community has helped turn a scientific challenge into a policy solution, not just by identifying a gap, but bringing the right people around together around the evidence. And it's also teased up really nicely our next conversation, because the same question keeps coming up again and again in rare disease. Even when the science is promising, what policy tools are needed to keep development moving for small patient populations? That leads us to this case study on the priority review voucher. Can you explain what a PRV is in plain language, Amanda, and then walk us through what it took to get reauthorized, which wasn't easy?
Absolutely. So, as background, a priority review voucher is a policy that's authorized by Congress to encourage the development of treatments for rare pediatric diseases. And essentially, how it works is when FDA approves a therapy for a rare pediatric disease like LGMD, the company developing it receives a voucher that can essentially cut FDA's standard review time from 12 months down to 6 months. And the company also has the ability to sell the voucher. And what they can do is use those proceeds to reinvest back into rare disease research. And in short, I would say it's one of the few policy mechanisms that directly supports companies who are investing in therapies for small patient populations who oftentimes need a financial incentive like this in order to do this work for very small patient populations. And we've actually seen that really bear out in the data. So what we've seen as we went through the last reauthorization is that since 2012, over 50 priority review vouchers have been awarded across 47 rare pediatric disease indications, which shows that this really has been a meaningful driver for investment in rare disease treatments, especially for small companies, like many of those who are doing work within the LGMD space. And to your point about this being somewhat of an uphill battle, the program expired at the end of 2024 and unfortunately was set to sunset in September of this year due to Congress's ability or inability, I should say, to get this reauthorized by that 2024 deadline. And with that potential sunset on the horizon, what that would mean is companies would not be able to receive a voucher if their drug was approved after that cutoff time. And at that time, it put many rare disease programs at risk for a lot of companies who really were relying on the ability to get a voucher or potentially sell a voucher to fund their work.
Thank you. Before we get into really how this played out, could you tell us a little bit about what was at risk for rare disease patients, LGMD patients, if this didn't move forward?
Yeah. So, essentially, like I mentioned, a lot of the programs who may not have received their approval by the FDA at that time were at risk to potentially not receive a voucher. And a lot of times, that voucher, as I mentioned, could be make it or break it for a small company who's really relying on that funding. And so, what I think is really interesting and significant about how this ultimately got reauthorized is this was not just, an industry or a company effort. What we really saw was the rare disease community banded together across all stakeholders and reacted to the true crisis that this was and called on Congress to renew the program. And I think what's really important to note for today is just the role that the LGMD community played in this reauthorization. I would say that this community was one of the most visible advocates. And really through this community's advocacy, it showed the power that one individual or one family or one community can have on actually spurring action forward within Congress. So a few areas I would highlight, we saw a lot of communication efforts. For example, Kelly Brazo from Cure LGMD2I had this amazing op-ed in Penn Live that was shared really widely across Capitol Hill, especially Pennsylvania. Offices took note about the importance of the program and about her family's story. We also saw the Dion family's extraordinary campaign that they had, making videos, with members of Congress that were aimed at the administration, talking about, educating about LGMD, the importance of the PRV program, and really getting attention to this issue. And I would say, The community really helped to echo and elevate these issues. There were so many sign-on letters, calls to members of Congress. Over 80 advocates, from what I understand, participated in LGMD Day on the Hill. And this was a key issue on the agenda, was to educate Congress about LGMD and the need for policies like PRV to support rare disease drug development. And we really saw results. We saw over 200 members of Congress ultimately co-sponsor the reauthorization bill, which makes this one of the most supported bills in Congress in the past 2 decades, which would not have happened without, truly seeing so much outreach happen from the community. And following all this advocacy that happened for the Give Kids a Chance Act legislation to reauthorize the program, we had actually specific members of Congress put pressure directly on House and Senate leadership when there was a vehicle moving forward in Congress to include Give Kids a Chance Act in that overall bill. And after, we saw the program lapse, finally, due to that pressure, it was reauthorized and signed into law earlier this year on February 3rd. And that program is now reauthorized through September of 2029. So that gives us several more years of really needed certainty in this space.
Well, congratulations to the community for all of their hard work. And that was quite a long and difficult path, even though it was ultimately successful. And now we have it reauthorized, but as you mentioned, only through September 2029. So what did we learn from this process, Amanda? What can we take forward as we think about, other actions that we'd like to, sort of get the community together on?
Yeah, it's a great question. I mean, I think what we learned is that even policies that are so widely supported and are such common sense policies and are bipartisan like this one can unfortunately stall in today's environment within Congress, where there's just so much gridlock and inability to move things forward. And so, this is a long-term effort, and what we saw from the rare disease community leading up to the bill finally passing this year was years, frankly, of really sustained and coordinated efforts. And the ultimate success would not have been possible without patient voices, including those of the LGMD community, reaching out to your member of Congress, sharing your story, keeping this issue visible and really adding the urgency to it. That matters and that counts. Without the patient and community engagement with your member of Congress and without your voice, this would not have been possible. This became a no-brainer. Congress couldn't leave it resolved after hearing from so many patients and so many of their constituents. And so making sure that we have this unified and sustained approach going forward will be absolutely critical.
That's really helpful, Amanda. And I think good perspective, these two great wins together, the scientific workshop that led to the proposed federal legislation, the Scientific Expert Act that you mentioned, and the PRV reauthorization. It's really about how the community works together. Can you pull together maybe one or two, common ideas or common threads for the community? You mentioned a few about how the collaboration actually moves this legislation forward.
Yeah, I mean, I would say we should not underestimate the power of the rare disease community because, as we always say in this community, rare disease is not rare. When you actually look at it, there are 30 million people in America that are impacted by a rare disease. And so when we band together, we can have a voice and we can have an impact that cannot be ignored. So I think that's a really key theme that came out from all of this. And I would also say, maybe circling back to the LGMD scientific workshop, is the role that science plays in all of this and how science is really what helps to earn a seat at the table. That workshop was a success because the community was so impactful in bringing data as part of the discussion and bringing data to bolster the really critical patient impact stories as well about lived experience and meaningful outcomes to help drive the science forward. And I think that resonated so strongly as well with those on the Hill. Ultimately, we have so much innovation in science that's happening within the rare disease space, and everyone supports wanting to see that move forward and actually benefit patients. So I think to summarize, really leading with the science and coming together as a community to really amplify our impact while also not underestimating the effect that one patient story or one patient voice can have on a key decision maker.
I think those are such important points, Amanda, that these wins didn't happen by accident. They happened because the community, kept showing up with their lived experience and, as you said, the credible evidence, right? The data. And because those advocates stayed engaged long enough to move an idea through a difficult policy environment, a quagmire, right? But as we all know, policy work doesn't end when one bill passes. The landscape keeps changing, and there are new decisions ahead that will shape rare disease drug development and patient access for years to come. What topics, Amanda, are top of mind for the rare disease community now, and how can people get involved?
Yeah, so I think everyone keeps asking what's next after PRV. And we actually did not have to wait very long after the reauthorization until FDA actually came out and said that they would support PRV being made permanent, which has to be done by Congress. So I think there's still more work to be done on the PRV front to hopefully avoid being in a situation like we were in last time where, the PRV unfortunately sunsets or expires. But I would say beyond that, we're really at an inflection point where the rare disease community is taking stock of what new reforms are needed. One big topic that we didn't touch on yet, but that's also really top of mind, that was passed alongside the PRV legislation was reforms to pharmacy benefit managers and how they are now required to pass the discounts that they receive directly to patients and enable patient access. That's also an area that the patient community has been really outspoken and advocating for and making sure there's more transparency around PBM reform. There was also legislation newly passed in February as well. So I think with both of those passing, we're looking at kind of what's next, especially as we now look ahead to the fact that we're entering the 2026 midterm election, where not as much is going to get done as members of Congress are really focused on their elections. So what I would say is that affordability continues to be a major topic in all areas, especially in healthcare within Congress right now. And, we talked a little bit about patient access. What was passed in February was PBM reform within Medicare Part D. But there are a number of other pieces of legislation being proposed that would essentially expand PBM reform further than just this one aspect of Medicare, and would also try to seek the transparency measures and the passing along of rebates to the commercial and the Medicaid markets to ensure that the patients are really getting the cost savings that they should be instead of having, the PBMs pocket the rebates, which is what we've been seeing for many years. Another area that the rare disease community has been engaged is on bipartisan legislation called the Help Copays Act, which would require commercial insurers to apply copay assistance toward a patient's deductible or out-of-pocket max, which actually, as of today, is not a requirement and seems like a very common sense policy that a lot of members of Congress are behind. I would also add another topic is newborn screening. Is really front and center for the community as well. And I know we were all very pleased to see earlier this year that HHS made an announcement to add Duchenne muscular dystrophy as well as MLD to the recommended uniform screening panel, also known as the RUSP, last year. And what that means is that states can now adopt both of those conditions to be screened for newborns. But there's a lot more that can and should be done to modernize the newborn screening system and make sure that we're diagnosing patients as early as possible and ending the diagnostic odyssey that so many patients face. So I would say, organizations like the Every Life Foundation and NORD have done a really good job galvanizing the patient community, sharing timely updates. I know they did that during PRV when there was a critical hearing coming up, or when there are times where Congress was voting on a bill where there was an opportunity to include PRV, they would send out really timely action alerts, but also convene the community to think about and align on what the key priorities are and align on strategies to drive them forward. Potential ways to engage is frankly just knowing who represents you in Congress, especially if your member of Congress is a member of the House Energy and Commerce Committee or the Senate Health Committee, which stands for Health, Education, Labor, and Pensions. Both of those committees oversee health policy, and engaging directly with your member of Congress and educating them about your story is also a really important way to engage.
Amanda, is there anything that people can do in their own communities? These are great ideas that talk about, advocating at the federal level. What can you do in your own community?
Absolutely. I think that's a great point, Neha. Members of Congress frequently have breaks where they're back in their district and they're working from, their office located in the state or in your community specifically. And they have full staffs located within each community in their office there as well. So I also think it's a great idea to, not just have to feel like you need to come to Washington, D.C. to meet with them, but also meet with them in their local offices when they're back in your state. And they really love to hear about, your real experience as a patient. And any data that you can share helps make the impact clear, especially if there are multiple constituents or multiple people within the community that can align and share that. Because really, at the end of the day, this is local. Members of Congress want to hear from their constituents, so the more that you can make it specific to how it impacts members of the community, the more that they'll care, and the more this will influence their decisions. And I would also say that there are a number of opportunities to engage from a patient perspective with what the FDA is doing. There are a number of proposed regulations or guidances with FDA that are open for comment right now, and these become part of the official record and can also inform policy decisions. So you very much do not have to be boots on the ground in DC to impact change. There are ways to make your voice heard at home in your state or by sharing your feedback directly.
Amanda, this has been such a thoughtful and informative conversation. Our time flew. Time flew by. I really appreciate how you've helped connect the dots between community advocacy, federal policy, and what it all means for people living with rare disease, specifically LGMD. Before we close, I want to ask one final question. Based on everything you talked about today, what are 1 to 2 things that you want everybody watching to walk away with? What are those top 2 things?
Yeah, so I would say, this is really a moment that matters. Decisions that are being made right now by Congress will impact how the federal government and how the FDA works for years to come, and how they support or do not support, efficient processes to review therapies for rare diseases and make sure that patients have access. So if rare diseases are not on the radar or part of the conversation now, they're much harder to add later. And I would also say, following getting PRV reauthorized, so much education was done on the rare disease community, and there's so much support for the patient community. And as we mentioned, not a lot is getting done within Congress right now. And this is one of the few areas where we're still seeing a lot of bipartisan, movement and activity and collaboration. So in an environment where it's hard to get much done, this community has the ability to really get things done. And so we should definitely take advantage of that collectively to make sure that we're continuing to move the ball forward. And I would also say a big message is that you really don't need to be a policy expert or be located in Washington, D.C. to make an impact. Just starting with what you know and sharing your story and your experience and knowing who represents you and engaging with them is really how all of this starts and what makes a difference at the end of the day.
That makes a lot of sense. Thank you, Amanda. From LGMD to broader rare disease policy, progress really happens when science and community move together. And with so many important decisions ahead, staying engaged will shape what comes next. Because this work is never truly finished, and the gains we make today only hold when the community and its allies keep showing up to protect them. So thank you to the conference speakers, to Amanda, to Kat and the Speak Foundation for inviting us, the entire Speak team, especially Jessica Evans and all the behind the scenes work on this summit. It was such a Herculean task, and we appreciate being part of the party. And finally, to the LGMD and rare disease community for continuing to show up and speak up and drive this work forward. Thank you.
Thank you, Neha and Amanda. As you've heard throughout today's discussion, progress happens when patients show up. The LGMD Scientific Workshop led to the proposed Scientific Expert Act. The Priority Review Voucher Program was reauthorized, and for the first time, we heard LGMD was included in House Appropriations report language. None of those things have happened by accident. It comes from patients, families, researchers, and our whole ecosystem coming together to make sure our community is heard. As someone who lives with LGMD, I know how easy it can be to think that one voice doesn't matter. But I can tell you from experience that every major advancement we've discussed today started with people who were willing to share their stories. Today, we are continuing that work. At the Speak Foundation, we're working with policymakers on issues that directly affect our community, including efforts to reduce the burden of duplicate natural history studies. We are advocating for greater collaboration between NIH and FDA so that high-quality natural history studies can be used more effectively as comparator arms so that patients do not have to go through long placebo-controlled trials. But none of this happens without community involvement. This is why I want to personally invite each of you to join us for advocacy opportunities, such as LGMD Day on the Hill and other future opportunities that occur later next year. Every year, members of our community meet directly with congressional offices to educate policymakers about LGMD. You don't have to be a policy expert, and you don't need to know how Congress works. You just simply need to share your experiences. The future of LGMD will be shaped not only by science and innovation, but by the voices of those who live with this disease. So my call to action is simple. Get involved. Join us at advocacy events and share your story. Thank you for being a part of our community, and thank you for being a part of the future of LGMD. Our next session is our FDA Roundtable and Patient Panel. We're talking about patient-centered trial design and regulatory harmonization. I'm pleased to welcome two leaders from the U.S. Food and Drug Administration. Our first is Robin Bent. We also are proud to welcome Dr. Michelle Campbell. Thank you for being with us. We also want to thank our patient panel, Carol Abraham, Kelly Brazo, Joshua Thayer, and Brad Williams. I also want to thank Annie Kennedy as our co-moderator. Now to our session. Welcome everyone to our session, FDA and LGMD. How do we advance feasible patient-centered drug development? I'm here today with our moderators and our FDA experts. I am going to also introduce our patient leaders and participants. But first, I'm going to send this over to Annie so that she can do some introductions of our FDA experts.
Thanks so much, Kat. So good morning, good afternoon, wherever you are. I'm really honored to be here with so many esteemed and distinguished leaders in the LGMD community, as well as two of the world's leading experts in the topics that we're going to be talking about today, who joined us from the U.S. Food and Drug Administration to help lead us through today's topics that we know are of really great importance and interest to our community. So joining us today are Robin Bent, who serves as the Director of Patient-Focused Drug Development, and Michelle Campbell, who's the Associate Director for Stakeholder Engagement and Clinical Outcomes within the Office of Neuroscience, both of which are nested within the Medical Product Center known as the Center for Drug Evaluation and Research, which we often refer to as CDER. So we're super grateful to them for lending their time today, but also their expertise. Many of the new pathways and programs that we're hearing about coming out of the agency have not just been coming from the programs that they work within, but also coming from their leadership and their expertise within rare disease. So we're going to be talking about that today and digging in. Today's discussion is really going going to be bringing together leaders throughout the LGMD community with these two leaders with FDA to talk about how innovative regulatory approaches, such as the newly described plausible mechanism framework, can be applied to LGMD. We are also going to discuss how the inclusion and integration of the patient voice, very importantly, can be applied to therapeutic development and regulatory decision making in therapies for LGMD. So, without talking about what we're going to talk about, let's jump in. So, for our first question today, we are going to have Brad Williams, who probably needs no introduction here in this community, but serves as the Director of Research for the Jane Foundation, to pose our first question. Brad.
Thank you, Andy. Sorry.
Brad, you've known me
thank
so
you.
long. Just call me Andy.
Thank you, Anna. Thank you, Annie. So the first question, has to do with the fact that there are approximately 30 different genetic subtypes of limb-girdle muscular dystrophy, and while the few with the most common, the largest patient populations have active drug development programs, many of them are neglected because there's very small patient populations and it's going to be very difficult to do a traditional trial. So, many, including the Speak Foundation, have pointed out that this might be a good, use for basket or umbrella trials that we've recently heard of. What is the FDA's thinking on, how these might be applied to some of the rarer subtypes of LGMD?
Well, good morning, good day to everyone, wherever you're located. As Annie said, I'm Michelle Campbell, and I'm in the Office of Neuroscience in Cedar. And it's a really great question, Brad, that you've asked today about the potential application of a basket or master protocol trial. There's a lot of different names that sometimes mean the same thing, and I really think what you're describing and talking about in this platform approach is how do we really pull together our different subtypes and try to um, do the needed drug development and clinical trial research to the broadest amount of people possible. And we are very much open to the use of a platform based trial or a master protocol or basket. Like as I said, it's really in how we define it. But we are open to this, and we have seen this being done in a couple rare diseases in the Office of Neuroscience. Probably the most famous one in neurology would be the ALS platform trial approach, which is run out of MGH in Boston. And so that is probably the most robust one that in the neurology space and in a rare disease space that is ongoing, that we have been obviously supportive of and have been engaged with that the entire time. But there are other rare diseases that are exploring um, uh, platform trials as well that we're seeing come in, have discussions, some in the other neuromuscular diseases as well. So I would say, yes, start those questions I think can be asked. And I think we would encourage a dialogue of what could a master protocol or platform trial look like? What are the things to think about? I do think when we're implementing, and from our experience in the implementation of this, there needs to be a very well thought out design plan. How do we add the drugs or participants to the studies, and in making sure that you have all stakeholder buy-in. So it's not just FDA, but as well as industry buy-in to wanting to support these platform trials. But we're very much open to exploring a design like that and would be supportive of it. But we want to then make sure that you have all the necessary other stakeholder support as well, is going to be critically important for success.
Michelle, I really love you also citing the example of the ALS community that's had, a lot of success, which also, one of the things that really is important to point to there is it takes a lot of coordination and a lot of leadership from the community to really launch those programs and get them off the ground, which that community did very well. And to your point, there are other communities that have done a lot of work there and done a lot to bring and convene the partners necessary to launch those programs, which the limb-girdle community would certainly be capable of, which if that were something that you really determined was a priority for the community. The next question is going to be posed by Kelly Brazo, which, again, somebody who probably doesn't need much of an introduction, but Kelly is the co-founder and CEO of the Cure LGMD2I community. Kelly.
Thank you so much. So great to be here with all of you today. I'd love to hear a little bit more from our FDA leaders here today of an overview of the plausible mechanism framework that we're pretty excited about hearing about this year. And we'd love to know how it might be applied to the limb-girdle muscular dystrophies. What potentially could it apply to individuals of rare diseases that are under the age of 5 or older, sometimes older, 55 or older, who might not be included in the current clinical trial designs for our community? And just, any advanced patients that might not be included, and how could we find an opportunity for them to be included as well?
Yeah, thank you, Kelly. And we do know that since this guidance has come out, there's a lot of questions people have and wanting to know a little bit more about this. So I will admit I got an assist on this one from my own boss, Dr. Teresa Bracchio, who is the cedar point on a lot of this plausible mechanism work. So the plausible mechanism framework is an approach to developing data for effectiveness and safety, support and approval for individualized therapy. It's typically therapies that are targeted to a well-understood genetic cause of a disease. Because of these therapies are directed to a specific genetic variants, they may only be effective for patients who carry that specific genetic variant, which may potentially be a very small number of patients. The guidance specifically addresses the situation where there are only a few patients and a randomized control trial may not be feasible. It's not specific to any particular age group. We do know, obviously, that some of the works that we have been seeing has been in the pediatric space. The guidance does not establish a new approval pathway, so I think that's important to understand. We still have to leverage our existing pathways, but this is really truly a framework of how do we approach the discussions needed to support needed regulatory approvals. But again, based on that existing approval pathways for either traditional or accelerated approvals, it's expected that the evidence for effectiveness would be established based on a single adequate, well-controlled trial with that confirmatory evidence, taking into account the need for flexibility in establishing the evidence on the rarity and severity of disease. Whether the plausible mechanism framework can specifically be applied in the LGMD community is going to depend on how the therapy is, what type of therapy it is, and how precisely it targets the known causal pathology or genetic targets of the condition. And as I already mentioned, there's over 30 subtypes of the genetic aspect. However, the ability for the use of that single adequate, well-controlled trial with confirmatory study is not unique to the plausible mechanism framework, and the approach for establishing substantial evidence of effectiveness can be considered for limb-girdle muscular dystrophy. So what we're really saying is that the pathway is really, or the framework, I should say. See, I have to get my language correct, as it was initially came out as a pathway and it's a framework now approach. Was really for a little bit of that individualized therapy efforts we're seeing. However, I think thinking about the first question you did ask about a platform trial, I think the message we want to get out to the limb-girdle community is that we understand the rarity of the disease, and how do we make sure we collect good, rigorous data that would be able to demonstrate substantial evidence of effectiveness of a potential therapy, and that we have enough confirmatory evidence to be able to support an approval. So I think that's really the message, is that we want to make sure that no matter what mechanism we're employing to do drug development and clinical trials in this community, that we are able to establish that substantial evidence of effectiveness. And so I think that's why the conversations with us, when we say come early and often, are really important to really discuss how do we get the rigor that's needed to be able to apply the flexibility that we can when it comes in our rare disease communities with our clinical trials.
Michelle, can I ask a quick follow-up to that? Which is, in a lot of types of limb-girdle muscular dystrophy, it's a loss of function. So, gene transfer therapy has been kind of the main focus on. So how would the plausible mechanism apply there, as opposed to, gene editing targeted at one particular mutation?
Well, I'm not going to be able to fully answer that question. That would be a great question for my colleagues and our Center for Biologics, or CBER, that regulate gene therapies and gene editing products. But I do think it's important to note that the guidance does it covers both the Center for Drugs and the Center for Biologics. So the framework approach is going to be similar for each. The uniqueness is, and to what your point is going to be those conversations with either CDER or CBER and what you believe the targeted therapy is going to be. So for CDER, we would be regulating an ASO product, and then in CBER would be that gene therapy type product. And so in CBER, it's going to be a much different conversation about what is needed. I think, you do highlight something that is with the loss of function aspect is how would we then approach, that individualized therapy or applying this framework in that. So I think the emphasis is in this broader aspect of how do we build that trial to get the evidence that we need, regardless of a framework that's being used. I think what's nice about the plausible framework is it is tied to, this aspect of that targeting underlying pathophysiology of the disease. And so I think that's where we want to make sure we're getting that good information to be able to apply this framework. But I cannot fully answer your question. That is definitely a great question for my Center for Biologics. But I would be remiss to be able to give you, a correct response for that one.
Michelle, thank you so much for sharing that because I think in our patient community, these are questions we ask one another all the time. And so it's such a great thing to hear. Robin, I also know that you're on as well. Um, Robin and Michelle, pivoting a little bit because we're going to get ready to go to question 3. But when you're talking about platforms, with question 1 and question 2, these are questions about platforms. And I want to mention too, when we do something like a basket trial, and definitely the Speak Foundation is looking at that very closely and working with some experts to do this for the community with a master protocol. I wanted to ask, one of the dilemmas I've seen in the LGMD space, especially even, is that what happens when or if there is a adverse risk scenario with one of our subtypes? We don't want the whole platform to stall. So when I'm looking at the idea of a basket trial, what flexibility does FDA have if there is an adverse scenario with one of the subtypes? Will it affect the entire basket trial? And just wanted to talk to you about that real quick.
Yeah, I do know why you're asking this question, Kat, and I think it's a really great question. I think it's going to get tied to the therapy that's under study. So, is this a therapy that is targeting broad aspects of limb-girdle, right? So maybe not the specific underlying pathophysiology, but maybe some more symptoms aspects, right, in a broadest sense. Or are we talking about an arm? And I'm making up a very hypothetical. So to our audience members, what I'm about to say is a very hypothetical scenario. But if you were to have different arms of your platform or basket trial that were to be a different subtype, right? Um, is it is what you're seeing from that therapy that's being studied in that arm of the trial, is that where you're seeing something, a safety signal per se, or an adverse event, as you say? I think, first of all, we're going to want to make sure we understand the cause and root of said event. I think that is critically important to the community at most, right? Our job is to, protect the safety, of patients as part of this when we're going through the IND space. So we need to understand, a little bit about what happened, and is it tied back to the drug? Or the therapy that's being studied. And then I think the nuance of it is, again, what is that therapy? What was it targeting? And does the other subtypes have similar targeting mechanisms? And so is there a plausible potential of maybe something happening, or was it really unique to this variant and the delivery system? So I think there's a lot of unknowns, but I do think that we would need to do everyone's due diligence to really understand what that safety could have been event, how is it impacting that specific subvariant, if it is a therapy that's targeted, and then how does that apply to the other subtypes? So again, we would want to do our due diligence to really explore the root cause of that. I think we want to make sure, we take very seriously the safety of our people who participate in clinical trials. Obviously, that is the root of our mission. And I think we would want to really, really understand what's happening for a trial to continue on, but also we really make sure that it's for the entire trial population. So it's a great question, and I think as you explore, if you were to develop a protocol, a platform basket trial protocol, I think that's a question and a consideration to take into place as you would build that. And I think that's a fair question to ask.
I know we have a lot of topics we want to get to, but I appreciate that this is a really important area, especially if this is a area of focus for the LGMD community right now. But this would be a very rich focus for a meeting unto itself to invite the agency to come in and benefit from the agency's expertise, but also from other communities that have gone before, those that have launched platforms and those that have not, but done a deep amount of work in this space. So this may be an area, and you may be thinking about it already, to conduct a workshop and invite some partners to come in and provide some insights and experience on this topic in particular. But Michelle just gave you a great jumping off point for that. So thanks, Kat. I think next we're going to go to Josh Thayers, the legal advisor to the Jane Foundation. He is going to queue us up for some questions around patient preferences and the risk tolerance threshold.
Thanks, Annie. The point I want to make is that we often hear sort of two themes from the LGMD patient community. First, we're generally quite risk tolerant. And second, we're not expecting miracles. So even stabilization of disease progression would generally be considered a clinically meaningful outcome for us. And it seems to us that both drug development companies as well as regulators find that somewhat surprising. So that my question is, how can we make other LGMD stakeholders more aware of our views in this area on risk and what's clinically meaningful?
Thanks, Josh. So I think that the ELT-FTD meeting that was held previously really clearly documented that stabilization was something that was important to the community, and really kind of clarified that the community was risk tolerant. I think what is really important, and I'm not 100% sure who you mean by other stakeholders, but I think one of the things that we typically recommend in just getting the information out there is, if there's a way to publish this information, publish what you heard at what we heard at the patient-focused drug development meeting, if there's an opportunity and an appetite for doing some sort of patient preference study, if necessary, or something like that, in order to really kind of meaningfully kind of quantify that those patient preferences and such, that could be really, really helpful in maybe making this more clear to the other stakeholders. I know here at FDA we certainly have heard this, and I think our medical product developer partners or who are working in this space are aware of this. So I think really just putting this into the public domain so that if people are doing literature searches or other things like that, they're able to see that information could possibly be helpful.
Yeah, and I would just add, I mean, in the Office of Neuroscience, we very much know that in almost all of our rare neurodegenerative diseases, stabilization, no change, slowing progression, we know those are clinically meaningful aspects for communities. And so I don't think we would per se want to question that. We know that and understand that, and we've definitely come out and said that publicly at meetings before as well. I think to Robin's point about getting information out to the broader stakeholder community, I think is really important. I also think where some of the nuances and where maybe it gets a little, where people sometimes are wondering this is when we get into having to make our benefit risk decisions and having to look at the data that hopefully has been rigorously collected to support substantial evidence of effectiveness. And we're looking at the change that we're seeing, making sure that the change really is clinically meaningful. So the concept of what you're saying, we definitely understand. Sometimes it's how do we look at the data and interpret it in a way that actually can make that same conclusion. And I would obviously be remiss, and with Robin here too, for us not to mention, our patient-focused drug development guidance series that does really talk about how to collect that patient voice in a rigorous way. And there are many ways and opportunities, including a patient preference study or other patient experience data to be collected that can help support that information being collected during a trial that then can be reviewed by us as well to kind of support this overall aspect of what is meaningful to patients and what is really their viewpoint on that tolerance risk. So I think that we've put out some really great methods and papers and guidances to kind of help be able to do that work in a rigorous way, but also, how do you inform us? And so I really look, Robin, adding in the, getting the word out to those other stakeholders that are important because that's it. But I can say that for ON, we know that slowing progression or just keeping in a current state is meaningful to our populations.
Thank you.
Thank you so much, Michelle and Robin. I'm going to pivot to our next question, and that is from Carol Abraham. And this question's about inclusion of advanced patients in clinical trials. And so I'm going to pivot over here to Carol to ask this question.
Thanks, Kat. For many of us who've lived with LGMD for many years, we are often excluded from clinical trials due to either our age or the requirement of having to be ambulatory. We understand that there are valid safety reasons in some cases, but we think the default policy should actually be one of inclusion. Excluding us from trials may discourage coverage from payers following approval. So my question to you is, how might the FDA ensure the inclusion of more advanced patients in clinical trials?
Thank you, Carol, for that question. We very much hear what you're saying and do encourage the broadest spectrum possible to be included of that patient population into clinical trials. I think I want to clarify there is no default policy from us for what you're describing. We very much encourage, as I said, the broadest inclusion possible into clinical trials. I think what we see often and what comes to us when we are providing advice is, from the perspective of the stakeholder developing that therapy and medical product, what they know and understand of the optimal population that they believe can be treated and should be able to demonstrate effectiveness and efficacy in their trials in the most safe manner. But we absolutely encourage when we can, we definitely give this advice more often, and I think people actually know to be as inclusive as possible. We definitely understand with limb-girdle and a lot of our neuromuscular diseases that have a transition from an ambulatory to non-ambulatory aspect of disease progression, that there's still a need for therapies across the board. And so how do we need to incorporate that into our clinical trial design and into our inclusion criteria? So we definitely understand that. We've been having some conversations ourselves. How do we continue to push this and include? Do we truly need to have, what we're seeing where uh, just an ambulatory population and a separate non-ambulatory? Can there be a mix? I think those are conversations we need to think about and applying, novel and innovative methodologies. What are the appropriate statistics to handle that? How do you select endpoints in that way? So I think there's some things that in the broader community we can explore and think about how you do this. But please know that we do want the inclusion to the broadest level possible, because we do want to make sure that if an approval decision is made, it will be able to target the broadest population possible for that. And I do understand what you're bringing up is really a potential access issue for if approved therapy was to occur. So I would say this, as Annie said, is another great broader conversation that I think all stakeholders could be a part of, and really in talking this out through about how do we be as inclusive as possible, but in a reasonable time of the trial length to be able to demonstrate that effectiveness. And sometimes, that we know that we don't want long trials. And so sometimes finding that population that's going to lead us to an appropriate trial length is maybe why we're seeing some of this. But I think this is a great next step question.
And I'd just like to maybe add a little bit, and kind of reflecting back to Josh's prior question, is that, sometimes we hear from medical product developers, sometimes the medical product developers have their rationale for the inclusion, their inclusion and exclusion criteria. Sometimes it's the institutional review boards who may have safety concerns and things like that. So really, the more you can make people understand the feelings of the community around this, that could potentially be helpful as well.
Yeah, and can I I want to reiterate something that Michelle said, that I think this is another good example of there's an opportunity for collaboration across the broader community. So wearing my broader rare disease community hat, that this isn't something unique just to the LGMD community, but we see sort of an absence of trials that are conducted in the non-ambulatory communities across the board, because the majority of the outcome measures that are studied and are better known are in the ambulatory communities. So it really underscores the need to develop better outcome measures in our non-ambulatory communities so that we de-risk the clinical trial study and the clinical trials in our non-ambulatory communities. So there's a real opportunity. This could be a RISE workshop. This could be a workshop that just we work as a rare disease community to do ourselves and invite the FDA to. But there's a real opportunity to come together and say, how do we do this as a community? What do we understand? What do we learn from these PFDD workshops within those communities that are experiencing the same issue? So there's a moment to maybe come together here.
I like that, Annie. And I'm going to go to the next question here, but I wanted to just comment about what you just said. I agree. It would be wonderful to have a RISE workshop on this type of topic. And please include patients who live with the disease. A lot of times our patient community feels frustrated because we see events at the FDA. There's big, large meetings, and we feel left out. We feel like we want to be there. We want to be the patient who lives with the disease talking about policy issues. We study these, we talk about these issues all the time in our community. Kelly Brazo, myself, Brad, Josh, Carol, and probably a host of about 25 other patients are talking about these issues. We feel conversant about them, and we just want to have a seat at the table for these types of meetings and be included. And I'm not saying you have to include me. I don't need an invitation. I would just like to see somebody with the disease, the person that lives with it on a day-to-day basis and lives with the burden, included. But that brings me to the natural history study question for our community, because participating in natural history studies is very burdensome for our patients, and it's a long-term commitment. I've been in 2 natural history studies. Kelly's daughter has been in 3. And when I say I've been in 2 for my subtype. Kelly's daughter's been in 3 for her subtype. We duplicate the data over and over again, but our only motivation is to see the results being used broadly and effectively. Oftentimes, we don't see that that's actually occurring. And so, with that in mind, how could various stakeholders, including FDA, address the following issue? The redundancy that we see, and it's not just our rare disease, we see it across all rare disease. And I'm sure Annie can jump in here too, what she hears. But patients with various LGMD subtypes are often asked to participate in duplicate natural history studies. And so some of these natural history studies have even included muscle biopsies. So we're talking muscle biopsies, blood samples, extensive testing at times. And I would just like to hear what FDA's thoughts are on that. And then how can we solve that problem? Because the broader problem is that we want to use this as a natural history comparator. Big question.
No, I think I also think about this a lot and I talk about this a lot. And, it's not under FDA's purview to kind of control the natural history studies and who does the natural history studies. But what I think about, especially from a patient focused drug development standpoint, is if you as a community are working with different investigators, there's power in numbers, as you know, and really ahead of the studies, really trying to get clarity from the people who are performing the studies about what's going to be asked, how that data is going to be shared, when that data is going to be shared, is really one of the best ways to control this. We all have seen natural history studies where the study has gone on and then it's either been done in academia and there's a paper that's published, but it doesn't have a lot of detailed information. And so it makes it difficult for it to be leveraged by others. We've seen challenges where it's done by a drug company and then that either the company is bought or the program is stopped and that information kind of gets placed behind locked doors, never to be really seen again. And we feel how frustrating that is. It's frustrating for us. I can't imagine how frustrating it is for you all, having put a lot of your heart and your time and your energy into doing this. So really, I think the best thing that we can do is kind of ask questions as at the beginning about how this information is going to be shared back to the community. Is there going to be a joint publication with the community? Where is this data going to go? Can it be shared to something like the Critical Pathways Rare Disease Chairs Accelerator platform so that information can be seen and interrogated by others? And so I would really just recommend that this is a great place. Annie has pointed out several different places that are really great for advocacy. And I would say this is another great place for advocacy to really try and, ensure that you have an understanding with the people that are doing the natural history study about what will be collected and when that will be shared and with whom.
I remember when I attended your meeting a couple Februarys ago, hearing how many different natural history studies were in the limb-girdle community, and I was just shocked because I hear you. I very much hear you, and this is something I echo everything Robin says because it is slightly. I mean, I'll say it's kind of our pet peeve when we hear, communities come and say, and I have a friend whose child has a rare disease. I have free permission to talk about this experience. Is I get a phone call, Michelle, why do you need to update? We're in 4 natural history studies. Who do I update? I'm too busy. Which one do I pick? Right? I know what I would want to offer that advice. I have to watch as a friend versus what I do and that advice I give. But that's a real problem. And you're not alone in this problem. And so, my wish is like, how do we can get that single, really well-developed perspective, make sense for the current standard of care that occurs, data collection opportunity instead of us having, Joe Smith's study here and Mary's over here and picking who do I pick to get into and then for us when we see people wanting to leverage natural histories with reviews and they come in and we're like, hey, I haven't heard of this one, whose natural history is this, right? Or, oh well, I thought there was another natural history study, because we try to stay pretty well read in the literature. And so we are aware of natural histories. And so sometimes we learn about new ones too, at the same time that you're learning about new ones as well. So I do think that, putting back and talking about data sharing and what are we signing up for and how is this information getting back, you're really hitting things that are definite pain points for Robin and I in terms of we wish we could change how that happens, but we can't, because it does frustrate us a lot too when we see this. So I think you're asking some really great questions. I think about how do we get to ultimately using the natural history study in drug development, which is what we really want, right? And we know the ultimate goal is, can we use natural history to be that external control arm, right? I know this where most rare disease communities have a desire. And so I think going back to what you're talking about kind of ties into how do we make sure that we're collecting good, rigorous data to support that? And is having multiple natural history studies impeding potentially that, right? Because of that variability among who's willing to participate, because they're just exhausted because they're part of 3 natural history studies. So great question. And again, I think true among so many communities. And I think it's a really important talking point these days.
I want to say thank you guys for like being willing to talk about these really difficult questions, because these are challenging and these are patient driven advocacy issues in our community where, again, it was one of those things we talk about all the time because we've got patients participating, children participating, and it's an ironic situation when we see a kid getting out of school to do one natural history study only to 2 months later go do another one. And so now I'm at a point where I want to actually protect our community, and then I wanna ask those questions and say, "What can we do? And is this something that we can do a RISE workshop about? And are there ways that we can work together, not just for us, but for all rare diseases?" And I know Annie Kennedy would love to do this as well because these are like common issues we're hearing across all of rare. And I know FDA wants to do things about this. I just know you do because you're just wonderful human beings, Michelle and Robin. I know you guys have such a compassionate heart. And beyond being great scientists, we all know that this is involving people's everyday lives, and it's very hard when we have to go through things and people are not listening to the patients. So it's I want to say thank you so much for being here, for taking the time, because I know how busy you guys are. And I want to say thank you just for this wonderful open conversation. It was just a wonderful thing. And I want to say thank you to Annie and her passion for rare disease. We need you and we need others like you as well, speaking up for the patient and what we go through. And thank you to the rest of our panel. Thank you so much. Anybody else want to share anything before we go?
I just want to say a couple of things. First of all, this conversation has been amazing, and thank you for the gracious things you just said. There were just a couple of calls to action that even if only one of the amazing threads that were discussed here happened, it would rocket the field forward. But there were a number of amazing ideas that really could be transformative in LGMD that I really do challenge the community to move forward. But I will also, we referenced the RISE workshop a number of times. The Federal Register notice for submissions for ideas is open. So if the community wants to come together and put together proposals, please do. And we will support you with that. Every Life will support you with that and help move those forward. I also want to flag that there is always an opportunity for representation at FDA meetings, both on the agenda and in the meeting. So please, there is no reason to feel left out for that. And the agency does their best to make sure that there's full representation. And again, if you need support in doing that, we're happy to do that. But also, Every Life has rare giving grants so that if you need support getting to a meeting, we will help get you there. So I just wanted to flag that as another opportunity. But platforms, outcome measures, representation for the non-ambulatory population, patient preference studies, we have so many tools and resources to help operationalize everything that came up here today, and think that these are amazing things, not just for the LGMD community, but for a broader rare for us to be really working to operationalize. So this was an incredible conversation. And thank you to Michelle and Robin for helping really move us through this, and how there are some real near-term actionable opportunities for LGMD to move forward. So thank you to the leadership of the LGMD community for bringing us together today.
Absolutely.
Thank
Thank
you.
you.
Thank
Thank you,
you.
everyone.
Thanks so much for having us.
Thank you to our FDA and patient representatives for this important conversation. This concludes the session on policy discussions. We will now take a 15-minute break before beginning the emerging treatments session.
Welcome back. We will now begin our third session on emerging treatments. Our first presenter will be Joanne Donovan of Edgewise Therapeutics.
Thank you, Brad, for that introduction. My name is Dr. Joanne Donovan. I'm Chief Medical Officer at Edgewise Therapeutics, and we've been developing fast skeletal myosin inhibitor for Becker muscular dystrophy. But it has potential implications for limb-girdle muscular dystrophy as well, so that's why I'm talking with you today. First, I need to show you my forward-looking statement. We are a public company, and so I need to let you know that, first of all, sevasemten is an investigational agent that's being developed for Becker muscular dystrophy and Duchenne muscular dystrophy, and it's not approved in any territory. This is for U.S. people only. And I'll be talking about some things that may happen in the future, which is why I need to show you this. So Edgewise is a company that's focused on muscle disease science. It's been around since 2017, very specifically for muscle disease, and initially for muscular dystrophies. We're located in Boulder, which is lovely. This is a picture from our back deck. And we are dedicated to the mission of changing the lives of patients and families that are affected by serious muscle disease. Now, the way we've approached this is to protect susceptible muscle fibers in muscular dystrophy. So not only in Becker and Duchenne muscular dystrophy, but also in limb-girdle muscular dystrophy. Certain muscle fibers are more susceptible to damage because of the lack of the either functional dystrophin or the dystroglycan complex in the case of certain limb-girdle muscular dystrophies. What we've done is to make an investigational therapy that's designed to protect those specific fibers from damage. And it's something that could be used regardless of mutation, so potentially for a number of diseases. We have been focused on clinical trials to develop sevasemten in Becker and Duchenne muscular dystrophy. And we have done limited assessment of the investigational therapy in limb-girdle muscular dystrophy. So I'm going to focus more on what we've done in Becker and Duchenne, but it also potentially in the future could apply to certain limb-girdle muscular dystrophies. Now, this is a schematic of the dystroglycan complex. And what the dystroglycan complex does is it connects the muscle fiber that contracts. Myosin is the protein that contracts, and it connects that across muscle fibers. And what that does is when muscle fibers contract, they don't contract all at once. So when one muscle fiber contracts, this dystroglycan complex spreads the stress across the muscle so that it isn't damaged with contraction. And there are a number of proteins in the dystroglycan complex, not just dystrophin, but the alpha, beta, gamma, delta sarcoglycan, as well as in limb-girdle 2I, when FKRP is not functioning properly, that also affects the dystroglycan complex. So there are several limb-girdles that could be potential for this therapy. So as I mentioned, muscle fibers, you have individual muscle fibers contracting. And when the force is spread over muscle, then that protects them. But when dystrophin or the dystroglycan complex is absent, then the individual fibers can get damaged. And what this shows here is actually a mouse finger muscle. It's very, very small enough you can see it under the microscope. And when the muscle contracts on the left here at 100% without sevasemten, you can actually see the contraction-induced injury that happens in muscular dystrophy. And what you see is these blebs. These fibers are damaged, and that causes ultimately the muscle damage that results in loss of muscle. On the right here, when it's contracting with sevasemten, it contracts. It takes the edge off. It's only contracting at 85%, so it's perfectly functional. But what you see is that it completely preserves the muscle structure and prevents contraction-induced injury, all of the visible contraction-induced injury. So that's what we're trying to achieve. With sevasemten, we have gone through a clinical development program, initially with Becker muscular dystrophy, an open label study, and I'll show you some of the results from these. A phase 2 study that has now evolved into Grand Canyon, which is a pivotal cohort that is going to be finishing this year. And that is essentially a phase 3 study in adults with Becker muscular dystrophy. And we hope to go to the FDA for registration after that. All of the patients in these studies have continued into an open label extension that assesses the long-term effects of sevasemten on safety, biomarkers, and functional measures. We now have men that are out 4 years on the drug. And we've looked at, because this concept of contraction-induced injury is there in a number of limb-girdle muscular dystrophies, we also did an exercise challenge study in patients with limb-girdle muscular dystrophy 2I as well. And I'll show you a little bit of data from that. What we've used to assess how the disease progression can be affected by sevasemten is the North Star Ambulatory Assessment. And you've heard of that in other clinical trials, or the North Star Ambulatory Assessment for limb-girdle muscular dystrophies. And it's a number of measures that really reflect activity in everyday life. And it's assessed by 1 point if you can do them, but with compensation. So for example, to get up from a chair, you need to use the arms of the chair to push up. Or and a 0 means you can't do it at all. 2 means you can do it without visible compensation. And so it has real world implications of the things that you do in everyday life. So what does, say, a 1 point difference mean? And it could mean the difference between being able to go down a step or up a step to not being able to go up a curve, for example, not being able to get up off the toilet to needing to help for that, and getting up from a fall. So those are the kind of things that 1 or 2 points on a North Star ambulatory assessment can mean. So what we did was look at, as I mentioned, this shows 3 years. We now have men out to 4 years. And this shows on the vertical axis the change in the North Star ambulatory assessment. It's a maximum score of 34 points. And after 3 years, the men in this initially open label study of 24 months, then they were in an additional study, and they really hadn't changed. They were very stable at this point. And we developed a model based on the individual characteristics of these men at baseline, and that predicted that these individuals would have decreased by about 4.5 points. So that's very encouraging that we're seeing them stable over time. We also looked at biomarkers which tell us they're circulating. We can use drawn blood to assess them. And we saw that they were significantly reduced early on. So the thought is, if we can reduce muscle damage, then long-term we can preserve muscle, just like we did in that with those mouse videos. So sevasemten, safety is obviously key. Sevasemten was generally well tolerated, and the most common things happened early and transiently: dizziness and somnolence, which typically only lasted a few days or even a day or less. And those were the most common. So it's been very well tolerated overall. We then went on to a placebo-controlled study with biomarkers as the endpoint. We looked at men who were ambulatory between up to age 50, as well as adolescents. And they were in the range where they would be expected to decline over time, and looked at a dose of 10 milligrams daily, and we looked at a couple doses in adolescents. And what we found was that it was well tolerated in terms of adverse events. There weren't serious adverse events that were related to the drug, and people stayed on the drug, which is very encouraging. What we saw was we hit the primary endpoint, which is, of course, very important in any clinical trial. We saw that CK, which is released from muscle, went down significantly. And importantly, fast skeletal muscle troponin, which is specific for the fast muscle fibers, also went down dramatically. And this is the primary endpoint, which is great. We also looked at the North Star in this study. And again, we saw the 27 men who were on sevasemten for a year were stable, and the placebo, people who received placebo actually went down. And it was consistent with this blue triangle, which is what we would have expected from natural history studies that show that North Star goes down between 1 and 1.7 points per year. And of course, every year that continues over time. So what happened? We went out further. We had them go on for another 12 months, now in an open label study, and they continued to be stable. So it's a theme, and we're very encouraged by this, that they were stable, continuing on drug for 2 years, and the men who were on placebo initially then stabilized. So to understand also a little bit more about limb-girdle, we looked at how the biomarkers of muscle damage changed after exercise. And we worked with Dr. Vissing, who you may very well know, in Denmark, who has a very specific protocol of exercise, and then looking at the circulating biomarkers to see how much, to see muscle damage. And could sevasemten then protect that muscle damage? So they did a very well-controlled cycling exercise after 12 weeks on the drug on sevasemten. And then we can let them continue in the open label extension. And what we saw was the biomarkers of muscle damage, fast skeletal muscle troponin I, went down dramatically in the sevasemten group. This is the folks that were on sevasemten. They went down 93% at week 12 compared to the placebo group. And their creatinine kinase also went down significantly with 25% reduction. So we were able to identify that the exercise-induced damage was decreased with sevasemten in another disease where the dystroglycan complex was affected. So this is encouraging long-term. We saw, again, the most common treatment emergent adverse event was dizziness. Again, very transient. People didn't discontinue because of it. It went away quickly. So to summarize, in what we saw in Becker, we saw that the biomarkers of muscle damage decreased significantly, and it both after exercise as well as after everyday activity. And in limb-girdle 2I, we saw that it decreased CK and TNNI2. Even the exercise decreased these 12 weeks later. But then when we looked longer term, we saw that they were decreased, which is quite encouraging. It's not enough to go and say, yes, we're going to develop this, we're going to go to regulators for an approval, but it supports future studies. We now have the Grand Canyon study, which is the pivotal study in Becker muscular dystrophy, which we hope to bring to FDA for an approval. That's 175 patients, and it's an 18-month study, again, looking with North Star ambulatory assessment as the primary endpoint. So that will read out later this year, and we'll have results in the fourth quarter of this year. So what's next? We're going to be sharing those results. And as of a few months ago, almost everybody had moved into the long-term study, the open label study. So we will have men with substantial experience to be able to look at not only the long-term safety, but the long-term durability of the drug in preserving muscle function, which is the hope to stabilize disease and prevent progression. So with that, I would like to thank the patients, the investigators, and everybody who helps make this possible. It isn't possible, of course, without people participating in studies, both natural history studies, as well as the actual clinical trials. So thank you to the community. And we hope in the future to be looking at how we can develop this, not just in Becker or Duchenne muscular dystrophy, but in other muscular dystrophies as well. So stay tuned. And next, I would like to turn this over to Dr. Simone Spuler. And thank you again.
Yeah, hello. My name is Simone Spuler. And I would like to thank Dr. Donovan for the nice introduction. My topic today will be the DELI 550 and the dual R3 clinical trials, gene-corrected primary human stem cells for therapy in LGMD R1 and R3. I'm from Berlin in Germany, from the Charité Medical Faculty. So you know about limb-girdle muscular dystrophies. These are more than 30 different genetic disorders. There are a lot of challenges to finally find treatment for these progressive, debilitating, and so far untreatable disorders. There is a high genetic heterogeneity. All subtypes are rare. We have a lot of muscles, more than 600 in our body. And there is also a question of how the best route to application to therapy will look like. We would like to treat muscle in a very early stage. And here on this picture, you can see a healthy muscle on the left side. It's a histological picture. On the right side, you can see a dystrophic muscle, which you can appreciate that muscle fibers have been replaced by here in a little bit in a greenish connective tissue and adipose tissue cells. And this is not reversible. So if we want to treat limb-girdle muscular dystrophy, we should start as early as possible. Our approach looks at a gene and cell therapy specifically designed for limb-girdle muscular dystrophy. So we start with the patient. We take very small muscle tissue biopsies from patients, and we purify muscle stem cells from these small biopsies. The muscle stem cells will carry a disease-causing mutation, a disease-causing LGMD mutation, and this will be corrected by techniques that are called CRISPR-Cas or gene scissors, and the repaired myoblasts will undergo a very thorough quality control and then be re-injected into the patient. So this is all based on the knowledge that muscle can only regenerate if muscle stem cells or so-called satellite cells are present. So we all carry these wonderful satellite cells. Satellite cells have a marker which is called PAC7, and the satellite cells have got their names because they are located in a specific spot on the muscle fiber, and this gave them their names. And what happens to these wonderful stem cells? So if there is an injury to the muscle fiber, this could happen through sports, This could happen to trauma, and this can also happen by genetic mutations. And then the satellite cell gets activated. And activation here means that these satellite cells that are quiescent for many, many, many years on the muscle fiber and suddenly undergo proliferation, meaning that they expand. And so there are suddenly a lot of daughter cells, but some of these cells will also go back and become new stem cells, meaning new satellite cells. And this is depicted here. So many of these cells will fuse with existing muscle and repair the injury, and some of these cells will become new satellite cells. These satellite cells are able to undergo the same cycle again once they are injured. So here, this has been known for quite a while, but it has not been possible to render these muscle stem cells towards clinical use. Why is that? Because if you try to purify these satellite cells, it happens that they divide a few times, that they become what we call early myoblasts, but then very quickly they become late myoblasts. They become senescent, meaning they get old, they fuse, and they will not be able anymore to regenerate muscle. So if we want to keep the regenerative potential of satellite cells, we have to catch them as early as possible, and we need to multiply these very early myoblasts, which still have their regenerative potential. So this is where we spent the last 15 years on to find a technique to purify and to amplify these early myoblasts with a retained regenerative potential. And if you want to have a look at these beautiful cells, then this is depicted here. So here you see that in green, you can see desmin, which is the marker of a muscle cell, early muscle cell, and the Ki67 as a marker of cells that are still retained the possibility to divide and to regenerate muscle. So if Ki67 is not present anymore, these cells will not be able to help muscle to regenerate. And then we also have this important transcription factor PAX7, and this is another picture from another culture. And here you can see the desmin in red and the PAX7 in green, and all the cells are still young and very potent for cell regeneration. Here, we show that in preclinical models, and this is, what we finished now before we can go into clinical trials, we can show that mice who tolerate human cells, that the mice, when they receive these cells that I just explained to you, that they can make new muscle fibers of human origin, and that they also can make new muscle stem cells. And these cells will be able to be activated again and to make even more healthy muscle fibers, which is crucial for a long-term repair of skeletal muscle. Okay, so here we focused in our work, after we had established the new technique of purifying and amplifying muscle stem cells, we focused on certain limb-girdle muscular dystrophy types that are frequent, and where we know, and the entire community knows, that certain mutations are much more frequent than others, and that we want to focus on so-called founder mutations. So here, this tells you and summarizes the LGMDs, the limb-girdle muscular dystrophies, that we are aiming to treat in our upcoming clinical trials. So it's the calpain deficient limb-girdle muscular dystrophy R1, or previously 2A, and also the alpha-sarcoglycan deficient limb-girdle muscular dystrophy, now called R3, formerly named 2D. And these are the mutations. So if you look at the next slide, you will see that the mutations we are focusing on in the calpain-3 community, which is the 550 del-A mutation. This mutation makes up to 40% of all patients that are suffering from the LGMD R1 variant. In the dual R3 trial, where we are focusing on the alpha-sarcoglycan deficiency, there are two mutations which absolutely stick out in their frequency, and this is alpha-sarcoglycan 229C to T and 739G to A, and these two mutations make up 60% of all LGMD R3 patients. So here we focused on mutations that affect thousands of patients, although both of them are considered rare, but taken together, the prevalence in the Western world, we know that thousands of patients are affected by these mutations. Interestingly, we know, now with the gene scissor or the CRISPR-Cas, we have now endorsed two different molecular tools to repair the mutation, different for those two trials I'm introducing here. So one is the DELI 550 trial for the calpain mutation, where we use so-called Cas9, and then the dual R3, where we use the adenine base editor. Please let me explain to you a little bit of the molecular details, because this needs to be understood, and it's very important that one is not just, completely ignorant of what is actually being done in a clinical trial. So with the Cas9, the scissor or the gene editing tool cuts the DNA strand, and the DNA strand then needs to repair itself. This is something that nature provides in all of our cells, but here, this is required by the cells and by the muscle. Once the Cas9 cuts the DNA, it needs to be repaired. With the adenine base editor, it's a little bit different, because here, this CRISPR-derived tool, the gene DNA is not cut, but it's only sort of nicked by something that I would call a forceps. One base is eliminated and replaced by another one, and this appears to be a little safer. And this is why adenine base editing is something that is really liked in all of our endeavors to bring gene editing into clinical trials. But I want to tell you that also here for the DELI trial, we know now that also the Cas9 can be a very safe tool to bring therapies to the patient. Now here a little bit more of molecular biology, showing you that with an 80% of editing efficiency in the calpain DELI 550 trial, where we have a deletion. This is why it's called del-A. It's a deletion of a single A, shown here. Here we have a healthy control at this particular spot in the DNA. We see an A and an A, and in 3 patients we miss an A. It's an A and a C, and there should be an A, C, and in all of these the A is missing. And then when we cut at this particular spot by the gene scissor, by the Cas9, then we create a cut, and the cut preferentially inserts the missing A, which is really nice and wonderful to see. So here we have a patient that has an AC sequence, and this has been transformed into an AA. And this happens with 80% editing efficiency. In the other gene, the alpha-sarcoglycan, we can see that a mutation that is 50% coming from mother and from father, we can transform by the gene editing to a 100% exact and corrected sequence and 0% for the wrong sequence. And this is also shown by protein expression and also by transplantation into experimental animals. So here, this is how we start to think about a clinical trial going into patients. And please be aware that the regulatory agencies ask for a lot of preclinical safety studies, and we have to make sure that these gene editing approaches do not affect harmful sequences in the rest of the genome. So here we underwent a extensive protocol of off-target analysis, and that has been completed for all the mutations I've shown you. And we can assume that all of the editing is being safe and does not affect any genes that are absolutely important and essential for healthy organ maintenance. Okay, so now we think that from all these preclinical work, we are ready to start with clinical trials. And so here, first, I need to point out that after the preclinical work, we still need to secure the finances for running a clinical trial. And I think for both of them, the finances have been secured by now. And the next very crucial and very important point is that we need to identify a site that is able to manufacture the drug product that we want. That is a primary human muscle stem cell genetically corrected. And this definitely was a little bit of effort. But now we are done with that, and I can tell you that the site has been identified and that tech transfer and teaching them how to do the procedure has started. And so this is the most very crucial step towards the clinical trial. This needs to be submitted to the regulatory agencies and, of course, approved, and then recruitment can start. So here, I can tell you, of some very important information on the trial designs. This will be a phase 1, phase 2A trial. So the phase 1 means that safety of the patients will be, absolutely crucial and in the focus of the trial. The 2A will also mean that we will do some efficacy measurements. But here, safety of the patients is absolutely the most important aspect. It will be an open label trial. It will have a 12 months duration, but there will be a long-term follow-up because here we are dealing with a genetically modified cell. So there will be a long-term follow-up for at least 10 years, maybe 20. So in the Delhi 550 patient trial, 6 patients will be enrolled in the phase 1. They will all have to carry the 550 del-A mutation, and all the patients need to be older than 10 years, meaning that we will also include minors, coming also from the aspect that I told you about earlier, that muscle that is still in a not too much dystrophic histological modification, that this muscle might regenerate better. The trial can be followed on this website. It's called delhi550.com, and here you can find information on the actual timeline of this trial and also the recruitment procedure. In the dual R3, there will also be 6 patients. They either will carry the SEP39 G2A mutation or the 229 C to T, or both. which is definitely frequent, as we know. And here the same age frame exists. So all the patients need to be older than 10 years. And here, I hope that everybody who listens realizes that it's crucial to know about the genetic information that you do have about the type and mutation of your limb-girdle muscular dystrophy. Okay, and here I would like to summarize and to come to an end. So here my work group has gone through a lot of difficult work, and so this is why this is the stormy cloud above us. But you see the highlight and the sunshine on the horizon, and I think we have really gone a long way. That we really can start now with the clinical trial. We had a lot of support from different agencies, and I really would like to point out the Speak Foundation that has been extremely helpful in advising, also for designing the clinical trial and for encouraging us to go forward. And thank you very much. And with that, I would like to stop.
Okay, thanks for your presentation, Dr. Spuler. My name is Casey Childers. I'm the co-founder and CEO of Kinea Bio. We're located in Seattle, Washington. Today, I'd like to walk you through how we at Kinea Bio are thinking about a possible genetic medicine approach for LGMD-R2, also called dysferlinopathy. And this presentation is really designed for patients and families, so I'm going to stay focused on the big ideas rather than the technical details. My goal here is to explain the problem we're trying to solve, why gene delivery is such a challenging issue in this particular disease, and how our program, we call KNA 155, is designed to address that challenge. So as we go, I'll talk about three main ideas first. What LGMD-R2 is. Second, why the dysferlin gene is difficult to deliver. And third, how a two-part delivery approach might help. I'll also share where the program stands today, which is still in the preclinical stage. So what is LGMD type R2? So LGMD-R2 is a form of limb-girdle muscular dystrophy that's caused by genetic changes or mutations in the dysferlin gene. Over time, that can lead to muscle weakness, especially in the hips, the thighs, the shoulders, and other muscles that are important for movement and daily activity. One helpful way to think about dysferlinopathy is that the role of the dysferlin protein within specific skeletal muscle cells is believed to be contributing to certain cellular functions that help regulate the muscle's ability to maintain normal health. So that factors into the cell's ability to repair after everyday use. So for patients, this matters because the disease is not just about one missing part in isolation. It's about how the muscles maintain themselves day after day. That's why researchers are interested in treatments that aim to restore the production of the dysferlin protein itself. And that really gets at the root cause of the disease. So one of the biggest challenges that we faced is in building a gene therapy for LGMD-R2 is that the dysferlin gene, like the dystrophin gene or the titin gene, is very, very large. And this matters because many gene therapies like AAV or adeno-associated virus as a delivery vehicle, and AAV can only carry a limited amount of genetic material. And this is shown on the next slide here. So even if the therapeutic idea is very straightforward, just replace the missing dysferlin protein, the practical challenge here is that the full dysferlin gene is just too large to fit easily inside a single standard AAV package. So that size problem is one of the central reasons that this disease has been difficult to address with what we would consider traditional single vector approaches. In simple terms, you could think of it as like trying to mail a large item in a box that's just too small. The problem is not the value of what you want to send. The problem is that the package is just limited space. So that brings us really to the next question. If the gene is too large for one package, what can we do instead? And this is where the two-part delivery idea becomes really important. So our platform is something called Simply GT, and it's built around the idea of splitting a large gene therapy payload into two coordinated parts. Instead of, sort of forcing everything into one vector, the goal is to deliver two matching pieces that can work together inside the same muscle cell. So here's the concept between Simply GT. At a very high level, Simply GT is a dual AAV gene therapy approach. And that means that two AAV vehicles are used together, with each one carrying part of the genetic instructions rather than trying to fit everything into one vector. Unlike other approaches to dual vector gene therapy, which combine two pieces of DNA or RNA, the Simply GT system combines the two halves of the dysferlin protein using something called split inteins. The easiest way to picture this is a two-part delivery system. One vector carries one part of the instructions, the genetic instructions, and the second vector carries the other part. So our design is intended to result so that both parts reach the same muscle cell, and the cell can then use them together to produce the therapeutic protein. That's the core idea between the product that we're calling KNA-155. It's not about changing the biology of LGMDR2. It's about finding a smarter, more effective way to deliver the instructions needed to address a gene that's just too large for a conventional, one vector approach. So why two parts can work? So the reason this approach, in my opinion, is very exciting, it's that it's designed around the real physical limits of gene delivery. Rather than treating the size constraint as a dead end, the two-part strategy treats it as really an engineering problem that may have a biological solution. So each vector is intended to do part of the job, and the therapy depends on both reaching the muscle cells successfully. In concept, the cell receives both sets of instructions, processes them, and then forms the therapeutic protein from these coordinated parts. So now I'd like to turn to a movie that sort of visually illustrates this in a form of a cartoon. Okay, so in this animation, we're showing the same Simply GT process you saw before, but this time it's being used for another important muscle protein called dystrophin. These two tiny delivery vehicles, called vectors, each carry part of the instructions for making the dystrophin protein in the same muscle cell. Once inside the cell, these instructions are set up as DNA templates. One template holds the first half of the dystrophin instructions, the other template holds the second half. Together, they contain the full recipe for the dystrophin protein. Then the cell reads these DNA templates to make something called messenger RNA or mRNA from each half. Think of mRNA as a working copy of the recipe that the cell uses to actually make the protein. Each mRNA strand is translated into a protein fragment inside the cell. Each is the first part of the dystrophin, the other fragment's the second part. At this stage, the pieces are made, but they still need to be joined together. And this is where we use these special helper segments. They're called inteins. They guide the two pieces together, help them connect. Then the intein splices themselves out to make one continuous dystrophin protein. So the end result is a single large dystrophin protein that the cell can use to support muscle structure and function. Even though the example I showed here focuses on dystrophin, the important message is that Simply GT process stays the same. Two pieces are delivered, the cell makes the protein parts, then they're joined together into one therapeutic protein. Protein can change, but the underlying approach remains the same. Okay, so of course, carrying the genetic instruction is only part of the challenge. The therapy also needs an effective way to get the muscle tissues that we care about. So this is where the AAV SLB101 capsid comes in. So, in simple terms, the capsid is the outer shell of the delivery vehicle. And in the KNA 155 program, AAV SLB101 is the capsid. It's been specially engineered to target muscle tissue. In fact, it's designed to reduce what we call off-target effects like liver toxicity or illness post-treatment that could make the treatment safer and better tolerated. That's why reaching for a more efficient dual vector technology really matters. So take it all together, Simply GT helps with the cargo size problem, while the AAV SLB101 capsid is intended to guide delivery to the muscle. Both pieces matter because a successful genetic medicine needs to find the right cargo and a reliable way to get the cargo to the right place. When we put these pieces together, KNA 155 can be understood in a three-part concept shown here. First, there's the cargo challenge. The dysferlin gene is too large for a single, typical vector approach. Second, there's the delivery strategy. Simply GT splits the cargo into two coordinated parts. And then third is the targeting strategy. The AAV SLB101 capsid is intended to help bring those vectors to the muscle. So the program is really about solving multiple problems at once. It's not just about the protein we want the body to make. It's also, how to package those instructions and how to deliver them to where they may be needed. It's also important to be clear about where the program stands today. KNA 155 is in what we call preclinical studies as of mid-2026, which means it's still being evaluated before any potential human clinical testing. Preclinical studies are scientific experiments that are done using cells and animals like mice. Nothing has moved from that preclinical stage into the clinical stage when the research studies involve, human participants. This stage is important because it's where researchers gather the information needed to understand whether the therapy is suitable and safe to move forward into human studies. The work can include questions around the delivery, expression, safety, dose, consistency, all of which need to be understood before a clinical trial can begin. For patients and families, the key point is that this is promising work, but it's still early. A preclinical program is not the same as an approved therapy, and there are still many steps ahead before it can become a treatment option. So, what are the key takeaways? To close, I want to leave you with a few key ideas. LGMD-R2 is caused by genetic mutations in the dysferlin gene. A major obstacle to building a gene therapy is that the gene is too large for a standard single AAV approach. KNA 155 is Kinea Bio's effort to address that challenge using Simply GT dual AAV platform technology with the AAV SLB101 capsid for muscle targeting. Big picture goal is to create a therapy concept that can deliver the right genetic instructions in a way that's practical for the disease. And finally, the program remains in the preclinical stage, so it's still an area of active research rather than an available treatment. We will share the work because we believe patients and families deserve to understand the scientific logic behind what's being built, where the field is making progress, and what questions still need to be answered. So, in conclusion, I'd like to acknowledge our key scientific contributors to the program, Dr. Jeffrey Chamberlain and Dr. Hisham Tasfout at the University of Washington. I also want to acknowledge the Jane Foundation team for their guidance, as well as to investors of Kinea Bio, including the Speak Foundation, the Cure LGMD2I Foundation, and the National Institutes of Health, NIAMS, for making this important work possible. So thank you for watching, and I'll now turn this over to Dr. Perlingeiro.
Good afternoon, everyone. I'm Rita Perlingeiro, professor in the Department of Medicine at the University of Minnesota. And I would like to start by thanking the organizers for the opportunity to share our recent progress in our cell therapy program with you today. It has been a while since I last spoke in this event in 2022, so I'm happy to share our recent progress. So I have been working on the development of allogeneic off-the-shelf myogenic progenitor drug product for the treatment of muscular dystrophies. And before I start, I need to disclose I'm a co-founder of Myogenic, it's a university startup company that has licensed the cell therapy technology we have developed. And before I start, I also like to share this important slide. That's the thank you slide for all the people involved in this important project. These are my lab members. I'll be illustrating them along the way. Many colleagues at the University of Minnesota highlighted here in the clinical side. You'll hear from Peter later in the talk, and the people from the clinical grade facility, translational therapy, and many colleagues, as well as colleagues from outside that were very generous with their insights, and of course the funding agencies. So otherwise we wouldn't be able to make the progress we did. Okay, before I discuss specifically cell therapy, I would like to spend a few minutes giving a little bit of background on skeletal muscle and the field of cell therapy in general, since there is a lot of hype on cell therapy and a lot of press, and sometimes it gets confusing to the audience to really discern what's real or not. So I just want to remind this audience the skeletal muscle is a highly regenerative tissue. And I have here on the left, you have a schematic of myofiber, and here images of a mouse muscle. And you can see here on the top is an intact muscle without any injury. You have these beautiful myofibers that are schematized here on the left. And you see these dots here. This is the myonuclei. This is without any injury, and they are just beautifully and steady state. We can come and promote an injury. It can be an acute injury. We can do a cryo injury or even injection of different substances that will damage the muscle. And you can study regeneration. And you can see 2 days after this injury, it's quite a mess in that muscle, right? You have a lot of those beautiful fibers are gone. And there is a lot of inflammatory cells, and you see this goes because we damage the fibers. But after 1 week, and you already see the tissue starting to get reestablished. And by week 2, 15 days after that dramatic injury, you see the tissue is back to normal, right? So there is no space in between the fibers. You see these beautiful fibers here. The only thing that you might appreciate is different. That is the myonuclei that before it was in the periphery of the myofibers, now are in the center. And this is actually an indication of a newly formed myofiber. And this is possible because of the presence of muscle stem cells, which we refer to as satellite cells because they are in the periphery of the myofibers. And here is an old illustration from the first identification, 1961, by Mauro. And here you have an illustration here just to give you a sense of where these cells are in the muscle fiber. So these cells become activated upon injury, be acute or chronic, and promote the regeneration, making new myofibers. Okay, so this is happening every time there is an injury. So what happens with the disease progression of muscular dystrophy is ineffective regeneration. So this is a human unaffected biopsy, the same as in the mouse, right? So you have these beautiful fibers, the nuclei eccentric. And what happened with these genetic diseases in general, all of them, right? You have degeneration because of different mutations with the myofibers, and these fibers with these mutations will degenerate, and the stem cells will become activated, and they will attempt to regenerate. And you might even appreciate in some of these areas in the early stage, you might see some centrally nucleated myofibers. So the stem cells are working to make new fibers to compensate for the degeneration. What happens is the newly fibers are still contain the mutation. So this process of degeneration and regeneration is constant, which brings even secondary defects to the disease in addition to the primary defects. So this constant regeneration in the end leads to exhaustion of the stem cell capacity, and you have persistent inflammation, and you have replacement of the muscle architecture by fat deposition and fibrotic tissue. So what's the point of this slide? What I want to convey really is to give a sense of what's happening. And when we discuss cell therapy, the ideal cell product to be able to deliver to the diseased muscle, functional muscle, which are the healthy myofibers, but also muscle stem cells, or how we like to call satellite cells. So then you would be able to promote long-term production of healthy myofibers. So this is not a new concept, but what kind of cells can you think of? Well, the cells are within the muscle. I thought I would just give a brief slide here in the hierarchy of the muscle. We talk about the muscle fibers, right? And then these are coming from the muscle stem cells that I mentioned to you. And through the process, you have intermediates. And the daughter cell of the satellite cells are the myoblasts. So attempts to develop a cell therapy started many, many years ago when there were studies by Kang and Partridge showing that if you transplant myoblasts, they can promote regeneration in mouse models. And this evolved very quickly to clinical trials in the early 90s. And unfortunately, those clinical trials had controversial or mixed results. But overall, the conclusion for most of these studies were for Duchenne muscular dystrophy, the results were not as encouraging as one would have wished for. And the reasons were more related to attributed to limited regeneration of these myoblasts because they are post-mitotic, they are not the stem cells, they have limited migration and poor survival. There are still strategies or many studies involving myoblasts, but not, mostly not focused for muscular dystrophy, but more self-contained areas like incontinence, for example. So these studies led to the idea, well, it's because the cells are not really the stem cells, and that's why they have not a full capacity to regenerate. So the ideal cell population, as you would imagine, would be the satellite cell or the muscle stem cells, since these cells have the ability to self-renew, make more of themselves, proliferate, and give rise to the daughter cells, the myoblasts, and they will be ideal for regeneration. There are caveats. It's nothing. It's easy. The cells, for obtaining satellite cells, you also need a biopsy, the same as myoblasts, but they exist in low frequency. They are these very specialized cells. They are usually sleeping. They're in a dormant state, and they just become activated to the job after the injury. So these cells exist in low frequency within the muscle, and you can imagine you could expand the cells, but they tend to differentiate it when you put it then in the dish. So they make the daughter cells, the myoblasts. So there are areas of investigation to try to bypass these hurdles. So this is studies with myoblasts and attempts to expand the muscle stem cells are underway in different laboratories. But with these challenges, one can imagine, well, are there any other alternative cell source that could be used for muscle regeneration? You might have heard about these different cell types. So I'll make it simple. You might hear about adult stem cells or the pluripotent stem cells that are cells with an embryonic nature that have the capacity to make any cell type in the body. So within the adult cells, you might have heard about bone marrow infusions or use of umbilical cord blood, as you hear a lot about stem cell tourism. So I will tell you out of the bat that bone marrow cells or umbilical cord blood cells don't make skeletal muscle at their native state. So if you get any cell from the blood or the bone marrow without any modification, any prompting, they will just make blood. So these are not really cell therapies that will promote muscle regeneration, cell replacement. There are mesoangioblasts or pericytes that have been studied for a long time by Giulio Cosso in Italy, and there are many studies with the cell population that can be isolated from the muscle, and these cells have been demonstrated to have regenerative potential. He has tested the cells in Duchenne muscular dystrophy in allogeneic context. the cells were shown safe, and more recently he has evolved his clinical trial, his research into autologous combining mesoangioplasty with exon skipping, and I believe that this trial is open for recruitment in Italy. Another cell population is the pluripotent stem cells, which will be the focus of my talk. That's the focus of our research in the lab. And you might have heard about embryonic stem cells, and that's how the terminology pluripotent really started, because these cells have the capacity, as you can see here, to generate all the cell types in the body, and you can instruct the cells in the dish to make specific cell types and theoretically generate very large numbers of a particular cell type of interest. And, as in our case, you are interested in skeletal muscle. So these cells have unlimited proliferation ability. So you can imagine that there is no issues in generating large numbers of cells. Well, embryonic stem cells really are not, although there are some clinical trials for different diseases using embryonic stem cells. What really advanced the field of pluripotent stem cell-based therapies is the advent of induced pluripotent stem cells that were first described in 2006, 20 years ago, by Yamanaka and colleagues, first in mouse and then in human cells, in which you can reprogram any cell in the body, any somatic cell. Here I'm showing you an example of a fibroblast cell, but this could be actually mated with blood cells or any cell type in the body. You reprogram the cells with these factors and keep them in culture, and the cells will be induced to become pluripotent. And this is shown here. This is fibroblasts, very specific morphology, very elongated. And after exposure to these factors, as you can see, they change morphology. We start seeing these colonies. This is a induced pluripotent stem cell colony, and you can isolate these cells and expand as an induced pluripotent stem cell line. And they are comparable, exchangeable to human embryonic stem cells. So basically, bypassing all the ethical issues that one would have with developing therapies with embryo cells from the embryo. So we work with induced pluripotent stem cells. We started actually with mouse cells, and our technology, just to highlight, uses conditional expression of important transcription factors for the myogenic hierarchy to, as I mentioned at the beginning, the cells can make any cell type in the body. So how I make sure the cells are just making muscle? So one of these genes, if you modify the cells, they will generate myogenic progenitors. And when you transplant and generate very large numbers of these cells, this is really the concept here. And when you transplant these cells, they make myofibers and they make muscle stem cells or satellite cells. This is examples from our early publications. We are looking here in a mouse model of Duchenne, dystrophin contribution, as well as muscle stem cells located in here, and donor-derived fiber with a donor-derived stem cells, improve function. Here we are looking at human iPS cells, human iPS-derived myogenic progenitors. This study was published in 2012. And you're looking here for human dystrophin and another human marker to confirm that the fibers you are seeing are coming from the human transplant. And you see an improvement in muscle function following the local transplantation of these cells into the muscle of mouse model of Duchenne muscular dystrophy in this example, and also the presence of muscle stem cells based on the localization and the marker expression. So how about translation, right? Enough of background. So all this work put us in a situation that you can see two lines of potential for translation. You generated cells from a healthy donor, no mutations. You make your iPS cells, you make the myogenic progenitor cells, and you have an allogenic transplant. Or you generate cells from the patients. You have the patient-specific iPS cells that have the mutation. You can correct the mutations. You have shown in different diseases, limb-girdle muscular dystrophy, we have published in fukutin-related protein, calpain-3, and more recently also dystrophy, and the proof of concept that this is possible. But as we started discussing translation about 10 years ago, many aspects came to our attention that we need to work on, like purification of the cells, even though the majority of the cells will be instructed towards the myogenic lineage. Purification is very important to make sure that the product is homogeneous and safe, and also the manufacture that's compatible for clinical application. And you can actually generate billions of cells that retain the function of the engraftment, and as well as testing safety. So as you discuss these approaches, with allogeneic, you have cells from one donor can be used for a number of patients, and autologous one cell product is applicable to just one patient. We thought that the best way to start the translation of our work would be with allogeneic, because the allogeneic transplantation, as I mentioned before, is mutation agnostic and disease agnostic. So if you prove the cells we have generated are safe and they're actually functional in a clinical trial, these cells that we have produced can be used for a number of diseases regardless of the mutation. And of course, once this strategy has been optimized, it can be used for the generation of autologous cell products, which we actually have also recently performed. So this is what is Myopaxon. It's a myogenic stem cell product that we have derived from iPS cells. We use an iPS cell line generated by the NIH, sponsored by the NIH, by Lonza, that was developed specifically for multiple labs across the country or the world to produce different cell products, to facilitate clinical translation. So we use these cells as actually, these iPS cells were derived from cord blood, but they were iPS. They were not cord blood cells. They're modified, as I mentioned, to make induced pluripotent stem cells. And now these induced pluripotent stem cells derived from blood have the potential to make different cell types. So we use PAX7 particles to genetically modify the cells with PAX7 that allows them to differentiate into myogenic progenitors. And this is just to show the capacity. I'm not going to go into details. This study has been published. You can see it here in the molecular therapy last year. These are the details of the different steps. It takes about a month. But importantly, this is an off the shelf. So the cells are in the freezer. And I want to mention just one specific point is that we have this large master cell bank, that one vial of this master cell bank produces a working cell bank that's immediately after the purification. And we have a large number of the working cell bank, or intermediate, if you might call. And then one vial of this working cell bank you expand for 2 weeks, and one single vial generates 150 vials of 50 million cells per vial. Let me summarize the math here. So one vial of this working cell bank generates 7.5 billion cells of the drug product. So this is so exponential that with this off-the-shelf product that we generate, we could do all the stability studies of this drug product, all the safety studies as required by the FDA, and all the efficacy and as well as safety studies in larger animal models. And we have a significant number of vials in the freezer that can be used for a first-in-human clinical trial and beyond. And if this is proven safe and efficacious, we can still use these vials, or we can go simply back to one of these working cell banks, and in 2 weeks we have more vials of the drug product. So this is significant, exponential in terms of production. Cell number is not an issue. Okay, so you're able to produce a lot of these cells. Are these cells actually behaving the same way? Because now we are not producing just a few millions, but we are producing billions of cells. So as you can see here, we test the efficacy of this drug product called Myopaxon in mouse model of Duchenne, and also in this case, mutation fukutin-related protein. That's a mouse model for limb-girdle R9 or AI. And as you can see, you have significant engraftment in this after the transplantation of these cells. We look at the specific force of the transplanted muscles, and you can see in both animal models, we have an improvement in muscle function after transplantation of myopaxon. And you see also the presence of donor-derived stem cells in the right location with the human markers. So indicating that this drug product is producing muscle grafts. The grafts are functional, and you are also able to, these cells are also able to contribute to muscle stem cells, as we have seen in earlier research studies. This is all very good, but what the FDA is really interested in seeing is safety. So we performed safety of myopaxon in animal models. We tested to make sure that the cells are not going to different tissues or making anything abnormal. And we are making sure the cells don't go to different tissues. We collect all the samples after these different time points. So we follow for histopathology. And here we are looking for signal for human DNA. And as you can see, the human DNA signal is detected only in the transplanted muscle. Well, one issue I had, this was all good news, is that the maximal feasible dose in a mouse, as you can imagine, is not many millions. It's just a few million. And this is very different from the dose would be used in the patient. So I was very excited to collaborate with Melanie Graham here at the University of Minnesota that directs the non-human primate facility. And you're able now to optimize the transplants in a larger animal model. We now, instead of testing few millions, we actually transplant many millions. So for these studies, well, as you can imagine also, the monkey is very similar to us. So there were some issues in terms of detecting our cells, because a lot of the markers are cross-reacting. You want to make sure that we identify the transplanted cells, not the cells from the monkey. So we labeled the drug product with a color. So in this case, it's a red fluorescent protein, so you could find transplanted cells easily. And we are very happy to see that in this pilot study, we saw a lot of red cells, that is the transplanted cells. This is a control, we see no red. And we are also looking, we identify another marker that's specific for human cells, and they co-localize. So we're very excited. So based on these pilot studies, we performed more studies and using exactly the same muscle that will be tested in the first in human clinical trial. That's the EDB muscle that my colleague Peter Kang will share with you in a few minutes. And of course, because this is a xenotransplantation, we are transplanting human cells into the monkey, we had a very strong immunosuppression regimen. And we look for engraftment and also make sure the cells are safe. So there were no changes in weight. The monkeys were stable. And after a month, we look at again, we see lots of red in the transplanted muscles. And you look at TRA185. That is the marker, another marker that's specific for human cells that we could detect in this graft, and they colocalize. We did dose escalation, so we got up to 60 million, which is actually a higher number. That's going to be the first dose in the patient. So we are very happy that we could test this large number of cells for safety. And we see myofibers, we see donor-derived stem cells. And again, we don't see the presence of myopaxon in off-target muscle tissue. So we don't see any tissues, we just see the presence of RFP, in this case, in the transplanted muscle. And we follow the immune monitoring of these monkeys with my colleague, Sab Hamachandra, and we didn't see the development of immune response during this period. So I went by summarizing the roadmap. It took us a long time, but we crossed all the boxes. This is a summarized list, as I mentioned. And Peter Kang led the efforts of IND filing and the submission, and it was approved. And it took us many years to actually manufacture and do all the testing, but we now have an off-the-shelf product that once you prove it's safe in this first in human clinical trial, hopefully we will be able to expand this trial, as Peter will talk about, to different diseases, different muscles. And so I'm very, very excited. And it took us 9 months to get here, but I'm very excited. Now I'll hand the mic to Peter. Thank you so much.
Well, thank you very much, Rita. That illustrates how much work goes into the preclinical studies leading up to a human clinical trial. And as she mentioned, we do have our IND now with the FDA to test this new therapy, Myopaxon, in Duchenne muscular dystrophy. And I'll explain later on how this is relevant for the LGMD community. So I'm really pleased to show this photo from of colleagues from the Muscular Dystrophy Center that we have at the University of Minnesota. And I would like to point out Allison Johnston, who's the second from the right in the front row, who is our primary study coordinator, has done a fabulous job with this study, and many others have helped with this study as well. Molly Stark, Sarah Hilbert, Jenny Meyer, and many others at the university. These are my disclosures, and none of them are directly relevant to this product. So what we have open now for enrollment is a phase 1 study for Duchenne muscular dystrophy. And this diagram just shows what the basic scheme of the study is. So there's a screening period that happens before the dosing. And during that screening period, we do a lot of assessments and tests on somebody who is interested in participating. And if everything goes well, then we do the injection at point 0, which you see a little bit in from the left. And then after that, we take a look and monitor the patient for the first month very closely to assess for side effects. And then we continue to look for side effects, but we don't have to look as closely after the first month. And then at the third month, we perform a muscle biopsy. The reason why we have to do a muscle biopsy is primarily for safety. This is a phase 1 study. This is the first time we'll be injecting this in humans. And so we want to make sure that it's safe and that there aren't consequences of the injection that we didn't anticipate. We really don't anticipate any significant adverse events, but we want to be prepared and we want to monitor for them very closely, of course. So the goals for this phase 1 study are to determine the maximal tolerable dose of these injections of Myopaxon. That's the product that Rita spoke about earlier, into a single muscle. We want to determine the safety of Myopaxon in humans, measure the engraftment, and engraftment means how many of the cells are actually sticking to the muscle and growing and surviving and being healthy. Measure any immune responses. That goes back to the safety issue. Is there an excessive immune response? And then determine feasibility and tolerability. How well are the patients tolerating this? Is this whole procedure something that they are potentially able to go through? And it could be repeatedly. So our current eligibility criteria are patients with Duchenne muscular dystrophy who are 18 years and older. They also are required to be non-ambulatory and to have an intact muscle, the extensor digitorum brevis, which is the target muscle in the foot, which we will assess by MRI. The screening that determines eligibility will include clinical, genetic, and imaging assessments. And then the tacrolimus that I mentioned earlier is an immunosuppressant medication, which people have had a lot of experience with. It's been around for quite a while, but that will help dampen any immune responses. And then, and that will begin before the injection. And then 3 months later, as you saw in the diagram, the muscle will be removed under regional anesthesia. We will be doing ongoing clinical and laboratory monitoring to assess for toxicity and side effects. If this initial dose level is well tolerated, during the course of this phase 1 study, subsequent participants may receive higher doses. And then for future studies, if our phase 1 is successful, we will shift towards dose refinement and efficacy. We will look at whether we can inject other muscles in subsequent phases. We'll be looking more at motor outcome assessments because we're hoping that we wouldn't necessarily have to do biopsies in subsequent phases and that we can then focus more on efficacy for patients and measure are their muscles actually getting stronger after they're injected. What are the implications for LGMD? The Myopaxon product that Rita's laboratory developed is not designed specifically for DMD or any specific kind of muscular dystrophy.
it's derived from a healthy donor. So it should be applicable to LGMD as well as DMD. I'm fully aware that there's a huge unmet need for therapies in LGMD. There are no FDA approved therapies, and we really need some. And we are actively seeking avenues to expand myopaxon research to treat LGMD. I showed the photo at the beginning. I just want to show the names also of people who've been incredibly helpful with this project. And there are others besides the ones on this list, but we rely on a lot of these people at the MD Center, the MDA clinic that we have. And I greatly appreciate the support we've been having for this part of the project from the NIH, the Greg Marzoff Jr. Muscular Dystrophy Center, Duchenne UK, and Parent Project Muscular Dystrophy. Thank you very much for your attention and your time today.
Thank you, Dr. Kang. At this time, we will take another 15-minute break before continuing with the session.
The Speak Foundation would like to take a moment to thank the generous donor who made it possible for us to give away 4 Will Model F travel wheelchairs. We're pleased to announce the winners of this giveaway: Glenn Kalai, Minjin Edward, Jeff Haugen, and Nicholas Compatello. Congratulations to each of you. We'd also like to remind everyone that the Speak Foundation offers several grant programs for individuals living in the United States with any form of limb-girdle muscular dystrophy. You can learn more by reading our LGMD news magazines or visiting our website at thespeakfoundation.com. Thank you.
Welcome back. We will now continue with session 3 on emerging treatments with Paul Martin from Nationwide Children's Hospital.
I'd like to thank the Speak Foundation and the meeting organizers for the opportunity to present our work today. I'm going to share with you a little bit of our data on a new dual gene therapy AAV treatment for LGMD-R9. So these are my disclosures. And so the reason we got into dual gene therapy development is pretty simple. And that is because gene therapies using normal gene replacement, which is single gene therapy, where a normal copy of the gene is used to replace a mutated copy in the disease state, is not always curative. If you take this little guy here and you treat him before he has any disease symptoms with gene replacement, you're going to have a very good chance of having a profound effect on the course of his disease. But if he comes into the clinic at 10 or 20 years of age, when he's already affected by the disease, gene replacement is going to be far less effective. And that we know from a number of clinical trials now in a number of neuromuscular diseases. So our approach is to create a therapy where we can intervene early in disease to improve symptoms, but also treat patients with more advanced disease to reverse symptoms once they've already occurred. And our approach to that is to use 2 genes at once in a single AAV, what we call a dual gene therapy approach. So this cartoon then just shows you the basics of this, where we put 2 different genes into a single AAV genome, one with gene replacement to prevent further disease, and one with a muscle booster therapy to build new muscle mass and strength to reverse pre-existing muscle loss. And we package that into an AAV capsid. This is our gene delivery tool, and that can be delivered in the blood and then travel to tissues and go and deliver that piece of DNA straight into the nucleus of cells where we want that therapy to be effective. And as most of you probably know, gene therapies can be very long-lasting therapies. So the idea of this is that a single treatment can provide for prolonged, decades-long, perhaps lifelong therapy for the patient. And so this kind of approach is applicable to many different forms of muscular dystrophy as well as myopathies, and also is even applicable to some non-genetic medical conditions like sarcopenia, which is the natural loss of muscle mass with aging. So the disease I'm going to talk about is LGMD-R9, which used to be called LGMD2I. This is a limb-girdle muscular dystrophy where patients have mutations in the FKRP or Facoutin-related protein gene. So in a healthy state, dystroglycan normally is glycosylated with a very long sugar called matriglycan, which you can see these little circles here represent that. And that allows laminin to bind to dystroglycan. Laminin is an extracellular matrix protein that wraps around every muscle cell. And then that strength of adhesion can be translated through the membrane via dystrophin to the F-actin cytoskeleton. In dystroglycanopathies, that glycosylation, there are several dozen genes that affect that glycosylation where the glycosylation is lost, and now laminin is no longer able to bind to dystroglycan in the membrane, and that leads to muscular dystrophy. So we have tested mice that have LGMD-R9. These are the P448L mice with a variety of treatments comparing dual gene therapies to single gene therapy alone. And these dual gene therapies have FKRP, a normal copy of FKRP as a gene replacement. And then they have this muscle boosting gene called follistatin or FST to build back muscle mass and strength. And we've compared these in every case to each single gene therapy alone. And we published a bunch of this work a couple of years ago in Molecular Therapy. And the take-home message of that work is that the dual gene therapy, when given early in the disease process, could restore normal ambulation to the same levels that you could see in wild-type mice. It could build new muscle mass and strength in muscles that exceeded what you found in normal mice. It could induce functional glycosylation and reduce muscle pathology. And because of the way we've engineered these therapies, they actually express more gene than single gene therapies alone by engineering them in the dual gene format. So I'm going to tell you a few experiments about early treatment here, where we treated at 1 month and analyzed at 7 months of age. But we've done a number of other treatment paradigms looking at mice that have middle-aged mice that have severe disease and aged mice that have very severe disease. And we're going to publish that work soon as well. And all of that work really points to this therapy being therapeutic throughout the disease, the span of the disease. My favorite assay for studying muscle disease in mice is just walking. So we do a 1-hour walk test in mice. This is be equivalent to the 6-minute walk test in a patient. The main difference being that mice are much better at walking than humans. They have 4 legs instead of 2, and they're very low to the ground. So we have to walk them for longer to see biological changes. And here, these wild-type mice, which don't have a mutation in their FKRP, can all do this walking for the entire hour. An hour is 3,600 minutes, in case you want to do that conversion. So all of these other categories here are the LGMDR9 mice. And you can see LGMDR9 mice treated with buffer alone lose about 2/3 of their ability to walk in this treatment paradigm. Now, if we treat with either single gene therapy alone at the low dose, we don't really see an improvement. At the low dose with the dual gene therapy, we recover about half of the ability to walk. And then with single gene therapy at the high dose, that is also true. But at the high dose for the dual gene therapy, 100% of the mice are able to walk like normal mice in this experiment. So that's what we're looking for. We're looking for a complete recovery of function. And in this case, that's what we were able to achieve with the dual gene therapy. So I'm just going to show you a few more slides. This is a slide showing immunostaining of muscle sections to show you that we can improve the functional glycosylation of dystroglycan. So in these sections, we have stained with dystrophin in green just to mark where the muscle cells are. And then with the 2H6 antibody, which recognizes the functional glycosylation epitope on dystroglycan that laminin binds to. And in wild-type mice, you can see that all of the muscle fibers, these are little circles or each a muscle cell, can express that functional matriglycan. Mice with the disease don't express that sugar, and you can see they're very disorganized because of cycles of degeneration and regeneration. If we put the muscle boosting gene in there, we can get bigger cells, but they don't increase glycosylation. If we put gene replacement in, of course, we can increase glycosylation, but we don't build bigger muscle cells. But the dual gene therapy does both of these things quite well and can recover the functional glycosylation as well as build much bigger muscle cells and muscle tissues. So we've done a number of measures of muscle pathology. I'll just show you 2 here in the diaphragm muscle of these mice. The diaphragm is the most affected muscle in the disease when in these mice. Percent central nuclei is a measure we use to look at a cycle of muscle degeneration and regeneration. So what you can see from this is the mice with the disease, about half of the fibers have undergone a cycle of muscle degeneration and regeneration at the time that we look at this experiment, and that both single and dual gene therapy at the low dose and also at the high dose can prevent that from happening. The dual gene therapy is a little better than the single gene therapy in doing that. Likewise, when we look at the percent non-muscle area within the muscle, ultimately when muscles lose the ability to continue regenerating, they become replaced by fat or extracellular matrix. So the muscle tissue is replaced by non-muscle tissue. And you can see in the diaphragm here, there's about twice as much of that replacement in the LGMDR9 mice as in wild-type mice. And again, that is improved back to baseline with the dual gene therapy. The big difference with the dual gene therapy, though, of course, is that we can increase muscle mass and strength. So this dual gene therapy at the high dose here has increased the absolute force of the muscle by about 50% compared to the untreated mice cohort. And so single gene therapy with follistatin does that as well, but it doesn't improve walking. And you might say, well, why is that? And the reason is because not every muscle responds in the same way in this case. And so you get an imbalance in the muscle growth. You also get an imbalance within the growth within the cells within the muscle. And only when you have disease prevention coupled with that muscle growth do you get a consistent rebuild across the whole body plan that leads to better function. So this dose is about 60% less than the dose of Elevidys or microdystrophin therapy that's given to boys with Duchenne. But that is still a dose that we think is a little too high to be completely safe. And I'm sure that if you're a parent of a patient with LGMD, or if you're a patient yourself with LGMD, you're well aware of the fact that there are a number of adverse side effects to AAV use at very high doses for gene therapy. And this is just a snapshot of the adverse events I took from the FDA website. It is obviously not up to date at this point. Just looking at Zolgensma, which is an AAV9 treatment for SMA, and Elevidys, which is an RH74 treatment for Duchenne. And what you can see, and these are just estimates of the number of patients treated, so these percentages are a little off, but you can see that their serious adverse events are quite high, and patient deaths are also very significant. So, we want to try to avoid that in our therapy, and to do that, we really need to give lower doses of AAV. So one way to do that is to change the capsid that you're delivering the drug in so that it binds better to muscle. And there are several papers that have come out recently showing that that can be done, where these two investigators took libraries of modified capsids, and they found that if there was an RGD peptide in one of the loops of that capsid, that the AAV could go transduce muscle at a much higher level. And so RGDs are well-known tripeptide motifs that Erkki Ruoslahti showed can bind integrins all the way back in the 1980s. And they've been shown integrins have been shown to mediate and potentiate the entry of AAV into cells. And Jeff Bartlett at our institution even showed that RGD put into the capsid as early as 2003 can change the properties of targeting of AAVs to tissues. So we made our own myotropic serotypes in RH74 and compared them to the serotype we had originally done our experiments in. And I'm just going to show you one piece of data from this. So here in purple is data from our original experiments. And what we're showing here is the amount of human FKRP expression from the gene therapy relative to the amount of normal mouse FKRP expression that you would see in a wild-type mouse. So if you have a 1-to-1 ratio here, you have complete gene replacement. So for every molecule of mouse FKRP that you lose, you've replaced it with the therapeutic FKRP from the gene therapy. So that's why we're getting our optimal dose. And this is in muscle. A TA is a tibialis anterior, which is a muscle in your leg. And you can see that these gene therapies, these myotropic vectors, are dramatically improved in terms of the amount of gene expression you can get at this high dose. And our myotropic vectors that we made, we compared also to an AAV that's already been reported in the literature as being a myotropic vector. And basically, even at a dose of 2e to the 12th now, we're still above that magic level of one-to-one replacement. So we can reduce the dose by about 25-fold by just changing the drug delivery device, which in this case is the AAV capsid. And when we do that, the amount of expression in the liver also is far lower than we would have originally seen. And that's true for both genes in the dual gene therapy vector. You see the patterns of these two for FKRP and follistatin are really basically indistinguishable. So we're in the midst of doing proof of concept on this at many doses, basically reinventing the wheel from the data I just showed you. And that data is very encouraging and suggesting that we should be able to dramatically lower the dose of AAV we need to use in patients to treat them with this dual gene therapy. So in conclusion, I have hopefully shared three things with you. By using a dual gene therapy, we believe we can stop and reverse aspects of LGMD at the same time, building new muscle mass and strength. And this should improve therapy for patients who already have significant disease. This approach is applicable to many forms of LGMD. While LGMD R9 is our test case, we believe that this could be applied to many of the LGMD disease forms, and we are working on developing those vectors and testing them now. And by utilizing a myotropic AAV serotype, we should be able to reduce the dose required for patients for optimal gene therapy, perhaps by as much as 25-fold. And this is very important because it will not only improve patient safety by injecting less AAV into patients, but it should dramatically lower the per patient manufacturing cost, which is another real impediment to the broad distribution of these therapies in the world. So I'd just like to thank my lab. Patricia Lamb, Deborah Zygmunt, and Macy Bennett did most of this work, and especially the patient funding organizations that really helped us kickstart this, the LGMD2I Fund and Cure LGMD2I. Without their support, we wouldn't have been able to do this work. We're also supported by the NIH, and we have a Wellstone Muscular Dystrophy Center that focuses on this work, and also the Office of Technology Commercialization at NCH. So I thank you very much for listening. I hope you have an enjoyable rest of the meeting, and I will now pass on the mic to Dr. Floer.
So thank you, Dr. Martin. I would like to thank the organizers of this scientific summit for giving me a chance to tell you about advertent biotherapeutics and our novel approach to combat degeneration and fatty replacement of muscles in dystrophinopathy. My name is Monique Floer. I'm the co-founder and CEO of Advertent Biotherapeutics. Advertent Bio was founded in 2019 with the goal of developing therapies that restore muscle function and improve the lives of patients suffering from a variety of neuromuscular diseases. Our approach targets growth factors to promote healthy muscle growth and regeneration and prevent muscle degeneration. Our most advanced drug candidate, ADA11, is a first-in-class therapy that combats fatty degeneration of muscles, which is a source of functional decline in patients suffering from dystrophinopathy, also known as limb-girdle muscular dystrophy 2B or R2. Fatty degeneration of muscles is the result of chronic muscle fiber injury and incomplete repair, which leads to replacement of muscle tissue by fat over time. And muscle fatty degeneration is strongly linked to functional decline experienced by muscular dystrophy patients. And while muscular dystrophies encompass a heterogeneous group of genetic diseases where distinct mutations cause muscle fiber instability or impaired regeneration, and while distinct muscle groups are affected in different muscular dystrophies, as you can see here, and while the onset, severity, and cardiorespiratory involvement vary, fatty degeneration is found in the muscles of most muscular dystrophy patients. As illustrated here in muscle biopsies from Duchenne muscular dystrophy patients, fatty degeneration appears early on, and eventually fat can replace entire muscle groups. We are developing ADA11 for limb-girdle muscular dystrophy 2B/R2 because there are no effective treatments for the disease. Moreover, limb-girdle muscular dystrophy 2B/R2 is a slowly progressing disease, and inhibition of muscle fatty degeneration could significantly improve patients' quality of life for an extended time. The disease is caused by mutations that lead to the absence of dysferlin, a protein involved in the repair of the sarcolemma, the membrane surrounding muscle fibers. Dysferlin is also important for calcium signaling, ensuring that muscles properly respond to stimulation by incoming nerve signals. As I said, there are no effective treatments for the disease, and corticosteroids used by other muscular dystrophy patients exacerbate the condition and are therefore not indicated for dysferlinopathy. Nevertheless, clinical trial readiness has been demonstrated through several natural history studies, the CAUSE-1 and CAUSE-2 studies conducted by the Jane Foundation. The rationale underlying our strategy is that mitigating fatty degeneration via ADA11 will protect against functional decline, and this is supported by our data from animal studies in a mouse model of limb-girdle muscular dystrophy 2B. However, as I will show you, ADA11's mechanism of action is disease agnostic, and it could therefore be used to prevent muscle fatty degeneration in other limb-girdle muscular dystrophies and adjacent muscular dystrophies. Changes in fatty degeneration in the muscles of limb-girdle muscular dystrophy 2B/R2 patients can be tracked by MRI. Here's data from the CAUSE-1 study showing that the fat fraction in the lower limbs of limb-girdle muscular dystrophy 2B/R2 patients increased as disease progressed. Several things stood out from this study. First, changes in the fat fraction of muscles can be detected within a 12-month period. And secondly, muscle fatty degeneration is strongly linked to clinical functional loss, as shown here in a 6-minute walk test, and also when other assessments for non-ambulatory patients were used. Significantly, patients who initially presented with an intermediate fat fraction saw the fastest progression of muscle fatty degeneration over the 3-year period of the study. And that suggests that as disease progresses, fat formation in the muscle accelerates and eventually outpaces muscle fiber repair, leading to loss of muscle function. Our goal is to extend this period of efficient muscle repair and delay the onset of functional decline. In later stage patients, we expect that inhibiting fatty degeneration will prevent or slow down further decline. Our hope is that ADA11 will have tangible benefits for patients and improve their quality of life and ability to function independently. Having a therapy that halts or slows disease progression would give doctors and healthcare providers added treatment options and alter the standard of care for limb-girdle muscular dystrophy 2B/R2. Hospitals and payers could see significant cost savings due to patients' reduced need for follow-on interventions and shortened post-procedural recovery times. And moreover, ADA11 has pharmaceutical appeal since it leverages a therapeutic modality with a proven track record of generating successful drugs for the treatment of other diseases. Muscle fatty degeneration has never been directly targeted by drug design, although its significance as a disease-promoting pathology has long been recognized. We identified ADA11 as a potent inhibitor of de novo adipogenesis, or fat cell formation, in a cell-based screen where we demonstrated that ADA11 inhibited 99% of adipocyte formation. In his screen, we used a progenitor cell line that can differentiate into fat cells and fibroblasts, forming fat and scar tissue. These progenitor cells share similarities with muscle-resident fibroadipogenic progenitors, or FAPs, the cell population that gives rise to pathologic muscle fatty degeneration. FAPs reside in the muscle compartment and are activated upon injury, and one of the functions is to recruit immune cells that remove necrotic muscle fibers. FAPs also play an important role in stimulating the activation of muscle stem cells, the so-called satellite cells, which then differentiate and repair damaged muscle fibers. However, overstimulation of FAPs can be problematic because of their propensity to differentiate into adipocytes and fibroblasts, and their constant activation in dystrophic muscles leads to muscle fatty degeneration. ADA11 prevents fatty degeneration because its mechanism of action is to inhibit the differentiation of FAPs into adipocytes. Importantly, ADA11 does not interfere with muscle fiber regeneration, and so it could be used by patients to ensure that their muscles regenerate without fatty degeneration. The agent ADA11 is a so-called ligand trap that specifically binds and inhibits two TGF-beta family growth factors. TGF-beta family growth factors are signaling molecules that govern the growth and development of all tissues in the body. The growth factors ADA11 targets are specifically involved in the development of fat cells inside the muscle, leading to muscle fatty degeneration. ADA11 is a modified human protein that can be purified from living cells. And important for our development strategy, ADA11 inhibits both human and mouse growth factors with similar potency. Because TGF-beta growth factor signaling is highly conserved across all mammals, efficacy of ADA11 in mouse models is a strong predictor of its potential as a therapy that halts muscle fatty degeneration in human disease. Lastly, ADA11 can be administered by subcutaneous injection, which will make it easy for patients and caregivers to self-administer ADA11 with a pen or prefilled syringe. We have shown efficacy of ADA11 in a mouse model of limb-girdle muscular dystrophy 2B. The model we have used are the BLA-J mice, which carry a naturally occurring mutation in the dysferlin gene that leads to the absence of the dysferlin protein. This is the model accepted by the field for testing disease-modifying therapies, and the mice were generously provided to us by the Jane Foundation. We have now performed 3 studies in the BLA-J mice, and in the most recent 11-month intervention study, we showed that ADA11, given at 10 mg/kg by subcutaneous injection twice per week, inhibited fatty degeneration of all affected muscles by 50 to 70%. This includes the quadriceps muscle, and here you can see cross-sections of the quads from control and ADA11-treated BLA-J mice, where fat cells are stained with an antibody against perilipin-1 in cyan, and muscle fibers are stained with wheat germ agglutinin in red. As you can see, there is fat infiltrating the muscle of vehicle-treated BLA-J mice, and ADA11 significantly reduced the amount of infiltrating fat. This inhibition of fatty degeneration by ADA11 led to a significant preservation of tetanic force of the quadriceps muscle. By 15 months of age, BLA-J mice have lost about 50% of their maximal tetanic force of the quads compared to healthy wild-type mice of the same age. An ADA11 treatment cut that loss roughly in half. This, to our knowledge, is the first time a direct effect of any agent on tetanic force of a muscle has been shown in this mouse model. In addition, we found that ADA11-treated mice also performed better in a run-to-exhaustion test on a rotorod, where they ran for about twice as long as vehicle-treated BLA-J mice. Together, this data establishes the therapeutic potential of ADA11 as a treatment that inhibits muscle fatty degeneration and preserves function in limb-girdle muscular dystrophy 2B or R2. In addition, we showed in a shorter intervention study that ADA11 effectively inhibited muscle fatty degeneration when Blage mice were treated for only 10 weeks. Significantly, ADA11 inhibited muscle fatty degeneration, although treatment was started after the onset of disease. Further progression of muscle fatty degeneration was reduced by 50 to 60% across all affected muscle groups, including the gluteus muscle, the iliocostalis, and the psoas muscles. These findings are clinically significant because patients are typically seen by a doctor only after they experience noticeable functional losses that are a result of disease progression. Our data indicates that ADA11 could halt or at least slow down any further functional losses. Moreover, in the same study, we also demonstrated that ADA11 basically prevented any new fatty degeneration after an induced injury of the tibialis anterior muscle. We injured the TA by glycerol injection, which is known to induce significant fatty degeneration as the injury heals, and we showed that ADA11 largely prevented any new fat formation. This is again clinically relevant for patients who often experience acceleration of disease progression after an injury such as a catastrophic fall. Our data suggests that ADA11 given directly after an injury could help patients heal without experiencing muscle degeneration and additional functional losses. In a separate study, we also showed that ADA11 prevented new fatty degeneration after an induced injury in older, otherwise healthy wild-type mice, demonstrating that ADA11's mechanism of action is disease agnostic. Therefore, the agent could be used as a therapy to prevent muscle fatty degeneration in other indications, including other limb-girdle muscular dystrophies. To summarize our preclinical development of ADA11, we have demonstrated efficacy of ADA11 in a mouse model of limb-girdle muscular dystrophy 2B/R2, where we showed that early intervention delays fatty degeneration and preserves muscle function. Treatment after the onset of disease reduces development of further muscle fatty degeneration. We further demonstrated efficacy of ADA11 in a mouse model of induced injury, where we showed that ADA11 prevents formation of any new fatty degeneration as the injury heals. We showed this in the Blage genetic background, but also in wild-type mice, demonstrating that ADA11 could work to prevent fatty degeneration in the muscles of patients suffering from other diseases where muscle fatty degeneration drives functional losses. In preparation of clinical development of ADA11, we also performed a pharmacokinetic analysis that indicates that the projected human dosing interval is every 3 to 4 weeks. Moreover, we showed that ADA11 is safe in mice with no adverse events. We performed a preliminary analysis where we assessed safety of ADA11 and found that blood and organ parameters were within normal range when mice were treated for 28 weeks at a drug concentration that was much higher than the intended dose for human patients. There is a clear path for ADA11. Last September, we started GMP development of the agent, which will produce drug material that will be tested in FDA-required dedicated GLP safety studies in 2 animal species. And a first study in rodents is planned for later this year. Our manufacturing process is advanced, indicating that we will be able to produce human-grade ADA11 for a first clinical trial next year. To bridge the mouse to human efficacy, we are currently testing ADA11's ability to inhibit fat formation from patient-derived FAPs. This will go a long ways to demonstrate ADA11's potential as a treatment for muscle fatty degeneration in human patients. A first clinical trial is planned for next year, where we will demonstrate safety of the drug as well as target engagement in patients. To demonstrate clinical efficacy, we envision a 12 to 18 months phase 2 trial with functional gains assessed by the adapted North Star ambulatory assessment and other functional tests. In addition, muscle fatty degeneration will be assessed by MRI as a biomarker and secondary outcome measure.
and this will be complemented by patient-reported outcome measures. The patient infrastructure for limb-girdle muscular dystrophy 2B/R2 is well defined with multiple centers across the US and Europe. And we anticipate a streamlined small cohort phase 2 trial in accordance with FDA guidance for low prevalence rare diseases and commensurate with the limb-girdle muscular dystrophy 2B/R2 patient population. And with that, I would like to end by highlighting BridgeBio's team and our sponsors. Eric Martinez-Hackert and I founded the company, and I have led BridgeBio as CEO through the early stages of drug development, IP prosecution, and fundraising. Eric is an associate professor of biochemistry at Michigan State University, and he serves as CSO. He brings with him deep expertise in TGF-beta growth factor biology with a strong translational focus and a track record of successful drug development, both during his tenure at Acceleron Pharma and at Talapo, a company he co-founded previously. Our strategic advisory board includes experts in business as well as GMP development and pharmacology. And moreover, we're also supported by two key opinion leaders, Dr. Kunkel, an authority on muscular dystrophy, and Dr. Partridge, a leader in muscle regeneration. Both advisors have helped guide BridgeBio's development strategy for ADA11. We have been working closely with the Jane Foundation, who provided the Blage mice to us and helped us in the study design. And we have been funded by the NIH through phase 1 and 2 SBIRs. More recently, we have also been supported by several Michigan-based VC funds as well as by private investors. And I would like to thank you for your attention and send it to Dr. Johnson for the next talk.
Thank you, Dr. Floer. It's really my pleasure to be here, and I really want to thank the Speak Foundation and the rest of the limb-girdle muscular dystrophy patient organizations for hosting such an exciting summit. It's great to see so much work happening in this space and so much hope and promise for the individuals living with these conditions. So I'm going to introduce myself. I'm Nick Johnson. I'm the CEO of Evolera Therapeutics, but more importantly, I'm a neuromuscular neurologist. I've been caring for patients living with limb-girdle muscular dystrophy for several decades at this point, and really hoping to continue that work and care in terms of developing gene therapies for the limb-girdle muscular dystrophies, which is what I'm going to talk about today. So Evolera Therapeutics was founded basically on the premise of developing gene therapies for the limb-girdle muscular dystrophies. That's the hope, goal, and promise of this company. It was founded out of VCU Health and the work that we've done in our center for many years. And really for us, we saw critical gaps in terms of LGMD treatment and access and failing patients. So we know that the limb-girdle muscular dystrophies do shorten the lifespan. They often, individuals often lose the ability to walk in their teenage years, and there's really very extensive treatments. And unfortunately, as we sit here in 2026, there's no available disease-modifying treatment for these individuals. We worked to build off of existing treatments. So, for example, the first generation AAV vectors are used to deliver gene products, but they do carry high toxicities, as we've seen even in the limb-girdle muscular dystrophies, and can target the liver. But that being said, AAV-based gene therapies are, to this day, the best way of delivering genetic material into the skeletal muscle. And we really founded the company with trying to be nimble and make sure that we are frankly taking care of patients. And so, we really work to prioritize patient input in terms of the drug development process. So our solution is to develop one-time gene replacement therapies for limb-girdle muscular dystrophies. We are using a next-generation vector, as I'll speak to, that detargets the liver and delivers the full-length genetic material to the muscle cells to allow for protein production to be restarted and really treat the disease at its source. So these are safer muscle-targeted next-generation therapies, and we know that these therapies can be given to patients across 18 different LGMD subtypes. And we believe that we've worked hard to align our stakeholders in terms of a trial-ready population. So I'm going to speak through our first program here, which is LGMD2C or 5, which is due to loss of function of the gamma-sarcoglycan protein. As many of you know, this protein is part of the dystrophin-associated glycoprotein complex, which is a mouthful. And when you lose that protein, you destabilize the complex and you disrupt membrane integrity. And you can see it here in the figure as the Greek letter there for gamma, but you can also see beta, delta, and alpha all present, working together as part of the sarcoglycan protein complex. We know that typical onset's between 3 and 10 years of age, and then often people lose the ability to walk before 20 years of age. And that also leads to respiratory problems and heart problems, and unfortunately, it does shorten the lifespan. So what we're using is something called an AAV myocapsid. So this is a historic, or this is a capsid that was engineered to specifically avoid the liver. And you can see this data here compared to, like, say, an AAV8, where you can see that MYO2 has substantially less expression in the liver, and actually, because of that, ends up with more protein expression in the skeletal muscle. Think about like a numbers game. So if you are not getting soaked up by liver, there's more of it to deliver to the muscle. And we, in this set of experiments, we used a missense mutation that's commonly seen in individuals living with a gamma-sarcoglycanopathy. But this is a mouse, and you can see that we gave doses at 1E13, 5E13, and then 1E14 viral genomes per kilogram, and then we measured the phenotype and then looked at a number of molecular markers, which you can see there. So first, at the top here, you can see this is the tissue delivery here for the skeletal muscle at the top, and you can see that all of the tissues analyzed did show a direct relationship between the dose delivered and the detected genomes, with good transduction at the 5E13 and most, especially at the 1E14 doses, in terms of the quadriceps, TA, and heart. And as you would hope or expect, you can see that really there's very little delivery in terms or transduction in terms of the liver, kidney, or lungs. So this is what you want to see, and we're hopeful that using this second generation vector, we're able to avoid some of the pitfalls that you might have seen previously in terms of causing the liver disease, liver toxicity. This is a different way of looking at this, where you can see that the delivery of the gamma-sarcoglycan protein leads to robust membrane localized protein expression in these mouse models. So two muscles shown here, the TA, which is a muscle in the calf, and the quadriceps, which is your thigh muscle. And you can see in the wild type, you see that nice red ring that kind of goes around the skeletal muscle. If you don't give anything, you can see that it doesn't really express very much at all. And then you can see as you go up in dose, and especially again at that 5E13 and most especially at that 1E14 dose, you can see good tissue delivery using this vector. And most importantly, of course, so we're getting it to the right place. Does it do anything? Well, here you can see both in the quadriceps and the TA that the total gamma-sarcoglycan levels reach near wild type levels in mice, muscles from mice treated with this dose. So there you can see the wild type and the blue on the far left, and then the dose levels there at 1E13, 5E13, and 1E14, and you're getting good protein expression in the skeletal muscle of these tissues. Interestingly enough, it does improve muscle endurance during wire hanging assay. This is actually seen, we can see this really actually at any dose. And for us, we are continuing to follow the great work that was done in this Brain article, where if you followed individuals retrospectively or prospectively, and they had greater than 30% protein expression. They did have a milder disease course than those with less than 30%, which is what you can see here in that paper. And so for this program, the toxicology studies are ongoing, and then the clinical grade manufacturing is also ongoing or nearly complete. We have a second program in LGMD2D, which is focused on restoring alpha-sarcoglycan protein expression. So we're using the same capsid and promoter as in 2C, which is what you can see there. And again, you can see the same exact experimental design using mice and then measuring their grip strength and then looking at protein quantification. And here you can see in this set of experiments, very similar data in that it improves the wire hang for these patients. And then if you're looking at the protein, or excuse me, at the skeletal muscle, which is that kind of bottom right set of graphs, you can see that you have pretty healthy skeletal muscle in these wild-type mice in the far left, and then you can see the signs of muscular dystrophy in the middle panel with the central nuclei, the fibrosis, the necrosis, and change in fiber size, and you can see that that also improves as you treat them with alpha-sarcoglycan. Same set of experiments, same idea. So you can see here that we have essentially normalized protein expression in post-treatment in these animal models, and likewise you can see, particularly on the bottom corners, if you deliver the gene, that you can see protein expression come back after treatment, which is not present in that kind of middle panel there. So we're happy and pleased to see that this replicates what we see in the gamma-sarcoglycan protein, in that you see very little hepato expression whatsoever, but that you get good protein, good transduction, and good protein expression in skeletal muscle in this, and that are some relationship in terms of functional improvement. So this program is about 6 months behind. So we're just getting our way through the dose range finding studies in this, and like I said, the clinical trial for alpha is about 6 months or so behind gamma. So for us, we have taken a very efficient approach in terms of getting our way through to getting these drugs into clinical trials, and then as quickly as possible getting them approved in such a way that they're really accessible to patients. And so this is kind of our first program here, and then you can see that the additional programs down at the bottom, so that we're working our way through our CMC toxicology and vector studies. We will be in clinical trials by the end of this year. Those clinical trials are designed to be very efficient and work their way through. So we're hopeful that by 2028, with that first program, we can be looking at our first biological license submission, and then approval after that. And as I mentioned, the LGMD-R3 program is about 6 months or so behind. So there we're looking at a clinical trial start sometime in the summer of 2027. We're fortunate in limb-girdle muscular dystrophy that there are about 18 to 20 or so different subtypes where you can basically insert the entire gene into the viral vector, and we are working on doing that for the third subtype that we're working on, and we'll continue to work from that time point. Couldn't do this without our team here. So you've been listening to me drone on here for the last few minutes, but I am an active practicing neurologist at VCU. I see patients there. I direct the Graswell Gene Consortium and a lot of experience in terms of clinical trial design and execution. Kevin Passarella is our business guy, who's been helping us and has a lot of success in that area. Melissa Hale has been working on the assay development. She's a PhD scientist and our chief technical officer, but largely focused on myotonic dystrophy, but muscular dystrophies in general. And then Ellie Carroll is a chief science officer who's an AV expert focused on developing safe and effective gene therapies, and has had 2 decades of experience in the muscle disease, and just really primed and ready to help us lead these programs forward. We have great partners. Again, it's a very small team, but couldn't do this without the supporting village. So Andelin Biosciences is our CDMO, so they're making the vectors themselves. Pearson's making the genetic material. Bioagilitics has been helping with assay development. Attentive Sciences, which is actually based in the UAE, has been helping us with the toxicology studies. Biotech Logic is supporting all the regulatory processes. And then Thermo Fisher, or PPD, is the CRO that's conducting the clinical trials on our behalf. We have a great group of ecosystems that's really been helping us. So in terms of advisory board, Jim Dowling, who's got a lot of gene therapy expertise in childhood muscle diseases. Saul Carpin is a pediatric hepatologist. So really doubling down on making sure that we do this in a safe way, particularly as it relates to liver toxicity. And then Petra Kaufman has been working in drug development in genetic neuromuscular diseases for at this point several decades. And we're leveraging, all of you, actually, frankly. We've got a great partnerships with the Speak Foundation, Cure SCG, and then the LGBT Foundation, as well as our other academic colleagues who will help us design and conduct these clinical trials. So I think in summary, for us, we're thinking about Evolura Therapeutics as transforming gene therapy for the limb-girdle muscular dystrophies. This is a single gene therapy for muscle restoration. We're very nimble and accelerated in terms of a 24-month path to patient treatment per cohort. We've got stakeholder support, and it's been a pleasure to meet with so many patients, individuals, and family members living with these diseases. I see you, and I'm certainly anxious to get these trials into therapy. And then, we're fortunate to be working on a modular system so we can scale this efficiently. We know from our interactions with the FDA that we will be able to not have to go through all the steps for every subtype, where we can really go directly from kind of the manufacturing process into clinical trials. So we can, because we're using a platform, we can really be quite efficient in terms of working our way through subtypes. And from work that we've done on the academic side, we also know that these clinical trials are going to look roughly the same, if not identical, across different subtypes. And with that, I would like to thank you for your time and attention, and hope you enjoy the rest of the conference and meeting. And I will pass it on to Dr. McNally.
Thanks, Nick. And I'm Beth McNally. I'm at Northwestern, and I represent also IKEKA Therapeutics. And I'm going to talk to you today about using genetic modifier signals to develop new muscular dystrophy drugs. These are some of my disclosures, and I am founder of IKEKA Therapeutics. So fundamentally in muscular dystrophy, the problem is that muscle gets replaced by fibrosis. And instead of seeing all the beautiful individual fibers here, we see in milder and severe forms of disease, there's just replacement of the tissue by fibrosis and in some cases fat. And this is common across all forms of muscular dystrophy. And what we've been trying to develop is approaches that address that common problem and that can be used across multiple forms of muscular dystrophy, including the limb-girdle muscular dystrophies. As we know, there are many different genetic causes of muscular dystrophies, including the limb-girdle dystrophies. And what I'll be talking about today is relevant to these sarcoglycans, which are part of the dystrophin complex. But as I said, fibrosis is an important part of many different forms of LGMDs, and so we think this really can be applied across many different forms. This work started many years ago when we identified a single mutation in gamma-sarcoglycan, which is a dystrophin-associated protein. It is known as LGMD2C, or now R5. Because patients had that same single mutation, it allowed us to see the variability in terms of how it could present across many different people, even though they had the same different form of disease. Some individuals stopped walking by the age of 10, others would still be ambulatory by the age of 29. And so that led to the the concept that there must be other genes that are contributing to how the disease manifests itself. And so we set out many years ago to identify those genetic modifier genes. We did it at the time using mice. You can do this now using humans because there's a lot more human data, but we did it in a form of disease in the SGCG null mouse, which is the gamma-sarcoglycan null mouse. We found that the DBA2J strain of mice made it much more severe, causing much more fibrosis to develop, and also leakage of a dye called Evans blue dye into the fibers, which is indicative of the surface of the cells being very, very leaky. We also found that there were a couple different strains, the 129 strain and others, that made the disease milder. And if you interbreed these mice, you can build a great big mouse family tree that looks like this, and you can use that to identify genes that make more fibrosis or more Evans blue dye. And so we did what's called genome-wide mapping. We look for signals across all the genome. Across the bottom here is 19. You see 1 to 19 because mice have 19 chromosomes. And you see there's a great big signal up here on chromosome 7, which was looking for modifiers that were regulating how much of the Evans blue dye was going into fibers. We did the same study looking for how much fibrosis was forming, and we got the exact same signal, also on mouse chromosome 7, with a very, very high LOD score statistical significance. And that showed us that it was this region that was responsible for modifying both the amount of membrane leak as well as the amount of fibrosis. We used sequencing approaches to identify the causative gene, and in this case, we identified LTBP4, or latent TGF-beta binding protein 4, as the modifier gene on mouse chromosome 7. What we found was that there were strains of mice that had a deletion in the hinge region where it was missing 12 amino acids, and there were also strains of mice in which there was an insertion of 12 amino acids into the hinge region of LTBP4. And the presence of that hinge insertion was associated with milder fibrosis and milder membrane leak. We know that TGF-beta is a very important modifier of disease because it is a master regulator of fibrosis. And the way the LTBPs work is they sit outside the cells in the extracellular matrix where they bind to latent TGF-beta and they regulate its first step of activation where it undergoes an activation step to ultimately become active TGF-beta. And so the role of LTBPs is really to keep TGF-beta from being too active. We also studied human LTBP4. So what I showed you before was in mice where there's an insertion-deletion change in this hinge region, which is full of prolines. But what we found in humans was there were 4 different positions, relatively common in the population, where you could have a valine or 3 threonines. Alternatively, there were humans that have 4 different positions, isoleucine, 2 alanines, and a methionine. And most individuals carry either 1 copy of the VTTT or 1 copy of the IAAM or homozygous for both. And what we found was having 2 copies of the IAAM led to much milder disease. Before we got to that, we actually studied that the presence of the VTTT or the IAAM version of LTBP4, it was in fact this version that has the isoleucines in it that binds much more of TGF-beta. So it underscores that basic mechanism that if you have the IAAM version, it is more tightly binding to latent TGF-beta and keeping more of it inactive. So to begin to look at whether those human changes had any correlation with disease, we reached over to work with Kevin Flanagan and study a large cohort of Duchenne muscular dystrophy patients because it's very hard to put together large cohorts of LGMD patients. There are just not as many different forms of disease. So we did this in Duchenne muscular dystrophy, and it demonstrates that, in fact, this modifier acts across multiple different genetic forms of muscular dystrophy, including the limb-girdle dystrophies in which we first did the genome mapping. So here is showing you in the Duchenne population, if they have the protective version, IAAM, they are this red line here, which shows that these individuals walk longer, on average about 2 years longer than what they would have otherwise. So this is demonstrating that genetic signals in this gene also correlate with improved outcomes in muscular dystrophy, in this case, Duchenne muscular dystrophy. So this LTBP4 IAAM genotype is protective in DMD. It's now been shown in multiple other studies, mostly all using Duchenne patients because, again, it's easier to get those patients. An interesting observation that we made was if the patients were taking glucocorticoid steroids and they had the protective allele, that was the best outcome. I will say that it's only about 10% of the population that carries IAAM homozygously. So overall, it's a pretty small percent that naturally receive protection from having LTBP4. But it still means that an antibody that I'll show you in a second that can protect LTBP4 could be useful across all different forms of muscular dystrophy, irrespective of what the LTBP4 genotype is. We also went on to just ask the question of, can you overexpress LTBP4, and does that make disease better? So this general idea of, you put more LTBP4 there, does it bind more latent TGF-beta? And that's in fact what we found. We did this in the background of the MDX mouse model of Duchenne muscular dystrophy, where you can see that there was less fibrosis present and that the muscles were also stronger. So here's a measure of fibrosis and a measure of the muscles being stronger. We also noticed that the muscles were bigger in these transgenic animals, and the TGF-beta family is shown here on the left, where TGF-beta 1, 2, and 3, all three forms do bind LTBP4, but we also assessed whether myostatin, the negative regulator of muscle mass, also had any differences. And what we saw here is in the transgenic form of overexpressing LTBP4, there was very little of the active form of myostatin present, which we believe explains why the muscles also can grow bigger when you modify LTBP4. We went on to show that LTBP4 not only binds TGF-beta, which I'm not showing you here, but it also does bind to myostatin, which we can see the full-length form binds myostatin, as does this N-terminal form. So this maps myostatin binding to this region of the molecule and TGF-beta binding to this region of the molecule. The general idea of stabilizing LTBP4 with an antibody comes from the concept of, if we make an antibody that's right to that hinge region, the region that the mouse genetics showed us was very important, that can stabilize the latent form binding latent TGF-beta. And that will stabilize what we call the closed conformational state, where latent TGF-beta and potentially myostatin are both modified by that. Almost more than 10 years ago, we actually did some studies using an antibody and showing that it could protect LTBP4 from proteolysis. And so we've used that as the basis of developing better antibodies, which Dr. Demonbreun will tell you about next. But in the last minute, I just want to say we started out by mapping lots of modifiers across all the genome. I told you about LTBP4 today, and in just 2 more slides, we did also map another modifier called annexin A6. Annexin A6, this is a muscle fiber that's been wounded right here, and annexin A6 is the molecule that actually forms a Band-Aid or protects this resealing, promotes resealing of the laser-induced wounding. And so we've also done some work generating recombinant annexin A6, showing that it can help reseal muscles, and we're optimistic that this also could be considered a gene-agnostic approach for treating muscular dystrophies. So our goal as ICAICA is to develop approaches to treat muscular dystrophies that don't depend on the primary gene mutation and can be used across multiple different forms of disease. And in the next talk, you will hear from Dr. Demonbreun. She will tell you about our development of high-affinity anti-LTBP4 antibodies to treat muscular dystrophies. Thank you.
All right, thank you very much, Dr. McNally. And now I am going to tell you about the development of anti-LTBP4 treatment for muscular dystrophy. I'm Alexis Demonbreun. I'm an associate professor at the Center for Genetic Medicine, the Department of Pharmacology at Northwestern University, and I'm the chief scientific officer of ICAICA Therapeutics. So our lab is interested in identifying genetic modifiers as therapeutic targets for muscular dystrophy. Previously, we performed a genetic modifier screen in a mouse model of limb-girdle muscular dystrophy 2CR5. We performed a genome-wide screen and quantitative trait loci mapping, and we identified a region on chromosome 7 that correlated with membrane permeability and fibrosis, which are two key aspects that drive muscular dystrophy. So in this region contained the gene LTBP4. So we are interested in LTBP4 because LTBP4 was a known regulator of TGF-beta. LTBP4 binds and sequesters latent TGF-beta, which reduces TGF-beta activity. This is important for muscular dystrophy because hyper-TGF-beta activity contributes to the dystrophic phenotype in many muscular dystrophies, including limb-girdles and Duchenne. So TGF-beta is a master regulator of fibrosis, where high levels of TGF-beta drive fibrosis formation, and TGF-beta also inhibits muscle differentiation. So we thought if we could regulate TGF-beta, we would be able to reduce fibrosis and improve muscular dystrophy progression. So the mechanism of action for our therapy was actually identified from the genetic screen. In the LTBP4 hinge region, we actually found an insertion polymorphism in the protective strain. So this insertion of the hinge actually prevented the full-length LTBP4 from protease cleavage, as you can see in the immunoblot in the bottom right, compared to the mouse model that had the severe form of LTBP4 with the deletion in the hinge. So by eliciting this protease cleavage, you are now releasing TGF-beta and driving fibrosis formation. So this gave us the hint of what we would like to do to develop a new therapy. We thought if we actually could design an anti-LTBP4 antibody directly to this hinge region, the antibody would bind the hinge and slow down the cleavage by proteases of LTBP4, thereby sequestering TGF-beta and mitigating disease progression, specifically reducing fibrosis and promoting muscle regeneration. So LTBP4 itself is found in skeletal muscle and in heart. It's found in the extracellular matrix of these tissues and also on the surface of muscle cells. This is important because antibodies generally on their own have a hard time entering cells. So since LTBP4 is located on the exterior surface, antibodies administered systemically is able to reach the target with systemic administration. So we performed an antibody screen and ended up with a lead anti-LTBP4 antibody. And so we wanted to confirm that this antibody actually bound both mouse muscle and heart muscle, since in limb-girdles and Duchenne muscular dystrophy, both of these tissues are affected, and we would like our therapy to target fibrosis in these two key tissues. So as you can see on the left, an immunoblot, we actually can see that LTBP4 antibody binds full-length LTBP4 in skeletal muscle and in heart lysates. In the middle panel, you can see in immunofluorescence microscopy that the LTBP4 antibody binds skeletal muscle on that exterior face in a costameric pattern, as I mentioned, and then it also is localizing to the LTBP4 found in the extracellular matrix. Importantly, we wanted to confirm our antibody could protect LTBP4 from cleavage, which was our main mechanism of action. So we performed a cleavage assay where we incubated full-length human LTBP4 protein with and without our LTBP4 antibody and subjected this to proteases. You can see in the immunoblot at 160 kilodaltons that the LTBP4 antibody protected the protein from cleavage compared to an IgG control antibody and PBS. And so knowing that our antibody bound the target and protected from cleavage, we wanted to evaluate efficacy in vivo. So we performed a short-term assay where we injected MDX mice once weekly with antibody, and then performed an eccentric contraction muscle assay. In this assay, the muscle is actually subjected to lengthening while the muscle simultaneously is contracting, and this induces muscle tears.
if our antibody was to stabilize the muscle membrane, which was one of the features we identified in our genetic screen, we would actually see that this muscle should be stronger and have less force loss because it is stabilized. And that's exactly what we saw in the right graph. The muscle treated with LTBP4 antibody had less force loss, meaning that it actually had improved stability and function. We then looked to see in this muscle if LTBP4 actually elicited this benefit through TGF-beta signaling. And we evaluated this through PSMAD, which is a downstream signaling molecule of the TGF-beta pathway. So in the top panel, we confirmed in green that the mice injected with LTBP4 did have LTBP4 antibody that localized to the skeletal muscle. And in those same fibers, we saw a reduction in PSMAD shown in green in the myonuclei in the center of the muscle. And so to further evaluate the effect of LTBP4 on sarcolemma stability, we performed another assay. So in this assay, we isolated myofibers from the mouse. These myofibers are alive. We incubated these myofibers with a dye, this FM464 dye, that marks regions of injury. So we took a laser and made a little micro injury lesion at the membrane. And if the muscle is stabilized and has enhanced repair, we should see a reduction in FM464 fluorescence. So the red area should be smaller. So on the right panel, you can see that myofibers incubated with the LTBP4 antibody did indeed have about a 50% reduction in FM dye area, suggesting that the LTBP4 antibody did stabilize the muscle membrane. We then wanted to perform an in vivo assay that looked at the effects on membrane stabilization and leak. So we injected mice with another dye called Evans blue dye. This dye is only taken up in leaky myofibers, which once again, this was one of the traits that identified LTBP4 in our genetic screen. And so the mice were injected with dye and then subjected to an acute injury. You can see that the mice that were treated with vehicle had a very large injury area with a lot of red fluorescence. And then the mice treated for 2 weeks with anti-LTBP4 antibody actually had a reduction in injury area as well as a reduction in dye uptake, illustrating that the antibody reduced the muscle leak in the MDX model. When we performed the same injury assay but allowed the muscle to recover for 7 days, we saw something very similar, that the LTBP4 antibody reduced the injury area, promoting recovery from injury by about 50%, which was super, super exciting to us. So then, importantly, we want to understand the efficacy of LTBP4 antibody in reducing muscle fibrosis. So in the MDX model, fibrosis accumulates over a long period of time. So to evaluate the fibrosis in natural disease progression, we actually performed a 6-month study. So we injected MDX mice once weekly for 6 months and then looked at the fibrosis accumulation by Mason's trichrome, which fibrosis would be shown in blue, and Sirius red, which fibrosis is shown in red. And you can see in the middle panel highlighted in purple that the mice treated with LTBP4 antibody had reduced fibrosis compared to the IgG control. When we quantified this using a hydroxyproline biochemical assay, we once again saw that there was a significant reduction in fibrosis accumulation in skeletal muscle with our anti-LTBP4 antibody. And you can see in the graph that we have one antibody that has an M and one antibody that has an H. Those are the exact same LTBP4 antibodies, just with 2 different backbones, one for mouse and one to human. And both of these antibodies did show similar reduction in fibrosis, which really gave us the belief that this was a true effect.
And so then in these same mice, we actually looked at muscle force and muscle size and saw that LTBP4 antibody increased muscle size and also increased muscle force. And so finally, in Duchenne muscular dystrophy, steroids are used as standard of care. So we were interested to see if a combinatorial approach where mice were treated with both anti-LTBP4 and prednisone could provide significant benefit. So we dosed MDX mice with either antibody alone, prednisone alone, or both our anti-LTBP4 and prednisone once weekly for 4 weeks, and then performed that similar eccentric contraction injury as I discussed previously. And as you can appreciate in the graph, we saw that mice treated with both anti-LTBP4 and prednisone had a significant reduction in force loss, meaning that the muscle was actually further stabilized and had further enhancement of muscle function with co-treatment. So this is really important as different therapies are coming out in the clinic that we would love to be able to, of course, dose with our LTBP4 antibody, but we also envision that this could be given as a co-therapy with other emerging therapeutics. And so in conclusion, what I've shown you today is that we have an LTBP4 antibody that binds LTBP4 protein in muscle and the matrix. The antibody blocks protease cleavage, thereby reducing TGF-beta activation. It promotes increased sarcolemma stability and recovery from muscle injury. The LTBP4 antibody treatment reduces fibrosis and improves muscle function in DMD. And the LTBP4 antibody treatment acts synergistically with steroids. And so with the similarity between DMD and LGMD and the fact that LTBP4 was identified in a model of LGMD, we are really excited to start evaluating the potential of this antibody therapy in the mouse models of LGMD. And what's also exciting is that using this mechanism of action, the LTBP4 antibody is gene mutation agnostic. And so we hope that this will be able to provide benefit to many patients across diseases as well as across mutations. And so with that, I'd like to thank Dr. McNally, who's done this work with me, and everyone who contributed to this work. Of course, IKECA Therapeutics, as well as our funding through NIH and the DoD. And so now next, we will move to our next session with Dr. Mozaffar.
Hi, I'm Tahseen Mozaffar. I'm a professor of neurology and pathology at University of California, Irvine, and involved with the Neuromuscular Center and the Muscular Dystrophy Clinic at the university. I am part of the limb-girdle muscular dystrophy Centers of Excellence through the Muscular Dystrophy Association and very involved with LGMD R9 therapeutic development. And today I'd like to talk to you about our recent efforts in terms of developing a gene therapy for LGMD R9. As you know, LGMD R9 is caused by mutations in fukutin-related protein. It is an autosomal recessive disease that impacts several thousand individuals in America. And age of onset is variable, generally from adolescence to early adulthood, and is characterized by progressive muscle weakness, loss of independent mobility, resulting in highly compromised quality of life, and early death due to loss of respiratory and cardiac function. LGMD-R9 is due to missense mutations in the FKRP gene. A more severe disease, which is congenital muscular dystrophy MDC1C, is caused by having no FKRP protein. FKRP in turn encodes for a protein called ribitol 5-phosphate transferase that is required for appropriate glycosylation of alpha-dystroglycan. Alpha-dystroglycan is a membrane protein in the muscle, and it needs to be glycosylated, which means extra sugar molecules added to it for its stability and integrity. The missense mutations in FKRP are responsible for the LGMD-R9 because it causes hypoglycosylation of the alpha-dystroglycan. In the absence of sufficient amounts of alpha-dystroglycan glycosylation, the interaction between muscle cells and the extracellular matrix is disrupted, driving the progressive loss of muscle function in LGMD-R9 patients. Due to these features, LGMD-R9 represents an ideal candidate for gene therapy and even pharmaceutical therapeutic approaches. The presence of residual FKRP enzyme activity has led to alternative approaches to increasing this activity by elevating the concentrations of the FKRP substrate CDP ribitol. One of these approaches is a dietary supplementation with ribitol, and that has gone through a phase 2 and is currently in a phase 3 study. The molecule is called BPB-418 and is developed by a company called BridgeBio. We've been very involved with both phase 2 and phase 3 studies and have a lot of experience with it. Given the current absence of any other therapy that can control the progression of LGMD-R9, it is highly anticipated that BB418 or ribitol will receive accelerated approval and be rapidly adopted as standard of care when it's approved. However, there are efforts going on with gene therapy. There are definite advantages of using gene therapy because you can deliver the correct gene in its entire size to the cells, and that gene can then obviate the need for any other treatment. It will start producing the correct amount of glycosylation. However, the first generation AAV viruses, which we are using for gene therapy, have limitations. The biggest limitation is that they have a predilection to go to the liver. Even with the slightest amount of gene therapy, most of it is given to liver, and it really needs to be directed to muscle and other areas with some effort. Because to get to the muscle, you need a higher dose of virus. There is a risk of liver toxicity, and there's a risk of heart toxicity. And those are some of the limitations and some of the challenges dealing with gene therapy. As I said, the gene also needs to be better targeted. It needs to be really targeted to the muscle more and heart more than the liver. Therefore, we need better gene therapy vehicles with better efficacy and much less dosing of the vector, which is the virus. So Cure Our Rare Disease is collaborating with a number of institutions, including UC Irvine, Virginia Commonwealth University, Yale, and Heidelberg University in Germany. They have chosen a different gene capsid or gene therapy molecule. This is AV-MYO2. MYO refers to its predilection for muscle, and it's a capsid that detargets the liver. So very little of it actually gets to the liver, and most of it gets to where it's really needed the most, which is the muscle. And I'll show you some data on that. Cure Rare Disease teamed up with UC Irvine and applied for a grant through the California Institute of Regenerative Medicine. It was funded. It was a $7.4 million grant. And the purpose of this grant is to fund the program through an IND submission. So all the preclinical work, including the work in the outside the animals as well as in animals, is going to be done through this funding. And this funding will hopefully lead to approval of a gene therapy trial that can be done in patients with FKR, with the LGMD-R9. The product that they have developed is now named CRD003, And I'll show you some data that should suggest that it doesn't go to the liver, and number two, it also mitigates the risk of toxicity to the heart in the initial non-clinical studies that have been done. So this is the first graph that I will show you. This graph is trying to compare the three different types of viruses, AAV8, AAV9, and AAV8 and AAV9 are the two most common viruses that are used for gene therapies currently. Most of the currently approved as well as experimental gene therapies are using AAV8 and AAV9, whereas the one on the right, extreme right, AAV Myo2, is what we are proposing to use in this CERM-funded trial. And this has a muscle specificity, so you can actually see that even with the smallest amount of doses, there is very high expression in the muscle. And in the bottom, the key tells you which particular muscles we looked at. So we looked at biceps, the hamstrings, we looked at quadriceps, we looked at the distal leg muscles. And in addition, we also looked at brain, spinal cord, lung, and the heart. So there is abundance of this virus being delivered to the muscle, and this is in a non-human primate study. Now, these graphs show you that these viruses are being delivered mostly in the muscle. So the left graph is comparing untreated treatment with low doses, mid doses, and high doses, and the traditional AAV-RH74, which is what Sarepta had used for their gene therapy platform, and this is a high dose. And you can see off the bat, if you look at the golden ones, the AAV-RH74 has extremely high expression in the liver and relatively modest expression in the muscle. Whereas when you use the low, mid, and high dose of the AAV Myo2, you have fairly good expression in the muscle and really no expression in the liver, which is very important on that. There is expression in the heart, but it's something that we will talk about in the next few slides. Now, this is a Western blot. Western blot is looking for protein, and this is protein expression of the FKRP for different muscles, the tibialis anterior, which is your shin muscle, quadriceps, which is your thigh muscle, the heart, and the liver. And one thing off the bat, you can see that there is really no expression in the heart. There's no expression, no expression in the liver. There's no expression in the heart, but there is quite robust expression with the mid dose as well as the high dose in the thigh muscles as well as the leg muscles. And that's very important. Now, in addition, despite all of this, there is improvement in the heart function in the mice study that had been done with these doses. And these graphs will look at different cardiac functions. So stroke volume is how much volume of blood you can pump. Heart rate is generally just how much the heart is draining. And then the cardiac output is also generally the function of the heart and how much volume it can put out. And you can see that there is normalization, or at least getting it closer to a normal with the treatment in these animals. And more importantly, we have done studies to show that there is no toxicity to the heart. So this is a series of experiments that were done looking at the amount of toxicity in the heart, and that's measured by how many macrophages or histiocytes are there. And you can see that generally with treatment, there is no macrophages or histiocytes seen in the heart, suggesting that they have little or no cardiac pathology. And then these are different doses and looking at contraction of the muscle and whether that contraction results in what we call eccentric injury or damage to the muscle. And all of that looks very favorable. There is little in the sense of eccentric injury in the treated animals, whereas when you look at the untreated animals, these muscles, even though they contract, they are very liable, very susceptible to injury. So again, we seem to be able to correct that with the proper gene dosing using the AAV-MYO2 gene therapy. And then final slide for the data that I'm going to show you is the histological data. So this is again on the left, this is wild type. This is the animals who don't have mutation. They have normal looking muscle, both the shin muscle, the thigh muscle, as well as the hamstring muscles. This is untreated animals who have severe muscle disorder. They have muscular dystrophy. They have a lot of degeneration and regeneration of muscle fibers. And then this is now looking at the low, mid, and high doses. And one of the things that we look for, especially as a marker of regeneration, are these internal nuclei, the central nuclei. And off the bat, you can see that compared to the low dose or the mid dose, the high dose really eliminates the number of muscle fibers that have internal nuclei, suggesting that there is improvement or correction of the heart muscle dysfunction. There are currently two different gene therapy trials. There is the AskBio trial, which is currently enrolling. They are taking patients with diagnosis of LGMD R9. They have to have confirmed gene mutation, ages 18 to 65. The main purpose is safety and efficacy. It's a phase 1 and phase 2 study. So they are looking for adverse events and serious adverse events, and if there is a dose-limiting toxicity. It is a randomized study. It's a double-blinded study, which means that patient will be receiving placebo, and it really depends on what the ratio of the placebo to active is. Atamio is a French company, and it's a very limited study in the US. I believe it's only being done in one center currently. It's a follow-up of an earlier French study on that. And again, patients with R9, they are looking at your pulmonary function or lung function as an efficacy measure. Again, it's a phase 1 and 2, so bulk of it is safety. And the phase 1 is open label, whereas the phase 2 is randomized, and there is a placebo. I already talked about the ribitol, which is getting very close to getting FDA approval. The main outcome measure was that North Star assessment, which has now become the standard for limb-girdle muscular dystrophy. And the study that we are proposing, and again, there's still a little work to be done here, but it'll be, again, a phase 1, phase 2 study because we need to make sure that this gene is safe and tolerable. We'll be targeting patients with LGMD R9, demonstrate the tolerability and safety, and mainly collecting information on adverse events, but we will be collecting some efficacy measure. And this will also have to be a randomized double-blinded placebo study. The exact number of patients, exact sites have not been decided yet. So just a few words about Cure Rare Disease. It's a group that is dedicated to developing genetic therapies and bring to treatment life-saving treatments for ultra-rare disease and to transfer the lives of the patients through that. It's a group of academics and individuals with experience in the industry along that are working together to find diseases that are suitable to develop therapeutic options, test them in animals and humans, and then bring them to trial and get them to license or approval through the FDA. So they are doing some cutting edge research and they're targeting rare and ultra rare diseases, including rare diseases that affect a few families at this one. It's a very patient centric approach right now. And this is their pipeline. They have been active in developing treatment. They're getting close to clinical trials as in the LGMD R9 space. And they're also very close to getting clinical trials in spinocerebellar ataxia type 3. But they also are doing diseases that are much rarer, like ADSSL1 or even LGMD2G or R7, which is much rarer than some of the other ones. There's a lot of interest in Duchenne muscular dystrophy as well, especially the somewhat esoteric and exotic mutations that are hard to treat and have generally been ignored by the larger pharmaceutical industry. So I'm going to end here. Thank you for your time.
We'd like to thank all of our presenters in this last session. So in conclusion, I wanted to mention that there are not only scientific and clinical hurdles which need to be overcome for successful drug development for LGMDs, but there are also structural barriers to treatment development, particularly for very rare LGMD subtypes. And to illustrate this, I'm going to give 3 different examples from rare subtypes of LGMD, involving whether repurposing a drug, that's approved for, a different LGMD subtype, or some different rare disease in general, how that can be applied to a different LGMD subtype or a different rare disease area in general. One can also consider, what if someone identifies something that, is a chemical compound, but needs to be turned into a drug for it to be used by patients, or something which can be bought over the counter, say as a natural product, that is not a drug. And as a reminder, what we're looking for is to have good scientific data on whether, this drug or compound actually works, what the correct dosage would be, any safety issues or side effects, and make it available to patients. Okay, so my first example is actually ribitol, which, we've already discussed in the summit. And it has now been applied to the FDA for approval in LGMD2I by BridgeBio, but it turns out it actually is applicable in theory to 2 other forms of LGMD, R13 and R20. Both of these are much rarer than R9 or 2I, but they involve the same chemical pathway. So the basic idea is that by giving a person ribitol, you're giving these enzymes that are less effective than normal due to a mutation more starting material to work with. So that can restore glycosylation of alpha-dystroglycan. So in theory, giving people with R13 or R20 ribitol should work in exactly the same way that it does in R9. However, there's some issues. So let's imagine that ribitol has been approved by the FDA for R9 at some point in the future. People with R13 or R20 can just be prescribed the drug. It's customary to prescribe drugs off-label in the U.S. Okay, so in theory, people with these subtypes could get the drug, but the payers are not likely to support this unless it's a very cheap drug, which unfortunately doesn't tend to be the case for rare disease drugs in general. So, in theory, they can access it, but they might not be able to afford it because the payer is going to say, well, it hasn't been improved for this indication, this particular subtype that you have, so we're only going to support it for it's been approved for by the FDA. It also, if someone just is using it, individually, that doesn't provide us any data, as the LGMD community, whether it actually is effective for that subtype. An alternative pathway would be for the drug developer to seek approval for a second in this case, subtype or subtypes of LGMD. Okay, that's decision will obviously be up to BridgeBio. I don't have any connection with them, so what they do would be their decision. But a couple points. One question is what would actually be needed to approve the drug for these much rarer subtypes where it's unlikely that one would be able to do a trial analogous to what was done in R92I, which is the basis for the current application. It turns out there's also a quirk in current US law which makes rare disease drugs exempt from the possibility of future price negotiation with Medicare or Medicaid, but only if they're approved for just one rare disease. If they get approved for a second one, that exemption no longer applies, which is actually a disincentive in general for any company with a rare disease drug for applying for approval for a second condition. And this would not only apply just to ribitol, it would, for instance, apply to any drug approved for DMD, which might also be applicable to LGMD. Okay, my second example is a compound called N-acetyl-glucosamine. And this is potentially applicable to a couple of other rare types of dystroglycanopathy, R15 and R24. These are caused by mutations in the genes POMGNT1 and POMGNT2. And for both of these, the molecule that they add to the glycosylation chain is N-acetyl-glucosamine. I'll just call it NAG. Now, this can just be purchased online or in a store. It's categorized as a nutraceutical. If someone with one of these subtypes actually takes it, will it actually help? The short answer is we don't know because, to my knowledge, nobody's tested it. The mechanism by which NAG would work for these subtypes is kind of very analogous to the ribitol R9 story, but because it's freely available, there's not much incentive to study since it's already available for a very low price. It can't really be commercialized, so no one has a incentive to set up a formal clinical trial, in addition to the number of patients being very small. So, we don't know whether it actually would work for these conditions. We don't know what the appropriate dosage would be. It may be that, it might not work because one simply can't take enough of this compound to improve the glycosylation significantly. We also don't know whether there's, any safety issues or side effects that one should be concerned about. So, until these questions are answered, it's not really a plausible treatment yet. Okay, the third example is a substance called mevalonolactone, and this is a recently discovered subtype called LGMD R28. This is caused by mutations in the gene HMGCR. What this is part of is called the mevalonate pathway that actually produces cholesterol as well as vitamin D, coenzyme Q10, and several other products. HMGCR is actually the target of statins, so muscle pain and weakness is sometimes a side effect of statins. In addition, so R28 is caused by mutations in the HMGCR gene. and causes LGMD type symptoms. So on the right, we see that the step that is involved in R28, it turns out the step immediately before that in the mevalonate pathway causes a different type of muscular dystrophy. So one treatment approach is to just give the product called mevalonate, which is produced by HMGCR. Well, a derivative of mevalonate called mevalonolactone has actually been used to treat a patient with R28. The problem is it isn't a drug, it's a chemical. The researchers who originally did the study figured out a way to purify it to pharmaceutical grade and then administered it for a brief period of time to a single patient under a compassionate use scenario. But what needs to happen now for it to become generally available is someone needs to synthesize it, confirm the initial result in one patient, and then get some type of regulatory approval so that it can be used by people with this form. Okay, so what we're seeing in all of this is there are fairly straightforward and attractive ideas for treatments, but there are structural barriers. Some of these have to do with the fact that the number of patients identified with these diseases, these subtypes, are very small, but another just has to do with some of the issues with getting either formal approval or repurposing status for different drugs. So with that, I will pass it to Catherine Bryant for some closing remarks.
As we come to the end of the 2026 LGMD Scientific Summit, I want to thank each of you for being a part of today's conversation. Your participation helps move the field forward and strengthens our shared commitment. Thank you to each of our speakers who took the time to present. We've heard powerful updates on clinical trials, thoughtful discussions on policy and advocacy, and inspiring insights into emerging therapies. On behalf of the Speak Foundation, I want to extend our deepest gratitude to our platinum sponsor, BridgeBio Neuromuscular. We also want to thank AskBio for their bronze sponsorship, and thank you both for your continued support to our entire community. While today's summit may be coming to a close, the work continues. We encourage you to stay engaged, stay connected, and continue advocating for progress in LGMD research and care. You can also visit today's content and explore additional educational resources on our dedicated LGMD YouTube channel, where we share ongoing updates and information for the community, and you can watch all previous scientific workshops and conferences. Thank you again for joining us. We look forward to seeing you next year at the International LGMD Conference in Orlando, Florida.